people will join the session?
Well, yesterday we had, at the session that I shared, we had oscillating between 25 and 30, but it really depends on the topic. Well, yesterday we had, at the session that I shared, we had oscillating between 25 and 30, but it really depends on the topic. Well, yesterday we had, at the session that I shared, we had... Oh, okay, it stopped. Okay, since it's 10 a.m. UK time, it's time to start. So as people arrive, I will just start talking. Good morning, everyone. I'm Matteo Mancini from University of Sussex, and today I'm sharing a very interesting session on the brain and the aging process. And we have three very wonderful speakers. And I also have as a back host Dan Wesseling, that I thank for the work that he will do, basically taking care of most of the things that will go on in the back end. So our first speaker is Feng Deng from Trinity College Dublin. And she will talk about how functional networks change through the aging process and how sex influence these changes through the aging process. Feng?
Yeah. So I'm just going to share my screen. Can you see my screen okay? Yeah, so hi everyone, my name is Feng. Today I'm very happy to share this work called Sex Disparity in Effect of Aging on Brain Network Integrity Across Lifespan. So we explored midlife change in brain network segregation because midlife is a critical period for the beginning of age-related neuropathology, which may lead to neurodegenerative disease such as Alzheimer's disease in later life. And it's also a unique disease-altering window. And brain network segregation quantifies functional specialization of different brain networks. And previous studies found that age-related reduction in brain network segregation relates to reductions in structural integrity and cognition. Therefore, it may serve as a proxy for age-related decline in brain health. We also looked at sex effect because 60% of people with Alzheimer's disease are women. So to answer those questions, we used the resting data from my data from a healthy lifespan cohort and divided them into 3 age groups with well-matched sex in each group. And then we computed the modular segregation to explore midlife change and the sex differences. So first of all, we found that network segregation significantly decreased with age, as you can see in the figure A. So y-axis is segregation and the x-axis is age. Here, higher value actually means lower segregation. Sorry about this confusion. And we also found that age-related reduction in the segregation related to reduction in fluid intelligence. and also two structural integrity measures, which are cortical thickness and gray matter volume. We then explored age effect on different brain networks. We predefined 10 brain networks shown in the x-axis in these two figures. And you can see that 6/10 networks showed significant age effect, with frontal-parietal and default mode networks showing the biggest effect. And we also found the age-related effect in those specific brain networks also relates to the reductions in fluid intelligence in a similar magnitude. The order of these networks for the two figures here are the same. And then we looked into the midlife change. So the x-axis is the same order of the networks with three age groups. What you can say is frontal-parietal and default mode and singular opercular networks showed significantly decreased segregation in midlife relative to young age. And in addition, Cillian's network and the mouth part of the motor network showed continuously decreased segregation from midlife towards older stage. And the last bit is the sex effect. In the figure A, I showed the sex effect on different brain networks. You can see that 5 networks showed significant sex effect, and I highlighted those 3 networks showing both age and sex effects, which are auditory, Cillian's, and frontal-parietal networks, so that we can look into sex differences in each individual network, as shown in B, C, D. So specifically in auditory network, we found that female had significantly higher segregation than male, especially in young and older stage. And whereas in frontal-parietal network, female had significantly lower segregation than male, especially in younger stage, which are young and midlife stage. And in Cillian's network, female showed significantly lower segregation than male in much older stage. So overall, this study showed significant reductions in key network segregation from midlife, such as default mode and frontal-parietal networks, which may suggest that those networks may be particularly liable to pathological agent in midlife. In addition, our findings also suggest that sex differences on brain network segregation represent both inherent differences presenting in more younger stage, such as like young and more midlife stage, such as frontal-parietal and auditory network, and also age-related differences on network deterioration, presenting in much older stage from midlife towards to older age, such as Cillian's network. Also, lower segregation in females from midlife in frontal-parietal and Cillian's network may provide an avenue for us to understand the higher incidence of age-related neurodegenerative disorders in females, such as Alzheimer's disease, in the future studies. And that's all I want to present today. Thank you for your listening.
Thank you, Feng. So let's start with questions. I have a first question that is, how do you interpret these sex-specific losses for the auditory network, for the frontal-parietal network, respectively, in men and women? Is there an actual interpretation on why this happens and why it's auditory in men and why it's frontal-parietal in women?
Sorry, my network is just run out a little bit. Can you repeat the question?
Yeah, sure. So since you showed that basically the auditory network is more impaired through aging in women and the frontal-parietal instead is more impaired in men, do you have any interpretation from this? Why this happen?
Yeah, so you mean for the auditory, like auditory network affect actually frontal parietal for the frontal parietal and salience network, females affected more.
Yeah. So why do you think this happens? So is there some sort of hypothesis on some structural difference that is driving this difference?
Yeah, actually, frontal parietal is a very interesting, and also deformal network is a very interesting, two very interesting networks. So previous studies found that males actually are affected more in frontal parietal network, both structurally and functionally. And whereas default modes affect females more. And also there are some structural and functional evidence. In a separate study, we try to explain this effect by looking at a brainstem structure called blue spot, neuroadrenaline, so locus coeruleus. And yeah, so we try to, because a lot of molecular studies found that these sex differences in the neurotransmitter. So we try to use fMRI data to explore the sex differences in a brainstem. And you know that the locus LC connect to the salience and the frontal parietal default mode network in a dense way. So we try to link that. Yeah.
No, it's interesting. So basically the reason may be because of the connections of those networks to more sort of more primitive structures. No, it's very interesting.
Yeah.
So while we wait for other questions from the participants, of course, if the other panelists have questions, go on and ask. But in the meantime, you looked at integration, and I wonder if you would expect some effect also in, sorry, you looked at segregation, and I wonder if you would expect also an effect in terms of integration, so the way the different network interacts, if maybe it balances what happens in segregation, or if maybe is also disrupted.
Yeah, so yeah, that's a very good question. The next thing I want to look at is find some good measures for the integration, because the segregation is a well-established measure, because a lot of studies found the relationship between the segregation with the cognition, for example, fluid intelligence, but not crystallized intelligence. In our study, I also looked at this relationship showed in the figure 1b. The relationship disappeared in a crystallized intelligence, and also it relates to the structural integrities. So the segregation is quite a good measure for us to look at the midlife change and the sex differences. And I will look into the integration, combine this to compensatory measures. Thank you.
Also, it's interesting that in the figure that you just mentioned, the one where you compare with the fluid intelligence, the thing is that you kind of expect that maybe there is also another variable that it's influencing that shape, because it's kind of looks that it may be non-linear, so it may be either something that it's making the figure more complex. Did you use any covariate to correct these results? There were any possible confounding factors that you took into account?
Yeah, so I controlled brain volume, because there is a relationship between the segregation and brain volume, and also brain volume with the fluid intelligence as well. So I controlled that, and I'm not sure that like other confounding variables there. I don't know whether it's worth to explore, adding maybe a secondary item in a model to fit this but it's just the linear model is very straightforward to interpretation.
Yeah, sure. And also the corrective for volume also makes sense because the volumes of the male and the female brain will affect these results otherwise.
Yeah.
So other questions from the participants or the panelists? Don't be shy.
Yeah, I will have a question for you. I was actually wondering, when you say midlife aging, what exactly do you mean? Like, what would you say is the cutoff for the like significant decline in, in brain network integrity?
Yeah, so we actually we want to look at midlife. We look at midlife in the healthy cohort in this study, but we have like another project looking at the midlife change in a risky population to later life dementia, especially.
Mm-hmm.
So midlife, like a lot of researchers start to look at midlife population because one of the Lancet, it's a very nice graph showing the risk factor in different life stages contribute to the late life dementia or neurodegenerative disease in general. Yeah. A lot of factors are in the midlife. So we focused on the midlife change in this study because we try to establish a foundation for us to explore, a more pathological agent in midlife and try to find maybe some markers before the symptoms showing up. If we can find some brain markers, we probably could understand more. First, understand more about the pathological mechanism, or like we can establish intervention markers. So that's kind of like a motivation for this study.
Thank you.
Okay, thank you, Feng, for the very nice talk. And now we will move on to our next speaker.
Okay, thank you.
So our next speaker is Marlene Haupt from University Hospital Jena, and she will tell us about what happens to our ability to stay alert when we are told to as we get old.
Yes, let me share my screen. So thank you. Can you see my screen? Yes, thank you so much for the kind introduction, and hello everyone, and welcome to my talk in which I want to present evidence for an association between the right frontoparietal network and phasic alertness in healthy older participants. So phasic alertness refers to a short-term increase in the brain state of readiness following external warning signals. Such warning signals could either be auditory, like the horn of an ambulance, or visual, like the changing color of a traffic light. In both cases, such warning signals indicate important upcoming situations, so it's beneficial for our brain to prepare for any kind of sensory input. Former studies have shown that these external warning signals speed up reaction times in participants, but in our study, we wanted to ask whether they also, for a short amount of time, increase participants' visual processing speed on a perceptual level, so without any motor response. And to measure that, our participants performed such a verbal whole report in which they saw target displays consisting out of 6 letters. We varied the exposure duration of these target displays, and the participants' task was to simply report all the letters they had seen. In the Q condition, these target displays were preceded by a loud auditory beep, while in the no Q condition, our participants simply fixated. So by using this task to measure phasic alerting effects on visual processing speed, we could find that in general, visual processing speed is reduced in older participants. That is a replication of former studies. But way more interesting to us, we could see that both healthy younger and healthy older participants profit from these auditory warning signals in the sense that they speed up their visual processing. We then related these phasic alerting effects to intrinsic functional connectivity. we performed an independent component analysis and dual regression to analyze the resting state networks we were interested in. In our case, that was the singular opercular network, as well as the right frontoparietal network. We also controlled for other attention relevant and sensory networks, but I won't go into detail here about the control analyses. In the two resting state networks of interest, we then performed a voxel-wise multiple regression. That means that in every single network, we go through the value of each voxel and check whether this voxel value is significantly related to our phasic alerting effect, so the behavior of interest, while controlling for the influence of age, sex, education, and head motion. And by performing this voxel-wise multiple regression, we found the following results. In the younger adults, phasic alertness was associated with the singular opercular network, while in older participants, it was primarily associated with the right frontoparietal network. You can see the networks here as blue overlays on the brain slices, and the red spots, I also highlighted it, are the peaks of these associations. That doesn't mean that these peak structures are the anatomical structures that are especially important for phasic alertness. It simply means that the association between phasic alertness and intrinsic functional connectivity in this network is best represented in this peak. So to summarize, while we found similar behavioral effects in healthy younger and older adults, different intrinsic functional connectivity patterns seem to underlie these findings. In the younger participants, we found the singular opercular network, which is in line with former studies who have shown this network's importance for visual processing speed. And here we are measuring differences in visual processing speed. In the healthy older participants, we found a right frontoparietal network, which is in line with a proposal by Robertson, who proposes that in aging, right frontoparietal structures are especially important for maintaining any kind of attentional functions that are related to the neurotransmitter noradrenaline that had already been mentioned in the previous question session. So this tight link to noradrenaline might also be an explanation why we find these different resting state networks, because a recent study has found that the connectivity between the locus coeruleus as the primary noradrenaline producing structure and different resting state networks also changes in aging, which makes it a potential explanation for our findings. But of course, aging also comes along with other brain changes like changes in gray matter, white matter, neurovasculature. So future studies are needed to find out what is the exact underlying mechanism that leads to these different findings. So with this summary and outlook, I would like to close my short presentation, and I'm looking forward to your questions.
Thank you, Marlene. It was very interesting. So let's start with questions. I would like to start, and my first question is, you showed this very nice separation in young and old participants, that you had this very frontal effect in young ones and a very parietal one in old ones. What would you expect if you could have more participants and divide them in 3 groups? So have a young one, middle age, and old ones. What would you expect in the middle group?
Yeah, that is a really interesting question. In general, there has been a former study that is from Adriana, who I think is even in the audience, my colleague, who looked at the association between visual processing speed, so without this phasic alerting cue, but just visual processing speed over like a whole age spectrum, so younger, middle-aged, and older participants. And she, in general, for visual processing speed, found an association with the singular opercular network. I could imagine, as the middle-aged group would be kind of in between, that it would be maybe especially interesting to look at the coupling between networks, and if that changes and then kind of one network at some point takes over. That would be what I would look at, but I don't have a concrete guess what we would see.
Okay, no, thanks. It was just a curiosity that I had, how this function would migrate through the brain.
Mm-hmm.
So For the participants and the panelists, do you have questions in the meantime? Marion, Feng, any question? No,
I could,
yeah, please go.
No, I could ask a question about why you chose to use an auditory external signal. Would it work with something that was visual, for example, or tactile? I don't know. Or is it something specific about giving, auditory? I don't know.
Yeah, that's a really good question. In general, phasic alerting effects also work with visual and tactile cues, so it's not domain specific. However, former studies have found that auditory alerting effects are usually stronger in older participants than visual alerting effects, while that difference has not been found in the younger ones. So as we were specifically interested in older participants, we kind of wanted to use the strongest cueing modality possible. And that's why we decided for that auditory cue.
Okay, cool. Really interesting.
And we have, go on, Fran.
Can I ask a question?
Yeah.
This is very interesting because I happen to listen to another talk very similar to this one. I wonder, like, for example, you mentioned potential links to the locus coeruleus. I wonder, like, is there any potential measures or like techniques you consider to measure this to test this hypothesis?
So I think one potential measure could be to look at the connectivity between the locus coeruleus as a specific seat and the different resting state networks. And that is also what the study that you see here by Lee et al. From this year does. And they find that the connectivity between the locus coeruleus and the singular opercular network is reduced in aging.
Oh, okay.
So I think that is the way to go ahead. In structural scans, you can also have neuromelanin sensitive sequences that allow you to tackle the locus coeruleus specifically. So I think there are some new MRI methods emerging to really get to these brainstem structures like the LC.
Yeah, and also like I know the pupil diameter is one of the well-established measure for the locus coeruleus activity. Do you think this measure will allow you to explore the causal relationship between the locus coeruleus and the brain cortex?
I think it would be great to combine them, to actually have a behavioral measure, in this case of alerting or any kind of arousal, to then have the pupillometry and maybe even skin conductance or something else. And then, of course, the fMRI data. I think that would give us more of a bigger picture of what is actually happening also at the interplay of tonic and phasic alertness.
Yeah,
I think that would be ideal.
Yeah.
And on a similar line, there is a question from one of the participants, Steven. It's more kind of a technical question. He's asking, currently, is it possible or not to evaluate functional connectivity on locus coeruleus using 3T fMRI?
Yes, I think it is. And I think, for everyone who's interested in that, I would refer them to the Lee 2020 paper. And also, I think in general, there's more and more literature coming up of people looking at the integrity and structure, as well as at the connectivity of the locus coeruleus, and they report the methods they use for pre-processing brainstem signals and all the difficulties that come along with looking at the locus coeruleus. So I think there is a growing literature there, and that opens up new avenues to actually look at locus coeruleus connectivity.
I guess that probably one challenge is when you can have some confounding factors, if it's really the locus coeruleus or if it's another one of the nuclei in the brainstem.
Yeah, so they are using really specific toolboxes that have circumscribed mask of the locus coeruleus. I think one big problem in fMRI is that the brainstem has a lot of vasculature. So you really have to control for all of these different noisy signals that come in your pre-processing as well.
And a very last question for me, and then we move on to the third speaker. You looked at networks that were specific, you hypothesized were specifically related to phasic alertness. Would you expect some concomitant effects in other networks that may be related also indirectly with phasic alertness?
Yeah, so we controlled for the other attentional networks as well as for sensory ones, because we had visual stimuli and the auditory cues. And we didn't find any effects in these networks with our behavioral variable of interest. We also didn't find any inter-network connectivity, even though I personally don't think that that means that there is no inter-network connectivity, but maybe our method doesn't excel at finding this inter-network connectivity, and maybe one would have to look at effective connectivity or other measures, because I'm quite certain that these networks are somehow communicating with each other.
Okay, very interesting. Thanks for the talk, Marlene.
Thank you.
And now we will move to our third and last speaker, that is Marion Durtest, de l'Institut de la Vision. And she will share some insight on why we get worse at navigate the world as we get older. Marion?
Yeah. Okay. Can you see my screen fine? Okay, so hi everyone. First of all, I would like to thank the organizers again for this really great conference. So my name is Marion and I'm a PhD student at the Vision Institute in Paris. And the work I'm going to present to you today is on age-related differences in regions of the brain that deal with visuospatial processing during spatial navigation. So just very briefly, kind of an overview of what is spatial navigation. It refers to this ability we all have to plan a path to a goal and then travel to that specific goal. And it's now really well documented that in healthy aging, there are prominent navigational deficits, and these can dramatically reduce older adult autonomy and mobility, as well as impact their quality of life and also accelerate cognitive decline. So this is really a major societal issue that really needs to be tackled. At the neural level, these navigation abilities are subtended by quite vast networks. So we have the hippocampus and its associated structures, but we also have more distal areas like the frontal, parietal, and occipital cortices. So I've highlighted also in orange 3 small regions that we call scene selective regions. So they respond preferentially to the presentation of scenes, and they're really interesting because they're situated kind of right at the junction between our visual system and higher cognitive structures. Now, what is quite remarkable is that in healthy aging, this network seems to be specifically impacted. So on the top right, you can see some hyperactivity in the hippocampus and associated mediotemporal structures. And we also have on the bottom right area this kind of strong frontotemporal pattern of atrophy. So what have these changes been attributed to? In the literature, largely they have been attributed to functional changes in strategic function, working memory, associative learning. However, the contribution of perceptual function has largely been overlooked, and this is actually quite surprising because we know that there is prominent sensory loss in healthy aging, and we also know that the visual environment is particularly key to how you navigate space. So this is kind of the overarching aim that we have in the lab, is that we want to understand how visual aging in kind of all its form contributes to spatial navigation deficits of older adults. So I really want to make it super clear that visual information is really key to how we process and navigate our space. And if you take a look at this picture here, we have what we would call landmarks, which are salient visual information that you use to anchor your behavior to in order to find your way in space. So in this case, it would be the window or the ATM. So this figure actually comes from a recent paper from the lab, and in real-world navigation, they found that landmark cues were significantly altered in healthy older adults. And this is kind of what we based our study on, and we wanted to go and take a look at the neural basis of landmark-based navigation in healthy older adults, and we were specifically interested in what happened in the 3 scene selective regions that I mentioned previously. So how did we do this? Well, we conducted an fMRI experiment. So in the scanner, we had 25 young subjects and 21 older subjects perform a virtual navigation task. So there were two conditions. There was a landmark condition where participants had to find and then learn the location of a goal that was hidden. And then they needed to retrieve that goal from different starting positions in the maze. And they could only achieve this correctly if they had encoded and they used the landmarks that were present in the center of the maze. So you can see them on the photograph in the top right. These were 3 discrete objects. And then we also added a control condition so that we could account for potential confounding factors like motor aspects of the task or very simple perceptual aspects. So I would now like to go over our results very briefly. This is just kind of overview. So one of the interesting results that we got is that when we contrasted the landmark to the control condition, we found that young adults had significantly more activation of a region of the anterior temporal cortex, which is mostly the anterior temporal pole. So that was quite interesting. And then we also found that in these scene selective regions, there is only a difference between young and older adults for the occipital place area. And it was actually increased activation in older adults, which was actually quite interesting and had never been found before. So how can we kind of interpret these findings? Well, first of all, this kind of higher activation of the anterior temporal cortex in young adults is really interesting because this region is particularly involved in visual object recognition and processing. And we could kind of speculate that changes in its activity in older adults could modify the way they use visual information and process kind of visual space, which could therefore hinder their spatial navigation abilities. And finally, this kind of second finding that we had was of this enhanced occipital place area activation in older adults. And this was in line with another study from the lab that had found that functional connectivity around that region was actually preserved in healthy older adults. So in our opinion, this is kind of the strong point of this study. It's really interesting, and it's something that we want to go and find out a bit more about. There are different hypotheses as to why it could be enhanced, so we could discuss that potentially. But overall, I just want to say that there are prominent age-related differences in the activity of regions in the brain that deal with kind of higher level visual function, and that these could contribute to spatial navigation deficits. And we want to go and look and find out a bit more about this. Thank you very much for listening. And I would like to thank everyone in my lab who worked on this with me. Thank you.
Thank you, Marion. It was a very interesting talk. I have a question about the experimental design. So in the training part of the experiment, where the subject will understand where their goal is, were they allowed to start from each possible path, or it was just one?
So when they trained to locate the goal, they always started from the same position. So basically, it was a task of reorientation. So they needed to have encode the kind of disposition of the landmarks in the middle with a goal. Like, for example, they remembered that the goal was situated between the square and the circle. And then in the retrieval phase, we started them from different positions. So either the same corridor or the other corridor. It's just a 3-iron maze, so not so complicated. But then, so yeah, we started them from the other corridor. And they needed to figure out that their position had changed, that the goal was from a different angle. Yeah.
Would you expect that, of course, not this one because it's a very simplified maze, but in a slightly more complicated one, would you expect a difference in performances if the subjects will start from the same place rather than starting from different places?
What do you mean?
So basically, when you go to a place and you start walking, if you do, let's say, a street always in the same direction, you will become very familiar with it. But then if you go the other way, it will look different. And so maybe you will perform worse.
Yeah, definitely. Well, that's actually what we kind of find here, because it's very difficult for older adults to reposition themselves once they have been kind of disoriented. So this is something that we see a lot in the literature, where older adults struggle a lot. And as you said, it's exactly that, is they become familiar with one orientation we call this kind of response learning. They just learn that they need to turn right, and then they turn right and right. And this kind of becomes a habitual behavior. So then when you change their position, they're completely disoriented and will just turn right again. And yeah, in simple mazes like this, it kind of works because it's very easy, and that was the point, to make behavior equivalent. But in more difficult mazes, then for sure you observe this behavior.
Because I assume that in more difficult ones, it will also happen for all subjects, that even if they do the same way, at some point will get lost.
Yes, for sure.
Okay, and we have one question from the participants. This is from Hugo Spears. Are there any differences in how older adults try to strategically solve the task? For example, verbal coding?
So that's a really good question. So actually, we had this questionnaire given to participants at the end of the experiment. So it's not the most robust questionnaire, but it gave us information as to how they used the landmarks to orient. And basically, we did manage to classify participants into if they had used a place response, we call in the navigation terms, and response kind of place learning strategy or response learning strategy, which means basically, do they do this thing where they just turn right using one landmark, or if they use kind of the configuration of landmarks to orient. And basically, it was very similar to what is apparent in the literature, where older adults use a very response-based strategy. They turn right at a landmark. They use just one landmark. This was 85% of our older participants, whereas for young adults, it was pretty much equivalent. You had half that had used the strategy and half that used a more place strategy, where they encode the configuration of landmarks. This was really, really evident from our data. Yeah. So response learning in older adults. Yeah.
And we just got two other questions. The first one is, I think, from Marlene's colleague, Adriana Ruiz-Rizzo, and is, did the activation of the opercular of the OPA or any of the other scene relevant regions relate to performance in older adults?
So really good question. So actually that's something that is a bit difficult to test with I'm just going to get this out, with the region of interest. So this, the scene selective region's activity did not correlate directly to navigational performance, but we need to say that the navigational performance, we made quite equivalent between participants. It was very similar. As I said, it was a really simple maze, and usually participants, well, older participants struggle much more in kind of a typical navigation paradigms, but here we really wanted the behavior to be equivalent. So, I mean, it's a bit difficult to say that these regions correlate with spatial navigation, but also because, as I said, there are scene selective regions, so they're kind of high-level visual areas. So not necessarily directly correlated with spatial navigation, but more with how information in the maze, visual information, is used and processed. So yeah, so that's not something that we found directly, no.
And the other question is from Christoph, and is, would you expect different results when the landmarks are easier to distinguish? For example, using color or very different objects?
So we actually did this experiment in young adults only, and the behavior is strictly the same. The brain activity is a bit different. So that's currently something that we're working on, but probably related to lower level processing. So definitely the saliency influences in young adults. It doesn't really change the behavior though, it changes the neural activity. In older adults, that's a really good question. So we actually had a second part of this experiment where participants were using geometry instead of objects. And geometry was impossible for them to perceive. We had to abandon that task. It was way, way too difficult. Which was a really interesting finding in itself, that we want to kind of go and explore, but the data was just unanalyzable. So, but regarding the saliency of landmarks, I would say that in this study, it was already quite salient. I mean, they all managed to do the task fine. I would suggest that if the saliency was increased, the color was present for older adults, so the behavior would still be the same. They would still use some kind of response learning. Would it change that so much? Potentially, the brain activity would be modified, and we have kind of this hypothesis about why the occipital place area activation can be increased. And we think it could be related to more like retinal information, so very low level information related to position of the cues in your visual field and things like that. So definitely there's something to go and investigate there.
To follow up, since geometry was very difficult for older subjects, what feature would you expect that for them would be easier to cope with?
Well, that's something that we're actually trying to understand, is what low-level information, so very basic visual properties of visual information, can help older adults orient. So this is something we're currently trying to understand. But I would like to point out that they didn't manage in our task, but in the real-world navigation task that we did in the lab, they actually preferred geometry. So this is something we're kind of puzzled about right now. It's very strange that in the MRI, they don't like geometry, but so there's something there to explore. I think it could be related to many different things, but definitely just increasing the saliency, I was said before, of cues, so making them bigger or enhancing the color. And we're kind of exploring how potentially where you position them, you position them in your upper visual field or lower, could that have an influence on how older adults orient? So all these questions are kind of the main goal of my thesis. So stay tuned for more.
Probably just to conclude here, probably it will be also a matter of familiarity. So maybe, yeah, and then it becomes very hard to study this at the group level, because what is familiar for someone, it won't
Actually,
be.
there have been some studies where they ask before participants to rate the cues according to how familiar they are to them. And familiarity helps 100%. If you're familiar with certain objects, and for sure us, it was completely abstract landmarks, so not the best, like nothing to be familiar about, but definitely familiarity helps.
Okay, very interesting. So, and with this, I think it's perfect timing. We don't have any more questions. And I would like to thank all the speakers and also my back host, Dan Vesseling, for working in the background. And I want to thank the participant for attending this session, and take a break and join the next sessions.
Very much.
Thank you.
Bye.
Bye.
Okay, we're broadcasting.
So hello everyone, welcome to this series of talks with theme computational modeling and techniques. My name is Ellie and I'm the MC for this talk. We have 3 speakers following the traditional talk format of 12-minute talk and then 3-minute Q&A. Please post your questions in the Q&A panel, ask anything if you like. If we don't have time to answer all the questions, I'm sure the speakers will be around to answer them just in writing in the Q&A. So don't be shy to ask questions, even if you think there may not be time for them. Also, there's no stupid questions. You feel free to ask what you want to know, and I'm sure the speakers will really happily explain things to you. With that, we're ready for the first speaker, which is Mikkel Lepperød from the University of Oslo. And Mikkel will be talking about inferring causal transmission probability from pairwise recordings and optogenetics. Mikkel, the stage is yours.
Okay, thank you. So I'm very happy to present this project where we try to infer causal transmission probability from pairwise recording and optogenetics. And this is a collaboration between Simpla here at the UIO and the Coding Lab at UPenn. I'm Mikkel Lepperød, and the principal investigator of this is Conrad Cording. So many neural networks in the brain seems to have like specialized network motifs that define the function of neural populations. And in order to understand how the brain works at the mechanistic level, it's necessary to map out these connections. And typical methods to find connections between neurons is to simply do in vitro, cut out the part of the brain and study the axons and dendrites under a microscope. But this can only tell if neurons are connected or not the strength of connections. Moreover, some cell types are functional identifiers such as place cells and grid cells, which calls for some method to identify strength of connections from activity. And by measuring spikes, a traditional way of determining how strongly two neurons is connected is by counting how many times one neuron fires just after the other, computing the cross-correlation histogram. And here the strength of connection is typically found by comparing the peak spike count against some baseline measure. However, this is a measure of correlation, and correlation does not imply causation. Or in other words, that neuron A very often fires just before neuron C does not imply that neuron A is actually causing neuron C to fire. Or in other words, there's a pink elephant in the room when using naive correlation-based method to determine connectivity. And to conclude a physical connection between neurons from correlation-based method would be similar to conclude that, for example, US spending on science, space, and technology caused people to take suicide by hanging, strangulation, and suffocation, which is of course not true. Even though they are highly correlated, as we see here, every year that US spend more money on science, more people hang themselves. So how can it happen that two neurons spikes are correlated but not connected? So if, for example, neuron A here in this picture is connected to neuron C while B is not. And if A and B receive the same input, they are correlated. And then B and C must also be correlated. And here we call this background activity denoted U a confounder. If you want to make sure that two neurons are connected, we can stimulate neuron A, since we then have experimental control over A, or do A as is typically used as an operator that we can control one variable. And we have the ability to perform randomized controlled trials, or RCT. And if only spikes from the stimuli are taken into account, the correlation implies causation. Similarly, if you take experimental control over B, C would not follow the perturbations in B, and there would be no correlation when only looking at the spikes induced by the stimulation. However, this kind of procedure can be highly technically challenging to do in vivo with, for example, patch clamp or two-photon stimulation. And in order to increase the number of neurons in a similar procedure, we want to use optoconnectics. However, then we also introduce a problem where multiple neurons are stimulated simultaneously, which again would introduce spurious correlations. So if we raster plot spikes from A, B, and C for each stimuli of the optogenetic stimuli, we see that the stimuli introduces a problem where AC and BC scenario seems almost equally correlated. So how can we distinguish AC and BC from these two scenarios? So in econometrics, they have a similar problems with confounding, which can sometimes be solved if you find an instrumental variable that emulate RCT if it fulfills some certain criterions. So to emulate RCT or do A, an instrument set must not be correlated with the confounder and have to be correlated with the presynaptic activity A, and only indirectly interact with the target C through the presynaptic activity A. And so if we set up a linear relationship between C and A here, then the strength of connection beta can be found as the ratio of covariances, and this is the instrumental variable technique between the instrument and C and the instrument set and A. So when a neuron fires a spike, it enters a refractory period in which it cannot produce a new spike. And when we stimulate during this period, the neuron does not respond to the stimulus. And this introduces random times which we can compare the activity of a downstream neuron, in this case C, to when the stimulus actually produced a spike in upstream neurons B and A, or A. So by simulating these three neurons, we can estimate the response in C when activating A and B with optogenetics simulated by current pulses. So first we compute the response of C when we successfully activated A or B, denoted here C given A equals 1 and set equals 0. So when set equals 0, this notes stimuli trials where there were no spike just before stimuli onset. And with this curve, we can also find C's response window. And then we can subtract the activity of C from when the stimulus in general did not activate A or B to get the ordinary least square estimate. But in order to get the IV estimate, we have to subtract the activity of C from when the stimulus did not activate A and B because of the refractory period. And this is the instrumental variable estimate. And you denote here set equals 1. That means it just fires a spike just before the stimuli. So by simulating these 3 neurons in multiple scenarios where we change the true connection weight, we can compare 3 different methods. So the classical cross-correlation based method called p-trans, and the ordinary least square method denoted beta hat, and the IV method. So when looking at the AC estimate here, see that the 3 methods largely agree and correctly estimates the strength of connections. However, when looking at BC, which are not connected, only the IV method seems to come with a causally correct result. So this shows that the method we developed using IV outperforms the classically used methods, and that those are not to be trusted in situations with confounders, which arguably can be abundant in the brain. So to assess how these methods performed in larger network, we simulated a randomly connected excitatory and inhibitory network of total 4,000 neurons with 80% excitatory neurons, which had synaptic weights log normally distributed. From the excitatory population, we selected 100 neurons, which we gave 1 million current pulses to simulate optogenetic stimuli with about 200 millisecond intervals to allow the network to settle back into its asynchronous irregular state between each stimuli. And to test the inference methods, we selected 2,000 outgoing connections and compared transmission probability against true weights. So the first result is that the cross-correlation based method, which compares the peaks by count against baseline, was heavily influenced, or denoted p-trans, was heavily influenced by the stimuli, as we would expect since the stimuli confounds the network. When comparing IV with the ordinary least squares, or OLS, we found that the IV has larger variance, but largely agrees with OLS. And this is expected that the IV has more variance, as it requires, it has much fewer trials to correct for confounding, as it only uses trials where the stimuli hits the refractory period. And it's also expected that the OLS should perform well in this scenario, as there are no other confounders than the stimuli. But when looking at the pairs which have no connection, it seems that IV outperforms OLS, as extremas are smaller. And this slide didn't load. Okay, I never had this problem before. Okay. Okay, sorry. There should be a figure here where the error of the IV method goes towards 0 and outperforms the OLS, but it requires very many trials. So here it requires almost 1 million current pulses. So yeah, and the error is computed by taking the minimum function of a scalar and looking at the norm, the L2 norm of the subtracted, or the difference between the true weights and the estimated weights. To conclude, confounding matters in neuroscience, especially during optogenetic stimulation, and the interaction between stimulus and the refractory state may be used as an instrumental variable to estimate the causal transmission probability. But this is like, it's not a completely finished project, and the problem we are currently kind of looking at is in which scenarios the IV assumptions are fulfilled, and if there are experimental techniques that can help ensure these scenarios and possibly increase the rate of convergence. So with that, I want to thank the Conrad, which has came up with the ID, and my collaborators Tristan Stubber and my local PIs here in Oslo, Marianne and Torke. Thank you.
Thank you very much, Mikael. We have some time for questions, so do pop them in the Q&A. You may also put your virtual hand up and we can allow you to talk and ask your questions that way as well. Obviously, fellow panelists, you're also invited to ask questions if you have any. Yeah, please go for it.
Mikael, very cool. I was wondering, so in addition to using this method as like a tool for observing or estimating the connectivity in the brain, is there any knock-on implication for how we think the brain works? So if you're like a downstream circuit getting a bunch of spikes from another region, should they be using these ideas from causal inference to help them infer, like, I don't know, some external variable or something?
And so you're thinking more at the larger scale, like two brain areas communicating, basically.
Yeah, if it's the V1 interpreting signals from the thalamus or something like that.
Exactly. Yeah, I guess one way to use this method then would be to kind of stimulate the projecting neurons and recording the kind of the receiving end.
Oh, sorry. Sorry to clarify. I meant separate to like us as neuroscientists. Could we take any insights from this for thinking about how the brain works?
Okay, in terms of behavior and stuff like that, I guess.
More like, how could should a neuron in V1 be using ideas from causal inference to interpret activity from the thalamus?
Oh yeah. I guess the closest is Conrad has explored this question a little bit, with that neurons can do kind of this regression discontinuity design, where it kind of tries to estimate its causal impact on downstream neurons. And yeah, so yeah, there's a paper about that which Conrad has out on bioRxiv. I can't remember exactly the title of it, but you'll probably find it if you. But yeah, that's a really cool idea, because for a neuron that sits in the brain, it's not so easy for the neuron to understand or to know if its activity is kind of being read out, right? So yeah.
Absolutely. Yeah. Okay. Thank you.
So
Thank you, Mikael. If it comes to you, feel free to share that paper later on in the chat. We have one more question from Rishika in the Q&A, and I'm not sure if they're still here. I can't find them in the participant list right now. But the question is, when is the IV case more useful than OLS, given that the inference ability was quite similar?
Yeah, so that's really the question we want to figure out, and it's not so easy because so I mean, the best would be if we could like have a experimental setting where we could really have full control over some slice or part of the brain, but that's really difficult experimentally. And then when we're simulating these large networks, it's really hard to ask the question if the kind of results we see is because of, for example, too few neurons, too little complexity in the neurons, there's no homeostatic kind of activities going on. So it's a little bit hard to induce a problem to see where kind of how well the method works, and then say that or claim that this is actually a biological case. So that's really the problem, in like finding a scenario where one method outperforms the other, and that's basically what we're trying to find now. We're really cool to find like this one case where this IV method like really outperforms OLS, but yeah, that's I'm not entirely sure how that would look like.
Thank you. There's one more question in the Q&A, and Michael, I invite you to type an answer to that because we do have to move on to our next speaker. Thank you again, Michael, for your really nice talk.
Thank you.
Next up is Kian O'Donnell from the University of Bristol, and he'll be talking on probabilistic estimation of correlation matrices from binary spiking data.
Great, thank you. All right, shall I just start? Yeah. Okay, great, thank you. So yeah, I'm based in the University of Bristol, and I'm going to talk about a method for estimating uncertainty around correlation matrices, and particularly from binary data, and I can explain why only binary data later. So first of all, just to motivate, so why should we care about correlations, or why does anybody care about correlations? One answer is that these days, neuroscience has entered the big data era. So for example, here's a sort of raster plot from Carson Stringer and Mary Castariu when they were in Ken Harris's lab in UCL. Where they've done 2-photon calcium imaging from 10,000 neurons simultaneously in V1 of a mouse as it was looking at some visual stimuli. So if you stare at this for a while, you can see some interesting activity patterns, but then what do you do after that? What do you go from there? And a common step is to, well, we'd like to understand these, and a common step is to compute the pairwise correlations between each individual pair of neurons, and then study that thing. So for example, you could try and do PCA on it, or ask how the correlations change with stimulus condition, things like that. So they're just like, I see them as a window into what neural circuits are doing and give us some clue about the neural code. Hopefully that we can relate to some theories, so some theoretical modeling about what the parts of the brain are doing. So what is a correlation, or what do correlation matrices look like? Here's an example. This is from the same data set that they've released online. So it's just...
We only see your title slide at the moment. I'm not exactly sure if your screen sharing is working correctly.
Oh, sorry about that. Maybe I need to... Let me try and just share my desktop. That's weird because it was advancing for me. Okay, let's try this. Does that work now?
Yes, much better.
Great, okay. Then I'll just quickly show what I was talking about. This is the 10,000 neurons raster. Thank you for the clarification, Eleanor. I would have just kept talking. 10,000 neurons from mouse V1. So you can see some interesting structure here, but often people will measure the correlations as the object of study to try and get some insight into what's going on, because just staring at this is not going to get you too far. And here's an example of a correlation matrix computed from that data that they've released online. And it's just 40 neurons in this case, so random 40 neuron subset. And typically, this is pretty typical of what you get if you measure correlation matrices in most brain regions, where with calcium imaging or electrical recordings, you see that a lot of the values are close to zero, some positive, some negative. On average, at least within a local region, neurons tend to be slightly positively correlated on average. So the mean is probably like 2 or 5%, something like that. But if you squint at these things, you see some structure in there, and we try and interpret that structure. So what's the issue? So people do this all day, every day. Why do I think it's a problem, or what do I think the issues are? So given a sample correlation matrix, kind of open or issues that people don't often worry about that I think are important are, we might want to ask a question like, is any given element of that matrix statistically different from zero? So this, if we compare the correlation between neuron 16 and neuron 8 or whatever that's in this element, is that number, like if it was 0.03, is that statistically different from zero or not? That's one question we might want to ask. And people often do ask that as a kind of way of thresholding these correlation matrices, or as in the last talk, we saw examples of people try to use them to infer connectivity between neurons. We might want to ask, take two correlation matrices from two different conditions or two different ages of an animal or something, and ask if they're statistically different, or what's the distance between them. And then there's more technical questions we can get with downstream analyses. If we're in the situation where the number of neurons, this is the wrong way around. If the number of data points is less than the number of neurons that we've recorded from, we get a correlation matrix becomes a low rank. And that could mess up downstream analyses, like for interpreting the output of PCA or something like that. And that's getting to be an issue now. So for example, those 10,000 neurons, I think they were recorded at about 2 hertz. So you would, every hour, you would get about, I'm going to get this right, 7,000-ish samples in an hour. So if you're recording continuously for 1 hour and you're 10,000 neurons, you still wouldn't be at the point where you had more samples than numbers of neurons. So that's a problem we'd like to fix. So my answer to this, and not just mine, but an answer to this, is to treat it as a probabilistic problem where we put uncertainty on all the numbers in the correlation matrix, and then use that to address these questions, or fix these questions maybe. So going through the basic idea, first of all, I'm going to assume that the spiking data is binary. And if you had calcium imaging data, you could get that by thresholding the fluorescence values. So everything above some value is 1, everything below is some 0. That's not uncommon. Or it might be the output of a spike inference algorithm that people apply to the calcium imaging data. For electrophysiology, a common way to generate binary data from spiking is to discretize time and then ask if the neurons spiked or didn't spike. And usually people take very small time bins, like 10 milliseconds, where the likelihood of 2 spikes is small. So most of the time they see 0, 0, 0, occasionally see a spike. So if we get in that situation where we've got a binary spike train, then if we think of 2 neurons here and time along this x-axis, this is when neuron 1 spiked, neuron 2 spiked, they both spiked this time bin, and so on. Then the trick is going to be convert that to some sort of binary sequence. So there's a 1 where there's a spike and a 0 where there's no spike. So at every time point, we've got a pair of values, 1, 0, 0, 0, 1, 0, or 0, 1, 0, 0, and so on. And now if we think for 2 neurons, if they're both binary, then there's only 4 possible configurations, 0, 0, 1, 0, 0, 1, and 1, 1. So what we can do is count how many times we observe each of those patterns, those binary patterns. And in this case, we've got 29 0, 0s, 6 1 0s, 3 and 2 of the other 2 patterns. And this is, again, pretty typical. A lot of the time neurons are silent. So we often see more zeros than that. Okay, and then after that, if we assume that the data, now this is where we make some assumptions. We're going to treat this as thinking about each of these binary pair of binary numbers as a pattern. And like I said, there's 4 possible patterns. So we think of those as samples from a discrete distribution with 4 states. Then the likelihood of observing any set of counts, like the ones we've just figured out here, is given by the multinomial distribution. So that's just the probability of observing that pattern. And the k's are the counts, so that's going to be our data. And the parameters of this distribution are the probabilities of the 4 numbers. So the sort of true probability of observing each of the 4 patterns. And in this case, that distribution has just 3 free parameters because we know that the sum of these 4 probabilities has to add up to 1. So it gives us a constraint. If we know that, and that, we can figure out the final one just by subtracting them from 1. So after that, if we assume some prior probability distribution, this is following a Bayesian model, prior probability distribution over these 4 probabilities here. And we can get the posterior distribution over the 4 probabilities given the observed counts. And the trick now is we don't really care about these directly, usually. What we're more interested in is the pairwise correlation between the pair and the 2 individual neuron firing rates or firing probabilities on average. And in this case, there's a unique mapping from any possible configuration of these 4 probabilities to the sort of marginal probability of neuron 1 firing, or the firing rate of neuron 1, the firing rate of neuron 2, and the pairwise correlation between the two. So we can just sort of directly write that down given these 4 numbers. And that means that if we if we have a method for sampling from this posterior distribution, which is fairly straightforward using Markov chain Monte Carlo methods, then we can transform those samples and get samples from the sort of marginal distributions for each of these 3 things. So now we can get joint sort of samples from the joint posterior over these 3 things. And that's kind of what we're after. So how does that work? If I simulated some pair of binary neurons, and imagine they just collected one second of data, 1 minute of data, 1 hour of data in 10 millisecond bins, and then asked what was the posterior distribution over the pairwise correlation in each case. And the true value I had set to, I think, 5%. You can see that dashed line here. If we only record for 1 second, the posterior distribution over the correlation is very broad, as we might expect, reflecting our big uncertainty. It could have been that data came from an underlying correlation of 0.4 or even minus 0.1, something like that. We're not sure. We have 1 minute of data that gets a lot tighter, converges on the true value, but you'll notice even the peak is not aligned with the true value, so there's some sort of random error in that case. But with loads and loads of data, like 1 hour of data, then the posterior distribution converges to like a narrow spike on the true value. And then the nice thing, again, we can not only get these sort of probability distribution over the correlation, but because we have the joint samples from all 3 parameters, we can plot them here. So we've got these 3 marginal distributions for each parameter, but we also got, here I've plotted kind of a heat map of the samples for each pair of parameters. So the correlation and firing rate of neuron 1, correlation firing rate of neuron 2, and the firing rate of both neurons together. So that's great. So now we can do things like ask if that correlation is statistically different from 0. So for example, thank you, how much of that mass is a certain distance away from 0 or crosses 0. In the red case, there might be a little tail that crosses 0. In the green case, we're almost certain it's not 0. In the blue case, we don't know, but we can put a number on that. So it's a fairly systematic way to assess that. So what do we do with more neurons? We have to, the issue is we can't sample from the true underlying distribution like we did in the 2 neuron case, because the number of possible states scales exponentially with the number of neurons. So we've got 100 neurons, we've got 2 to the power of 100 possible states, and that's just computationally intractable. So the trick I'm going to propose, and just almost finish on that, is to approximate that full posterior distribution with a multivariate Gaussian. So we assume there's some latent multivariate Gaussian distribution. Okay, I'll finish now, thank you. Some latent multivariate Gaussian that we're trying to estimate all these parameters over given the data. And so here's one example where simulated data from 10 neurons and drew samples from the posterior. And the nice thing is there's relationships between each pair of values in this that reflect their joint dependencies in the posterior distribution. So again, we can use that to ask questions like, are these 2 correlation matrices different from each other? So finishing, there's still some issues I need to solve, which are, it doesn't scale well. Need to figure out how to solve that. I need to come up with a way to guarantee the samples are from valid correlation matrices and how to handle the prior in a principled way. Thanks, and sorry for running over time.
Thank you very much. Kian, I'll leave it straight to the questions. We have a question already in the Q&A. Sean, I've allowed for you to talk, so you could ask your question live if you want to.
Hi. I think you may have somewhat mentioned it actually in your final slide, but I was just wondering about the prior that you use for the Bayesian analysis.
Well, I think in the example I used a flat prior over those 4 states, but sure. The tricky thing is we'd like to maybe set priors on the correlations directly, and then for the sampling we'd have to sort of back transform those to ask what prior they imply over the underlying states. So that's not too hard in the 2 neuron case. For the full correlation matrix, it's tricky to know what to take, but there are some standard prior distributions over covariance matrices that we could use, but I haven't coded up, I should say.
Yeah, great.
Thank you very much. The next question is from John. John, I've also unmuted you. You could ask your question now.
I was just wondering, you're assuming that the spikes are perfectly aligned, but there can be a delay or some variability in that. How would you include that in your model?
That's a good question. It's not in the model at the moment. So if you shuffled all the time points here, you would get the exact same results. It's ignoring time. And that's not uncommon for most types of analysis, including like when people do PCA. Maybe you could have a model that knows about time dependencies. So some sort of Markov underlying model for the dynamics of the spike trains, but I haven't considered that yet. But it's very difficult. It just explodes the state space even more.
Yeah, no, I could imagine.
Thank you very much. We are having loads of questions come in. We have time for one more brief answer. This is from Mark. I'm just allowing you to talk as well. Brief answer, please, and the rest, Kian, please answer in the chat later or in the Q&A function.
Thank you. So, Kian, I was pleased that you were trying to address the realistic case where you have many neurons. We often look for some sort of multivariate structure in the form of eigenvectors. How close are your eigenvectors from your multivariate model to the underlying true values?
Oh, that's a good question. I mean, in the limit of infinite data, it's going to converge. We know that's true. And all I'm doing here is matching sort of moments of that final distribution. And your question is like, how good a model is the multivariate Gaussian? And that's just going to depend on the data. I don't know. It depends on the type of structure we get in the data. If there's higher order correlations between triplets and quadruplets of neurons, then it's probably going to start becoming a bad approximation. Mm-hmm. Yeah. Or maybe we can fix that in the transform as well between the multivariate and the final covariance matrix. But that's a problem I haven't really figured out yet. Thank you.
Okay.
Thank you very much, Kian. Thank you again for your talk and answering questions. Please stick around and answer questions in the Q&A. And with that, we have one more speaker for the session, which is François Chinotti from the University of Nottingham. François is going to be talking about a Bayesian map of the striatal microcircuitry reveals different wiring processes for D1 and D2 spiny projection neurons. Go ahead, please.
Thank you. First off, I'd like to thank the organizers of this conference. It's great pleasure to be able to present this work, which I've done under the supervision of Mark Humphreys at the University of Nottingham, as just mentioned. So we are interested in a part of the brain which is called the striatum. It's known as the main input nucleus of the basal ganglia. And the basal ganglia is of particular interest because it is supposedly implementing a form of decision-making or at least action selection. The striatum in particular is made up in its great majority of two kinds of spiny projection neurons. So these are neurons which project outside beyond the striatum towards the globus pallidus in particular. And they fall into two different categories. You've got the indirect spiny projection neurons and the direct spiny projection neurons based on their long distance targets. They're also known as D1 and D2 neurons, which is how I will refer to them throughout the rest of the talk, based on the expression of the type of dopamine receptor which they have in their membrane. The classical view of the basal ganglia is that it is a feed-forward circuit in which the cortex stimulates different channels in the striatum. So these different channels correspond to different behaviors or actions. And the role of the striatum is to relay these inputs in two different streams, the indirect and direct streams, towards the globus pallidus. And this means there is any potential collateral connections between the neurons is completely ignored. So we only assume that the output activity is a reflection of the input activity, and any internal computation within the striatum is ignored. And fortunately for computational scientists who would like to look into the computational properties of the striatum, there are experimental procedures which have been carried out to look at the rates of connections between spiny projection neurons. And here you have a picture from the study by Plenaire 2010, where they labeled fluorescently to either the D1 or the D2 neurons. They then patch different fluorescent or non-fluorescent neurons, stimulate potential presynaptic neuron, and see if they can recall the response in the postsynaptic neuron. When they do, they say, okay, these two neurons are connected. They can then report their results in the form of the rates of success of their tests. So if you look at the D1-D1 connections in the study by Taverna, you see that in 15% of pairs, they found a connection. And in the case of D1-D2 neurons, it was only about 7%, so half as much. Based on this finding, you have work by Burke and colleagues 2017, who built a simple firing rate model of the stratum in which they looked at two different subpopulations corresponding to two different behaviors which were being encoded in the stratum, each divided into the direct and indirect pathways. And they assumed an architecture which reflected these discrepancies between direct connections, which are more numerous between direct spinal conduction neurons, and between direct spinal conduction neurons and indirect spinal conduction neurons. And thanks to this architecture, when you have equal cortical inputs, you can find a pattern of behavior which is interesting insofar as even though the inputs are equal, you'll still manage to find a winner. Unfortunately, if the experimenters had taken the trouble of including confidence intervals, you see that the probability of connection between D1-D1 and D1-D2 pairs is actually almost certainly the same. And so the question which we want to address is finding a way of correcting this oversight by exploiting as much as possible the experimental data which is given to us to determine the properties of connection. I'm going to adopt a Bayesian inference framework. So it differs from a frequentist inference, insofar as the frequentist inference framework gives us only a single point estimate. So you had the height of these bar plots, which I showed before, which is p hat, hopefully, although it wasn't the case, surrounded by a flat confidence interval. In addition, the single point estimate is usually the maximum likelihood estimate, which is kind of thinking of things backwards, because it takes the data to be the random variable and the p which you're looking for as the variable, which is already fixed. Bayesian inference, on the other hand, instead of giving us a single point estimate, derives a full probability posterior distribution, FP of p, which tells us how likely every possible value of p between 0 and 1 is, given the data and a certain prior. So we're going to start with a prior, which is a beta distribution parameterized with values alpha and beta. You find, for instance, 9 connected pairs out of 34, and you update your beta distribution into a new shape, which has new updated parameters, which corresponds to alpha plus the number of successful connections and beta plus the number of unsuccessful connections. So to illustrate this, we're going to take a prior distribution, which is based on previous literature, and this is one of the key strengths of Bayesian inference. You can use previous knowledge about the system. In this case, we have a study by Crosse and colleagues 2004, which determines that spiny projection neurons connect to one another with a probability of 0.12 without distinguishing between D1 and D2 neurons. So we draw a prior which has this shape centered on 0.12, and a certain broadness which is in this case arbitrary, but we don't want it to be too peak or too broad. And we then plug in the experimental results from the study by Taverna, and we obtain these posterior curves here. So as I said, we now have a probability of a description of the probability connection for all values between 0 and 1. And you can see how the blue curves, which represent the posterior curves for D1 neurons, segregate more to the left than the red curves. And so we could say at this point that D1 neurons probably make less connections than D2 neurons. But we can be more quantitative about this by defining a difference between two properties of connection and the PDF for this difference, for which you have the formula here. So we can start by asking the questions, do D1 neurons actually prefer to connect to other D1 neurons, as was claimed by Taverna and exploited by Burke, by calculating the PDF for this particular difference. And we see that it actually includes 0 in its kind of main domain. And we can also ask the same questions about D2 neurons, and it's the same result, meaning that neither neuron subtype, D1 or D2 neurons, prefer to connect to a particular postsynaptic target. Now that we found there is no postsynaptic targeting, how about we collapse the neuron counts according to the presynaptic neuron? So collapse the 2 red curves and the 2 blue curves and see whether the D1 neurons make less connection with D2 neurons, which is the result which we found here. So here you have the collapsed postsynaptic posterior curves, and here you have the PDF for the difference. And you can see that D1 neurons make connections with the property of about 10%, where D2 neurons have a rate of connection of about 20%. This is a difference which we can track across development thanks to a recent study by Krzyzewski and colleagues 2019, which looked at rate of connectivity at 3 different stages of postnatal development in the mouse. And you can see here that D1 neurons make their connections very early on. They're already present at postnatal stage 6, whereas D2 neurons gradually develop their connections over time and eventually overtake the rate of connections of D1 neurons. So if you remember in my first first slide about the experimental procedures, I mentioned a study by Plenner and colleagues, and we could do the same extraction of posteriors for this study. And what we found is that we had posteriors which were smaller than those for Taverner. So this isn't obvious here, but you have to take my word for it. If you compute the PDIS for the differences between the rates of connectivity, you can see they're all slightly offset from 0, except for the light red one here. And so this is an effect which might be due to the different experimental procedures. Taverner limited themselves to a maximum distance of 50 microns, whereas Plenner used a maximum distance of 100 microns. And if you're sampling in a larger area, and if you assume that neurons which are further away are less likely to connect, then this might explain the discrepancy between the two studies. And so we wanted to find a way of reconciling the two studies. And to do this, we're going to use the following model. It's based on 2 components. First, you have the assumption that the property of connection as a function of distance is an exponential decay function. And secondly, you're going to assume that the experimenters are sampling within their maximum area equiprobably. You combine the two, and you get this equation here, which relates the property of connection on this axis here, and the beta parameter, which defines how fast the property of connection decreases with distance. So, to recap, we have these two studies which have a slight disagreement about the property of connection, which we assume is due to distance. And we want to make sure the 2 studies are in fact in agreement. And to do that, we want to relate the property of connection with the decay rate. So our first ingredient to do this is a way of transforming this property of connection into a decay rate, so we can solve this equation numerically. Secondly, now that we have these posteriors, we want to transform this posterior into posterior for the decay rate, and this can be done mathematically once again, thanks to this equation here. And so if we look at D1 neurons, here you have the posteriors for D1 neurons between the 2 studies converted into posteriors for decay rates. And you can see that they overlap quite nicely. It's not perfect agreement, they overlap. And here you have the exponential decay curves, which correspond to the peaks of the posteriors here. And you can see they are quite close. And the here you have the half distance shown here, which is quite close in both studies. We can do the same for D2 neurons. The agreement is less evident, but we're at least satisfied the curves are close-ish together. So the estimates lie in the same ballpark. So to conclude my talk, so I ended by saying that there was relative agreement, but it wasn't perfect. So one thing we'd like to test is, instead of using the equiprobable sampling, we're going to use a nearest neighbor distribution. So we're going to suppose that given a choice of neurons within the maximum distance, experimenters are going to prefer to connect neurons which are close together. Psychologically, you'd expect that you're motivated to find connections, so that kind of makes sense. So I've already tested this, and you can reach a better agreement with this method, given parameters which I won't get into that. Secondly, we'd like to complete our map of the striatum by including interneuron data. So we've already done this. There's data from a study by Gittis and colleagues 2010, and there's a very recent paper as well about the cholinergic interneurons, which I have to add to this ongoing work. Thirdly, so what we're going towards right now is a map, connectivity map of the striatum, but we'd like to complete it also with a Bayesian analysis of the strength of connections. So for instance, Taverner found that D1 connections are relatively rare, but in addition to this, they're also relatively weaker than other types of connections. So this is something which we could treat with a Bayesian analysis using, I suppose, a Gaussian prior. So this is not done at all. This is really out there. And lastly, if given time, all of this data could then be used to draw a new model of the striatum in which we take into account the lateral connections and their potential effects on the computations which take place in that part of the brain. Thank you very much for your attention.
Thank you very much, Francois. everyone, please feel free to ask any questions. You can put your virtual hand up or use the Q&A function in the Zoom. And that was some perfect timekeeping, by the way. I didn't even have to put my hand up. Again, panelists, feel free to also ask questions if you do have any. Okay, I don't see anything coming up at the moment. I'm not sure if people are typing very long questions or if your talk was just very, very clear, Francois. All right, I think with that, we're done then. Do feel free, obviously, to get in touch with the speakers and ask anything if things come up. Thank you again, all the speakers. Really well done. And hopefully see you around in some of the other talks this week.
Great, thank you, Eleanor and Ellie.
Thank you for inviting.
Thank you.
Thank you.
A postdoc at Dartmouth College in Hanover, New Hampshire, USA. I've been told we have a few moments while people trickle in, so I'll use that time to tell everyone in the US to vote on Tuesday. Make a plan, have a plan. If you've already got a plan to vote, call your friends and make sure they have a plan. And if you're not in the US, call your US colleagues and make sure they have a plan, because it's super important. So we have two wonderful talks today. One of our speakers is not going to be able to make it, so we'll have a little bit of extra time for discussion. These are interactive talks. So the speakers are going to get 5 minutes to give an overview of what they would like to talk about, and then we have 10 minutes for discussion. If you'd like to come on stage to ask a question, there's going to be time for that. So post your question in the Q&A, and we can, and whether or not you'd like to come on stage, and I'll bring you on. Otherwise, you can post in the chat. We'll be monitoring that too. Just make sure you have your messages set to panelists and attendees, so that way we can all see what you're saying. Because this is an interactive session, I would really like it if you guys introduced yourself in the chat right now, so that way everyone knows who's here. This is a great time to do that before we start. And again, use the Q&A tab at any time. For those who are on Twitter, you can tweet about, or we'd like you to tweet about this session, so you can do that using the hashtag NMC3. And with that, I'll introduce our first speaker. Benjamin Silver is a PhD student at Columbia University. He recently published a paper on sex-biased patterns in breathing and their influences on resting state fMRI recordings. But today he's going to be talking to us about something different, which is titled Neuroscience Education from Home, Challenges and Opportunity. So Ben, you can take it away.
Cool, thanks Adam. And thanks everyone for being here. I'm really excited to be speaking at this conference. It's really cool that something like this is happening during a global pandemic. I am a psychology PhD student at Columbia University, but I also work as an assistant instructor for a neuroscience education program, and that's going to be what I'm talking about today. But what I wanted to do with this talk was really create a place for people who do neuroscience education to discuss and share their experiences overall, but also specifically this past spring as we had that really crazy transition to teaching online. So please feel free to use the chat throughout, talk about your thoughts and your experiences. I'll be posing some specific discussion questions throughout actually. And you should just feel free to type your answers to those in the chat. And when I finish speaking, then maybe, people can raise their hands and people can be brought on stage, as it's called. So before I dive into our experience, I just want to talk quickly about why neuroscience education is important. I'm sure we all believe this to be true, but just some evidence. Neuroscience PhDs have grown more quickly than any other field of biology in the past 30 years. And this is really exciting, right? Because we can fill great conferences like this one with really cool research. But as we grow, we want to make sure that we can maintain that growth and also make sure that we're growing in a way that is fostering a community where everyone feels welcome. And we know that neuroscience education, and specifically neuroscience education in the form of extended research experience, can really encourage students to enroll in a PhD program. So this is a graph from a study of a program that was specifically for Black high school students. And it shows that students who had more research experience were also more likely to enroll in a PhD program. So neuroscience education is really important, and specifically neuroscience education in the form of training in terms of research experience is really what we need to make sure that we can maintain our field as a diverse, exciting place to do research. So that is where the program that I've worked on comes in. My program that I worked on is called, or that I still work on actually, it's on hiatus right now. It's called Brain Research Apprenticeships in New York at Columbia, nicknamed Brainiac, which is quite clever. It's run through Columbia's Zuckerman Institute, which is Columbia's new neuroscience institute. And in this program, high school students spend the spring learning neuroscience, learning laboratory skills. And then in the summer, they have an internship where they work in a lab on their own research project. And this culminates in a final poster presentation. You can see here students from a couple years ago dabbing with their posters. And the students get paid to participate in this program. And we also specifically, try to get students to participate who go to schools who might not have the resources for a program like this otherwise. We work specifically in Harlem and Upper Manhattan. So we're really trying to make sure we are giving underrepresented students the opportunity to have these experiences. And I think of the benefits of Brainiac kind of in three main planks. There's the academic benefits. You get to learn neuroscience, you get to practice laboratory skills. There are the professional benefits. You get experience having a job. You get experience public speaking, interviewing, practicing science communication. And then there's also the social benefits. You get to form a community with like-minded students who are just as curious and bright and enthusiastic as you are. And of course, make connections with actual scientists. So this is how Brainiac normally works. Then, of course, as we all know, spring 2020 happened and Zoom took over our lives, as it is currently doing right now. And Brainiac became this, completely online. So before I talk about our experience with Brainiac online, I want to just pose all these discussion questions now. And I really encourage people to write about their experiences in the chat. And when I'm done talking, I can read some of them, and people who want to share more can be brought on to speak about it further. But so just tell us all a little bit about your experience this past spring with neuroscience education. So what was your program? Or was it with high school students or undergraduates? What was really difficult about transitioning to online learning? And then also, were there any aspects of online neuroscience education that you found worked better online? And that's kind of the key thing that I really hope we can discover together, some ways we can improve neuroscience education going forward because of this wild restriction that's been put on us. So for Brainiac, I'll talk about the drawbacks and benefits in terms of these three planks again. So academically, as I'm sure we all experienced, a huge drawback of online learning is that students ask way fewer questions and can be much harder to engage online. With content that's often as new and confusing as neuroscience, it was really clear when we were in person how much students benefited from getting to ask questions. And just being online makes students less likely to do that, which is unfortunate. And of course, there's no opportunity to practice these hands-on skills like dissecting a sheep brain, which is always a fan favorite, or practicing pipetting. We actually, our very last day before we all went remote was a session where we were all practicing pipetting skills. And then students didn't actually get to finish that, that activity because we went remote. Professional drawbacks. It's really hard to balance your home life with a job. We're all experiencing this, but some of these students don't necessarily come from the best home situations, and it can be tough to balance, some really rigorous academic learning with what's going on at home. And also, we're all learning right now too, right? Part of this program is to impart professional knowledge onto our students, but we're all really unclear about what the best practices are for virtual professional skills. And so it's really hard to, act like an expert in that when it's all new to us as well. Social drawbacks, students really expressed that they missed having in-person interactions with their peers. They didn't really feel connected to their peers. And of course, harder to form a relationship with mentors. Some students, didn't have video capability on their computer. And so their mentor actually never saw, what they looked like, never actually video chatted with them. So it's harder for those relationships to form. But of course, there are also some things, and I'm already seeing some of them in the chat, some things that online learning actually made us, reimagine or rethink the ways that we do neuroscience education. There's some ways that online learning actually was a huge benefit. One of those in particular is in our program, we really focused, when we pivoted to online learning, we began to focus on computational skills. And especially with high school students, it can be really hard to dive into that. It can be scary to do that. And it's often sidelined for the more exciting, pipetting, hands-on stuff. Wet lab stuff, but obviously computational skills are really important, as NeuroMatch knows, since I think it's originally a computational neuroscience thing. And so we got to really put a focus on that. And this coming year, actually in Brainiac, because we're going to be online in the spring, we're going to be baking that into the curriculum from the start, which will be really exciting. And also when we work online, you have more opportunities to work at your own pace, which is actually what being a scientist is really like. So that was nice for students. Professionally, we were able to get really creative with multimedia science communication, thinking about all the ways that you can utilize online tools to make science engaging, which really is the future of science communication and science engagement. We also helped students sort of cultivate their online presence, their professional online presence with LinkedIn profiles and that sort of thing. Socially, even though students didn't get to see each other, we tried to shore that up with planning like online social events. And not a lot of students would always come to those, but when they did come, so they expressed that they were some of their favorite parts of the program. And we got to get really creative with those. We had an online trivia night. We had an online Pictionary night. So that was fun. And also, there were more channels to interact with students, whether it was via Slack or WhatsApp or Google Classroom. Because we were online, we had to really get creative about how we were going to engage students. So that's our experience with Brainiac. I would really love to hear from people in the audience your experience. So I'm seeing some things in the chat. Does anyone, either if you put something in the chat or not, I think that you all have the ability to raise your hand. And so I would love to hear some of these discussion questions. What is your program? What did you find most difficult? What aspects worked better? So if anyone wants to raise their hand and speak about that, that would be really great. We can bring you on stage.
I guess I'll just just start off the discussion, perhaps with
please.
The idea, like the Skype a scientist, which is what I posted in the chat. But it made me realize how much, like how different of experiences you can give to students by having them talk with people who are in different communities that you wouldn't necessarily be able to have at your own institution, or if you're working with students of color and you want to give them exposure to other scientists of color, you can bring those people in if they're not in your home community, which is something that like Dartmouth, for example, really struggles with. So I think it's been really interesting to see like the or it's been really beneficial to be able to bring in that outside influence as a means of diversifying the experience, because that seemed to be a major complaint, was that the experiences were like very homogenous if people weren't creative, which it sounds like you guys are really trying to be through Brainiac.
Yeah, we did something similar as well. We actually brought in someone who does science communication full time. And again, it was something that we wouldn't necessarily have maybe done if we were in person, but because we were online, we could get more creative with what types of people we're bringing in, where they're coming from. I'm sure we're all experiencing this now with our own institutions, like weekly seminars or sessions. You know, you're now not limited to having to pay people to travel, right? You can have anyone come from anywhere, which is really exciting. And yeah, can definitely be applied to neuroscience education. And the homogenous thing is a really good point. Like Zoom gets really, really tiring. But if you can figure out ways to make every Zoom call not the same, you can mitigate that a little bit.
Yeah, that's a great point. And one other thing that that has kind of struck me as I'm preparing to teach a class this winter. And so I come from cognitive neuroscience, which gives you the benefit of like making your experiences more human. Whereas if you're a molecular neuroscience or someone who requires being in a wet lab, you're going to be in a very different boat. But we can develop experiments that the students can like test on each other as long as we help them install the right software. So they can still have this interactive experience with cognitive neuroscience experiments using like online platforms and things like that, which you don't necessarily get to do in a wet lab. Doing the wet lab, yeah, getting a similar wet lab experience would be challenging.
Yeah, and some students express that, and you know, some students have their mentors just sort of like Zoom them in or FaceTime them in while they were doing wet lab stuff. But of course that's difficult. So I know we're a little bit more flexible with time because we only have 2 talks, but I also know that I'm running up on my time. So I encourage people to, oh, go on.
Oh, I just messaged Paula privately and said we would let you run until 9:20. Okay. If you'd like to do that, or we can pick your discussion or have more of a group discussion at the end, if that would be better for you.
Yeah, either one is fine. I think we can let people continue to think about this and put thoughts in the chat. I did see one other message that I wanted to comment on. Martin said the use of whiteboards is really helpful. I would totally second that. There's so many great online tools that we're trying to find online so we can make online learning more exciting, but that can also be brought into in person. I remember my dad is a high school teacher, and when he got a smart board for his classroom where he could like write on, he was so excited. It was so exciting to him. And I think we should take that same excitement with us, hopefully when we move back in person for neuroscience education. Think about how we can bring those tools into the classroom. So I'll just share a couple takeaways and then I'll finish up. Neuroscience education and training for pre-college students is obviously really important. And it's particularly important when significant research experience is involved, and particularly important for students of color or students who are traditionally underrepresented in the sciences. We all know that the transition to online learning was really abrupt and really difficult, but hopefully we don't have to always associate, March 2020 or January, February 2020, if you're coming from somewhere else other than the US, with online education, right? Online education can be a tool for improving neuroscience education, and we can hopefully use this moment to reevaluate our practices. And I also want to encourage us, just like we all are so good at sharing our research with each other, part of having a vibrant research community is also having really good educational practices. And so I really want to encourage us all to share our education practices and experiences with each other so that we can, all improve together and make sure that our research community is one that is exciting and makes people feel comfortable and welcomed. So this is the Brainiac team. From counterclockwise from the left is Diana Lee, who's the lead instructor of the program, Alyssa Mayers, who is a program manager, Paula Croxon, the associate director of public programs at Zuckerman, and then down here, Nayeli Valdez and Matthew Ramos, who were both alumni of the program who came on as interns for instruction. So feel free to keep posting your thoughts about this. I see there was another question in the chat. Yes, definitely finding a PhD position at this time is part of this discussion, because that's just a continuation of the training and education experiences we've had. So keep putting your thoughts in the chat, and hopefully at the end we'll have more time to have a larger discussion in tandem with the next talk to think about these issues. Cool. I will stop sharing my screen now. Thanks, everyone.
Great. Thank you, Benjamin. That was really a very interesting talk. And for those who have only recently arrived, our third speaker was not able to make it today. So we're going to give both of our speakers their traditional 15 minutes, and then we'll have time for more discussion at the end, and still close the session at 9:45. So now we'll move on to our second talk, which is from Paula Denota from the University of Toronto, Mississauga campus. She has asked that when I asked her if there was anything she'd like me to announce, she asked that I send her hope that you have all been and continue to be taking care of yourselves in these unusual times. And I thought that that was a wonderful sentiment to share. And Paula is now going to be giving her talk, Neuroscientific Investigation of Police Perception, Cognition, and Behavior.
Thank you guys very, very much. Let me get my share screen happening. And great job, Ben. You really set the stage, set the bar high for me here. So I hope I can keep everyone engaged here. As Adam said, my name is Paula. I'm from Toronto, reporting live here. And I think, representing our Canadian kindness, I really hope that everyone is doing well. And I kind of want to flip the script a little bit. I really like how Ben had the discussion questions and you kind of continued on. So for those of you here, if you check in the chat, I've actually put the questions up already to kind of give you a sneak preview, or if you want to just start thinking about, how can we as neuroscientists kind of contribute to conversations regarding policing, which is sort of top of mind, especially here in North America, and just things that we can think about. How can the different methodologies that we use in our research, how can those answer questions relevant to society and to policing right now? So just to get the ideas marinating, but I will get started. So as we know, police and other law enforcement repeatedly face potentially violent, dangerous, and traumatic encounters in their day-to-day jobs. And various sources of threat can include sensory cues from the external environment or internal psychology, including a sense of uncertainty, unpredictability, or memories of past experiences in similar circumstances. So together, these different sources of threat trigger a general sense of unease, and they kickstart a series of coordinated physiological stress responses that largely affect the brain and the body. So once threat is perceived, the autonomic nervous system initiates the fight or flight response, which, among other things, increases heart rate to mobilize the individual for action and survival. These effects also influence several perceptual and cognitive processes, and there are a lot of research studies in both novice and experienced police officers that they report a lot of perceptual distortions, including neurodetention and tunnel vision, as well as different sensory distortions like slowed down time, amplified or even dampened sound. And these are during both real and also simulated incidents where they had to fire their weapons. These acute stress responses, they also change the focus of attention to the target that needs to be neutralized in the short term over longer term goal-directed behavior and decision-making processes, again in the favor of immediate survival. So together, various cognitive processes can be lumped together. So perceiving, understanding, as well as predicting possible outcomes in second-to-second encounters is extremely important, and it's referred to as situational awareness. And it's one of the most essential skills that police have, and especially with respect to use of force decision-making, as that's where a lot of my research lies. So following the fight or flight response, it triggers a slower neuroendocrine response. So that releases a cascade of neurochemicals that once again travel throughout the brain and body and exert a variety of effects. So cortisol is perhaps the most commonly known stress hormone that we know with long-term chronic exposure can really negatively impact both physical and mental health. And this is commonly seen among police and other emergency service and like first responder workers. So important from a police training and performance perspective, cortisol blocks access to brain regions involved with learning, memory, and executive functioning, like the prefrontal cortex, and that accounts for some of the perceptual and cognitive effects that I mentioned in the last slide. At the same time, the acute stress it causes us to rely on more automatic, reflexive, and implicit decision-making strategies or habitual muscle memory skills that are really deeply ingrained in mid and hindbrain structures. So therefore, this very lovely stress continuum that I'm going to show you here, knowing what we know about how stress impacts the brain and learning and decision-making, it's really important that in super high stress situations, so seen here in the far right of the continuum, that skills are trained in such a way that their performance is going to be robust to these interfering effects of stress physiology. So under more lower stress conditions, like it seems here on the far right, that's more conducive to lecture-based training or classroom learning of declarative knowledge like laws and regulations. So somewhere in the middle, in that sweet spot there, at moderate levels of stress, there's a growing body of applied research that both myself and my postdoctoral supervisor, Dr. Anderson at the University of Toronto, shows that adaptive learning through scenario-based training, it exposes officers to this feeling of stress that they would experience in the real world, but it allows them to safely practice these dynamic cognitive skills in a variety of very realistic settings. So as you can see here, there are live setups, with fake blood and props and actors that are either professional actors that can be hired, excuse me, or they're often experienced use of force instructors that have been in the real world, have real world experience, and act out various scenarios that will require the officer to either have to shoot the person, because it's an unfortunate reality of police work, but it is something that is trained very heavily, as well as training other de-escalation skills. So how can you verbally speak to someone, or how can you demonstrate in your body language as well to kind of bring the situation down so that we don't have to use lethal force. So together with partners at Finland's Police University College, Professor Anderson has developed a performance and resilience training program. So it actually uses heart rate variability biofeedback to condition adaptive stress responses. And that's through the use of various breathing techniques and cognitive skills like situational awareness during these critical incident scenarios, so that you can see the heart rate here when someone suddenly appears with a weapon. Officers are able to adaptively use these behavioral skills to bring their heart rate back down to a place where they're able to make more conscious, explicit decisions. So while we've published research on the effectiveness of this program in reducing lethal force errors, so that's shooting when you're not supposed to be shooting, so that's always a good win. And I do want to note that we are dealing with police officers that are well trained, and they already have very low error rates, which I know to us as civilians, maybe it might seem that any kind of use of force is totally unacceptable. There should be 0% error rates, but these are significantly lower error rates than what you see in other trained professionals, like in surgeons and medical professionals, as well as in military friendly fire error rates, which there was one report. I'm sorry to our friends down south there, but the error rates were about 19%. I don't think you want to be shooting friendly fire mistakes 19% of the time. So I'm very happy to say here that already we were starting with a very low error rate, and these were maintained for up to 18 months following this training program, which is great. And also we show sustained improvements in recovery from stress. So following, firing your weapon, you can imagine that an officer's heart rate is run super, super high. So recovering from that, because the reality of their job is they have to go to the next call. It doesn't just end at that call. So being able to physiologically recover from that stress, again, for their own mental and physical health, has been really significantly reduced, especially up to a year following the training. So these are really excellent empirical results that we've shown for this specific training program. However, the neurophysiological mechanisms by which this resilience training is effective still remains unknown. So there's still a lot of room for discovery on our part as neuroscientists. So through my applied research experience, I do have a PhD in cognitive neuroscience, but now have been working with all these great first responders. I've identified several practical considerations for conducting scientific experiments in real world or field settings. So first, there are many potentially confounding variables when investigating certain causes of stress or threat, including organizational stressors. So those are workplace type of stressors like the culture or stigma and attitudes towards different groups of people, including racialized, marginalized groups, and as well as among first responders and police, especially. These are hyper-masculine cultures, so female officers are often underrepresented or stigmatized and face their own unique stressors, as well as operational stressors. So these are things introduced in the day-to-day duties, so either shift work related fatigue or the risk of psychological or physical injuries as a result of their duties. Another practical consideration for police research is controlling for pre-existing mental disorders and as well as traumatic exposures, both of which are extremely common among police and is also severely underlooked in existing research and would certainly confound any types of investigations on both learning or performance outcomes, as well as any kind of training effectiveness. And as well, a major problem in policing currently, and maybe some of you others have more to share, but currently there's a lack of standard definitions or practices relating to the use of force, not only between individual agencies, so what's happening in Toronto might be different than what's happening in Montreal, but also around the world, between countries. So that includes a lack of standard definition for what includes use of force, what's included in use of force training, how long it's trained, as well as how often. And this limitation, it also introduces an applied research problem for, and I was practicing, and as a native English speaker, I was fumbling on this word, but operationalizing measures of police behavior and cognition with either very objective types of measures or subjectively, as they're commonly evaluated and rated now, is definitely another problem in existing police research. So based on all of the information that I've presented, hopefully haven't run through it too quickly, and I really want to draw from the unique perspectives of everyone attending, whether you're from Canada or the United States like us here, but I really welcome any other international perspectives as well of how some important, or what are the important issues in policing in your societies, and what are some things that can be answered from a neuroscientific perspective, since that's what we're all here to share. So how can we as neuroscientists really meaningfully contribute to some really important societal issues? And I welcome any questions or comments, discussion points. Anyone?
Thanks, Paula. That was really interesting talk. I guess I wanted to start with something that sticks out in my mind as a as a person from the US, where police action is being scrutinized at the moment. And you see these, you see a lot of videos of the moment when the violence begins, right? Like this is often what we see, and we sometimes see it from bystander cameras. I personally have not seen so many that actually come from police officers wearing body cameras. Yeah. But often what I'm wondering when I see a video like that is, what is the officer paying attention to in the moment before the violence precipitates? Mm-hmm. And it seems like one interesting method that could be deployed both in the experimental setting and perhaps in the real world setting is like real world eye tracking, or doing something like this, so we know, so that way we can begin to characterize the behaviors that would precipitate a use of force in an inappropriate way. Do you have any thoughts about that?
Yeah, and that's such a fantastic observation, because I think, we hear police or police use of force, you immediately start to think of all these terrible viral videos. And it's hard to explain each one of them. Each situation is so unique. The officer is unique. The context is unique. But absolutely, there is definitely a growing use of eye tracking in police research. I think there was actually recently like a systematic review as well of kind of all of the research that's been done, and I just see my PhD supervisor post a question. Hi, Joe. And so very excited to say also that in our partnership with Finland's Police University College, I have done research with Chief Inspector Juha-Matti Huhta. I'm not sure if he's online, but he's a PhD candidate as well, and has used eye tracking to compare both novices and experts to try to see how do experienced officers that might have additional training and experience, where do they focus their attention? But as I mentioned when I was kind of describing the stress response as well, that immediately has an influence on where you're looking, what are you attending to? So the fact that a lot of police training currently does not integrate any type of stress management training or any type of consideration for how stress influences these cognitive processes, as fundamental as your attention and gaze and where you're looking and what you're attending to. And, we're all famous, very familiar with that famous science experiment where you have to count how many basketball tosses there are in the gym, and you don't notice the monkey suit guy walking in the background. So it's totally that idea of blindness where, you see something that either your brain or your training is telling you, hey, like this is a suspicious looking figure, but you don't notice other important cues in the environment that should be informing your decision making. It should be guiding other subsequent decisions. So you often see these snowball effects of when one thing goes wrong and things continue to keep going wrong. And unfortunately, police are uniquely tasked. They're the only profession where they're legally entitled to use a lethal force. Other professions you're required to try to save a life and preserve life as much as possible. But police are the only ones, as well as us citizens in self-defense and in defense of others' lives, if we believe that they are in danger, at least here in Canada, that's the law, that police are the only ones that have the power to pull a trigger. So the gravity of the consequences of these decisions, it's really important that we ask these questions from an empirical perspective and use exactly these types of tools. So I think you're jumping ahead to my next question there, Adam, but exactly looking at what methodological approaches would be effective. So eye tracking is definitely one of them. It's definitely very heavily utilized in lab settings. But in the real world, can we get Google Glass or things like that to see where officers are looking? And you mentioned also body-worn cameras. There's a great deal of controversy surrounding those as well, for privacy issues, for resource issues. A lot of police agencies can't afford to equip every single officer with a body camera. And then becomes the issue of, are you generally surveilling the public? What are our individual rights as civilians for being on video? Things like that. So maybe from the audience, I invite you guys to think of other approaches like EEG or MRI. I know it's hard to stick officers into a scanner and expensive as well, but that's something that I hope to engage in the future. And I do want to ask the question from the chat. I'll ask from Dr. Joe D'Souza. So how can your results educate future police officers? And that's kind of our hope. And in partnering with the Police College in Finland and other agencies here in Canada, we hope that the findings of our research can be directly involved in implementing better police training, especially use of force training. So again, these life or death decisions. And the wonderful thing about our partners in Finland is that they've seen the research, they've seen that this eye prep training works, and it's actually been integrated as part of a mandatory part of their basic police training. So every single police officer receives training in stress physiology, like basic physiology, cortisol, fight or flight response, how it affects your perceptions, how it affects your cognitions, and different decision-making strategies. And through the adaptive scenario-based training, that allows you to experience that stress and work through that and actually practice these skills of like literally just taking a breath, bringing down your heart rate so that 1 second can make the difference between life or death of making a better decision. So I'm going to stop talking now and just let someone else maybe ask a question. But any other
one thing that just to give people time to post anything that they're thinking, one thing that I noticed in your data was this kind of upswing in errors in use of force that happened after 6 months. So I was wondering what you think was was causing, like, might have led to this? Is it something where it's like a vaccination where we need to like do a 12 every 12 months, do a refresher to make sure that, you know, a person remains continuously inoculated? It's just to continue the vaccination metaphor.
Absolutely.
Yeah. What were your thoughts on that?
Yeah, that's absolutely right. So like with any kind of new learned skill, whether, you know, we look at, you know, more basic motor learning research where you have these adaptations of reaching behaviors, you know, there's kind of a slow learning curve. And then once learning is sort of optimized, it does wear off. And we know from MRI scans that the kind of the motor representations for any newly learned skill will degrade. You know, it's kind of the use it or lose it principle, that if you don't use these skills. And another really important thing too that I want to be able to convey on behalf of our police partners is that these use of force incidents, even in America, I'm very familiar with the Canadian statistics, so I do want to say that the use of force incidents are extremely rare. So it's obviously not rarefied in the popular media. These are, you know, the first images that we see when we think of police. But thankfully for many of them, they might go their entire career without ever having to fire their weapon in the course of duty. And that's, you know, obviously what we would hope for. So these are extremely rare scenarios, but it is nonetheless something that they have to prepare for and train for adequately. So especially given that, the fact that they, police officers, the resources might only have 1 day a year in which you practice a training scenario, and it'll also coincide with your certification and recertification of firing your weapon and having the basic competency basically in using your firearm. So also pairing evaluation with learning, and I think that kind of ties in nicely to Ben's talk as well, is that, you know, we have education kind of in one box, but training and evaluation, it introduces its own source of stress. And it's like, okay, I have to do this perfectly while I'm learning it for the first time. That you would almost never kind of teach someone that way. So the standards that we use for teaching and educating police officers should definitely be held up to the same standard as we do for surgeons and other people that are involved in life or death decisions. So I think that actually will bring me nicely to the last discussion question is, if I can move my chat window here, is how we as neuroscientists can contribute to evidence-based policing. So what is evidence-based policing? Well, it really is what it sounds like. So using policies and practices that are rooted in some sort of either scientific evidence or empirical evidence. So like the research that I just presented to you on the use of force training. We see that this works. We followed up with police officers up to 18 months. We know that around 12, 18 months, the skills do start to degrade. So what we try to do with all of our peer-reviewed research studies is to try to write some practical recommendations at the end that practitioners and stakeholders, people that are involved in making the rules of how police are trained, that they can actually use some of these recommendations to improve current training practices. Another thing we've also done is writing policy reports. So these are to the ministries, to the government bodies, again, that govern how police are trained, and writing these very lengthy recommendations, like the answers are there. What do we do? What do we do in response to all of these errors of lethal force? Well, this is the science. We've established the science that this is what happens to your brain and your perception in these critical incidents. And these are ways to train officers that they're better prepared, protects their safety, protects the safety of the public. But it's up to that part of the chain to work and to put these practices into action. So that's what we hope to do. And I'm happy to answer any other questions, but I do just want to quickly acknowledge and thank my postdoctoral advisor, Dr. Anderson at University of Toronto, Mississauga, our fantastic partners at Finland's Police University College, funding sources, and the ministries, Ontario Ministry of Labour and Ministry of Solicitor General, that have supported our lab in generating some of these science-based insights, as well as some of our partners across Canada that are dedicated to researching and treatment for military and public safety people that protect us and have really kept us all safe, and especially during COVID as well. It's just an additional stressor for them and for all of us. So I want to dedicate this talk to their service. So thank you guys very much.
Great. I think that was two really fantastic talks. We're just about at the time when, unfortunately, I need to cut us off. But thank you both again. I think this work is on both of your parts is very timely and important. And thank you for talking with us about it today. And thanks everyone for listening. And we're looking forward to seeing you at the rest of the Neuroscience Neuromatch conference this week. Ciao.
The psychology, at least.
Well,
So,
I mean,
yeah, it's going. Given the circumstances, I think it's going fine.
Yeah.
Yeah.
We've had
We are live now on YouTube, so I'll start the webinar in a second. And I'm off screen. Chat.
Good.
Thanks again. Hello, everyone. Looks like we're live on YouTube and we're live on the webinar. It's my tremendous pleasure to introduce Viola Stiermer, who's a professor at Dartmouth College. She's just moved there actually from UCSD. And she's going to tell us about her work looking at how salient sounds boost visual cortical processing and also enhance visual perception. So I'm really looking forward to hearing about this. Viola, go ahead, take it away.
Okay, thank you very much. It's a great pleasure to present some of my work to you all today. Welcome everyone, good morning or good evening, wherever you are in the world. It's a nice morning here in Hanover, New Hampshire. When we look around the world, detailed visual inputs get rapidly projected from the retina to the visual cortex, which then processes that information that leads to our rich visual experience. And traditionally, visual processing is studied in isolation from other sensory systems. However, that research misses one important thing. In the real world, visual objects do not occur in isolation, but they're often paired with other sensory inputs, such as a sound, smell, taste, or tactile information. And so it is important to investigate how visual perception and visual processing more broadly is shaped by inputs from these other modalities. And so today's talk, I want to ask the question how visual processing is influenced by other sensory events, with a particular focus on audio-visual interactions. There are numerous studies that have shown that sounds can influence visual performance. And I'm here focusing just on studies that have looked at spatially specific effects of sounds on vision. And so much research has found, for example, that participants perform better when a salient peripheral sound appears at the same location as a subsequent visual target, for example, in oriented grading, as shown in these examples here, relative to when that sound appears at another location, for example, in the opposite visual hemifield. And so this has been shown in a variety of tasks using different types of behavioral measures. For example, participants respond faster for same location objects relative to opposite location objects. There's also an increase in the detection rate of visual targets when they're co-localized with a sound. And we've also shown that they can enhance perceived contrast of low-level and also high-level stimuli, and they also improve discrimination accuracy. So there's a lot of research already showing that we have these influences on sounds on visual performance in these types of very, very different types of visual tasks. The question that I want to focus on today is what is driving these behavioral effects? So how do salient peripheral sounds influence visual performance? And to do this, in the first part of my talk, I'm going to use human electrophysiology, and I will show that sounds bias visual cortex activity, and that these biases that we see in visual areas contribute to the behavioral effects that we are seeing. In the second part of my talk, I want to dive a little bit deeper into what these biases are, and I will provide some preliminary evidence suggesting that these are primarily due to enhanced processing at the sound's location, with no real signs of suppressive effects. I will talk a little bit about what this means for spatial processing across modalities more broadly, and also for spatial attention. Okay, so let me start by walking you through kind of like a typical cross-modal perception task that we very commonly use in the lab to study these types of effects. So participants in this task perform a visual discrimination. They are asked to indicate whether a briefly presented Gabor patch is oriented clockwise or counterclockwise at the end of each trial. To reduce location uncertainty, that Gabor patch oriented grading is always briefly masked after it appears. So participants know exactly what happened and they only do the orientation task. We threshold performance using a calibration task at the beginning of the trial and task, just to make sure that participants perform kind of at a rate of 70 to 80% correct. And so critically, prior to the occurrence of these visual targets, we present a peripheral noise burst, so kind of this salient sound either on the left or on the right side. These sounds come from external speakers that we mount to our monitor, so they're really an external space, but they're not critically predictive of the target location or of the target orientation. So these sounds are entirely task irrelevant. And in fact, we usually tell our participants to try to ignore these sounds as good as they can. They're pretty salient, so it's difficult to ignore them, but we really don't want them to kind of engage with the sounds at all. And so when we're looking at performance, we can still split up our trials based on whether those sounds happened to occur at the same location as the visual target, which we would call validly cued trials, or whether they occur at the opposite location of the target, which are invalid trials. And so what you can see here is that we do find a performance difference such that participants perform more accurately when the sound happens to occur at the same location as the visual target, or valid trials relative to invalid trials. And so this is what many other studies, what I just alluded to, have also shown, that these task irrelevant sounds that we think capture attention to the left or right side of space actually influence performance, despite the fact that they're not relevant, that they don't tell participants anything about these visual targets. Okay, and so now we want to see what happens actually, how these sounds are processed and how they affect visual cortical processing. And so you probably noticed in this task that the sounds are always very rapidly followed by these visual targets. So we have a sound and about 100 milliseconds later, or sometimes even less, the visual target appears. And so that makes it actually very tricky to look at the neural activity elicited by the sound alone without being confounded by the visual processing of the visual target. But we can do a little trick here. So in this task, we can insert trials where the sound appears simply without a target. So in this particular case, actually on half of the trials, we present a sound and then there is no target occurring until the next trial starts, so for about almost a second. And so we intermix these different trial types. So sometimes participants do see the visual target. On other cases, there is no visual target present. This is what we call sound-only trials. And they're all randomly intermixed, so the participants don't know in advance which trial is about to happen. But now we can use these sound-only trials to look at EEG activity elicited by the sound themselves without being contaminated by any visual processing of the visual target. Okay, so that's what I'm going to show you next now. Now I'm going to be plotting the event-related potential over occipital, parietal scalp sites, and we're going to be time-locking these ERPs to the left and right sounds. So we're going to be splitting them up depending on where the sound was coming from. And in particular, I'm going to collapse here the data across where they were coming from, so left and right side, and also left and right hemisphere, to reel the waveforms that are recorded over the cortex contralateral to the sound, which I'm going to be plotting in red, and ipsilateral to the sound, which I'm going to be plotting in blue. And so here, the sound onset is at 0 milliseconds. Our x-axis shows the time from that onset. And on the y-axis, we have just the amplitude in microvolts, positive plotted downwards here. Okay, and so this is what we find. This is how the ERP waveform looks at. It looks a little funky because we're actually looking at the auditory evoked potentials, but over visual areas, which is not very common. Usually you would maybe look for the auditory N1, but here we're actually looking over visual areas. But the really critical part that we're interested in is, do these waveforms differ depending on where the sound was? And what you can see here very clearly is that around 200-300 milliseconds, the contralateral waveform becomes more positive relative to the ipsilateral waveform. And so this effects last for about 200 milliseconds. It is distributed over parietal occipital lobe. That is very clearly visible when we look at the topographical map of the subtraction of these two waveforms. So we can subtract the ipsilateral waveform from the contralateral waveform. This is what the topography here shows. We're projecting that difference to the right side of the scalp here. And so you can very clearly see that this difference in amplitude, so really what's basically causing the spatial effect, is distributed over parietal and occipital cortex. And so this particular ERP component, so this difference between these two waveforms, we're going to term the auditory contralateral occipital positivity, or in short, the ACOP. Okay, and I'm just going to keep using this term throughout the talk just to make it a little bit easier and to be clear what I'm referring to. Right, this term is really just descriptive. It's just saying basically that it's elicited by an auditory stimulus, but we find this component over contralateral occipital cortex. Now, I just showed you the data for a case where these sounds were completely non-predictive and task irrelevant. And in another task that the same participants performed, we actually made just a slight change and we actually made those sounds predictive. So on 80% of the trials, they actually were informative to where the target would be occurring. And so in that case, we have longer stimulus onset asynchrony. So it takes about a second until the visual target appears. And we really here just wanted to test whether this early activation that we see, starting around 200 milliseconds after the sound, whether that's modulated by a kind of top-down goal. So by maybe endogenous attention, or by how informative these sounds are. I'm just going to show you the data for that version real quick here as well. And you can see that we really don't see a difference, right, between the first version where the sounds are non-predictive and this. We can see an overall difference in the shape of the pattern of the waveforms. That might be very interesting, but really these spatial effects, so the difference for contralateral versus ipsilateral waveform, essentially look the same. So what does this tell us so far, the data? We find that the sounds can activate contralateral visual cortex relative to ipsilateral. So we find that these peripheral sounds really induce a spatially specific bias in early visual cortex. These biases start at about 240 milliseconds post-sound onset. They occur regardless of whether the sounds contain some spatial information about the subsequent visual target or not. And possibly one interpretation here is that they're basically prepping visual cortex. They're basically getting visual cortex ready to perform this rather challenging visual discrimination task. Because even though here we're looking at trials where there's no visual target present, participants are nonetheless in a context where most of the time the sound is followed by a visual target. So it might make sense that these two modalities closely interact in this particular task context, because the sounds carry at least temporal predictability, for example, about the visual stimuli. So this really raises the question of how broad this effect is, right? So do sounds only affect vision when the brain is kind of expecting these visual inputs, so when participants are in this cross-modal context, or is this an effect that extends to other types of tasks and other types of kind of expectancies and contexts? And I think this is an important question because it can really tell us about how sounds, and in particular spatial information about sounds, is processed and about the connectivity of our sensory systems. And so to test this, in another experiment, we changed the context rather radically, and instead of participants doing a visual discrimination task, we actually asked them to do an difficult auditory discrimination task. So exchanging our kind of lateralized visual target stimuli with lateralized tone stimuli. So participants are doing a pitch discrimination task in this case, but these auditory target tones, they're still preceded with these peripheral noise bursts, okay, on the left and the right side, exactly like in the first experiment that I showed you. And so now we can look at the activity elicited by these left and right peripheral noise bursts that are task irrelevant, that participants are not responding to at all. But again, participants are now in this auditory context. So they're listening to these left and right tones that subsequently appear here much later, around 600 milliseconds later. And so I'm going to show you exactly the same plot that I showed you before. Okay, so again, recording ERPs with the onset time locked to the left and right sounds, and dividing them up contralateral or ipsilateral to the sound. And so here we find exactly, again, our ACOP component, okay? So again, we find that these peripheral noise bursts elicit a kind of biased visual cortical activity in a spatially specific way, even in this task where participants never see a visual stimulus. They do have their eyes open, they're staring at fixation cross, but they never do a visual task. They never see a visual peripheral stimulus. So all they're doing is hearing different sounds. And nonetheless, these peripheral noise bursts activate and bias visual cortex around the same time as we've seen before. However, in both of these tasks, the targets, in one case the visual targets, in the other case the auditory targets, still appear at a predictable time. And the task itself is somewhat spatial because the targets themselves are actually presented at the left or the right side of space. And so kind of to eliminate those two things, to also take away the predictability in time and also the spatial part of the task, we ran yet another version, in which case basically we're just presenting a random stream of auditory stimuli. Okay, so there are either these left and right bursts of noise, and they're also about half of the time there's a central target tone. And all participants are doing is listening out to the central target tone that's very easily discriminated from these peripheral noise bursts, and all they do is press a button whenever they hear that target. And so here the idea is really that the task is non-spatial anymore. It's a pretty trivial task for participants. And you can also see that we're jittering the timing between all of these stimuli. So really it's just a random stream of different sounds that participants are listening to. And the question here is, do these peripheral noise bursts again elicit and kind of bias visual cortical activity? And what we find is that yes, they do. So once again, around the same timing, we find this ACOP. So we find that these peripheral noise bursts elicit a larger amplitude signal over contralateral relative to ipsilateral cortex. So now I've shown you a bunch of experiments where we find that a salient sound can boost contralateral relative to ipsilateral visual activity in cross-modal versions of this task. So when participants actually performing a visual discrimination task, in purely auditory task, but also in this, what I'm not going to even call really task, but just in this like kind of random stream of sounds where participants just once in a while press a button whenever they hear a target tone. And so across many different experiments now, I just showed you some of them, but across many, many different experiments in my lab, we have found that a salient sound can boost visual cortex activity. And it seems to be the case that this effect is very robust across different task contexts, really suggesting a tight link between audition and vision that cannot be overcome very easily, at least within a single experimental session. And so these interactions, I think, suggest that they're either very well learned throughout life or even possibly hardwired. And one question these data raise is what functional pathways that are involved that lead to this effect. We know this is pretty tricky to get from EEG, but we do know that there are direct connections between auditory and visual cortex based on other literature. But I think the rather late timing of this ACOP, so around 240 milliseconds on average, suggests that not these direct pathways underlie the effect, but rather indirect pathways are involved. And so possibly it could be that these salient sounds essentially act kind of as an exogenous cue that activates the attention network, which then sends kind of these attention hotspots back to visual cortex. And this interpretation is also supported by the spatial specificity of the effect, right? So the brain region that is responsible for the sound location gets activated relative to the unattended or opposite location. And so I think this spatial selectivity is really the most exciting part of this data and has really important implications about spatial processing across modalities. It suggests that there possibly exists a sound-based spatial topography coded in visual coordinates, possibly because of the spatial dominance of visual cortex. And such a mapping seems to exist at a minimum at the hemifield level, so left versus right, which is what we're looking at here. And it might well be the case that that's all there is. It's just this kind of hemifield mapping, and possibly also only kind of in the periphery where vision is particularly poor, detrimental. But of course, another possibility is that there is actually richer spatial information about sounds that is projected back to visual cortex, possibly across the entire visual field. These are all, I think, important and interesting questions for future research. But what I want to focus on today is the link to selective attention. And in particular, the question whether these biases that we're seeing are due to enhanced processing at the sound's location or kind of suppressed processing at the other location. And so many theories of spatial attention, prominent theories of spatial attention, have proposed that attention operates via excitation at the same time via inhibition, based on the idea that visual processing is a strictly limited resource. And so if processing gets enhanced somewhere at one particular location, it basically consequently necessarily has to be decreased elsewhere. And there's a lot of evidence that kind of in top-down attention, so when we tell participants what and where to attend to, that we find kind of these two flip sides, excitation and inhibition. But very little is known in the context of these cross-modal tasks, and also in the context of kind of exogenous attentional orienting more broadly. So if we think about these salient sounds as kind of an bottom-up salient cue that orients our spatial attention reflexively to the left and the right side, that such that captures our attention, that could be kind of thought as a cross-modal exogenous attention. And so I think it's important to explore this question, whether these effects are really due to excitation or inhibition. And so this is what I'm going to focus on now in the second part. And I'm going to do this in three different ways. We're going to look not only at ERPs, but now also at alpha oscillations, which are a very prominent marker of studying suppressive effects in particular. I'm going to try to link these different neural measures to behavior and see if we can kind of learn from the pattern what type of behavior they predict and whether they can tell us something about excitation or inhibition. And then lastly, I'm going to show you some very recent experiments that we ran trying to isolate neural enhancement and suppression using experimentally. So occipital alpha oscillations are sensitive to visual processing. It's a very prominent, well-known marker, and it's very well known that this 10 Hertz oscillation around 10 Hertz is associated with kind of the amount of visual processing, very broadly speaking. So decreases in alpha power have been associated with more visual processing, and increases in alpha power have been associated with less visual processing. And there's kind of prominent theories that really suggest that these alpha changes, in particular increases in alpha, reflect kind of top-down control of suppressive mechanisms in particular. And so in the task that I just showed you, really in all of the tasks that I just showed you, we can not only look at the event-related potential, what I've done so far, but we can also look at alpha activity and see if we find changes in the occipital alpha rhythm that have been very strongly associated with these kind of biases in visual cortical processing. And so I'm just going to show you here one example of this last task that I showed you, where we just present these kind of random stream of sounds. And we're going to again be time locking now our EEG activity to these task equivalent noise bursts appearing on the left and the right side. But instead of looking at the ERPs, I'm going to be now showing you time frequency plots. So here on the x-axis again, time, 0 is the onset of the sound. And then on the y-axis, you now see not just amplitude, but you see the frequency. Okay, so from kind of 0 up to 30 Hertz, what I'm plotting here. And the different colors denote how much activation there is. So blue is a decrease in alpha power, and red would denote an increase in alpha power. So here I'm now showing you the time frequency plot for the activity recorded over the hemisphere ipsilateral to the sound. And you can see that we kind of see a small decrease in alpha power around 10 Hertz. Now I'm showing you the activity contralateral to the sound, where you can also see kind of this clear blue line around 10 Hertz of activity. It's a little bit easier to see if we do the difference between these two time frequency plots, which then very clearly shows that we have a stronger decrease in alpha activity over the hemisphere contralateral to the sound relative to ipsilateral. And you can see here that this decrease in activity really happens around the same time window that we saw for the ACOP, so around 200 to maybe 500 milliseconds. Of course, there's some temporal smearing here, but it really closely actually resembles the time course of this ACOP component that we measured. And so I just want to point out a few things here about this data, right? So we see that alpha activity contralateral to the sound decreases relatively quickly and transiently. And relatively quickly and transiently, I'm here thinking about a ton of work on endogenous spatial attention that also finds these lateralized biases, so when participants are told to attend to the left or the right side, but usually they occur much later in time, around 500, 600 milliseconds is maybe when these alpha changes are beginning, but here they're relatively fast, really suggesting that alpha is not necessarily a sluggish response, but can actually be triggered and elicited rapidly by salient peripheral stimuli. And these alpha changes, again, just to remind you all, are elicited by task-irrelevant peripheral sounds, right? So we're looking at activity here, the occipital algorithms over visual cortex, but these changes are triggered by sounds that are presented about 100 milliseconds beforehand. And so they're not due to kind of this top-down control suppressive mechanism that often alpha changes are related to. And one other point that I just want to kind of allude here point people to is that what we see here is that these alpha changes, decreases are bilateral. So we see decreases ipsilateral and contralateral with a stronger decrease over contralateral visual cortex. And so this is actually very different from many, many reports and kind of top-down endogenous attention that very often report changes that are due to ipsilateral increases in alpha and or contralateral decreases. But what we see here is really just that overall kind of alpha is decreased, just biased to be more decreased contralateral to the sound. And I think that could be potentially suggesting that there is kind of a difference in how these alpha modulations occur between different types of tasks. Okay, so overall, the fact that we do find these decreases are consistent with the interpretation that what we're seeing here is a visual enhancement, right? If we think about alpha decreases really kind of reflecting in increases in visual processing, not suppression of visual processing, then this pattern of data is consistent with the idea that the sounds increase or enhance cortical processing in visual areas. Now let me move to the kind of second point here. So in another task that's really similar to the ones that I showed you, we try to link the ACOP and also alpha activity trial-by-trial performance. And so here I'm showing you the task that we're using. Again, it's really similar to the task that I've showed you before. In this case, it's not a Gabor patch that participants are discriminating, but it's an L versus T task. But really the critical difference that you have to pay attention to is here that we expanded the time a little bit between the sound and the visual target. So here we have 300 milliseconds between the sound and the target, much longer than what we usually use. Usually it's about 60 milliseconds, much, much more rapid than that. But if we only have 60 milliseconds, that does not allow us to link trial-by-trial variation in performance to trial-by-trial variation in the ACOP or in alpha activity. But here we have just enough time basically to look at neural activity and behavior within the same trial. And so this allows us now to split up the trials based on whether they were validly cued or invalidly cued and whether participants responded correctly or incorrectly, and kind of look at how magnitude of ACOP or alpha activity relates to behavior. And so let me walk you through the logic here. The prediction here is that if the ACOP reflects primarily enhancement, you would expect to find a larger ACOP, so more positive activity in the hemisphere contralateral to the cue relative to ipsilateral, preceding correct, then incorrect discriminations for validly cued targets only, but not necessarily any difference for invalidly cued targets. Conversely, if the ACOP reflects primarily suppression, you would expect to find a larger ACOP, so more positive again in the hemisphere contralateral to the cue, preceding incorrect then correct discriminations of invalidly cued targets. Okay, so really what this means is that we basically want to kind of figure out here what the ACOP predicts, right? Does it predict performance on valid trials or invalid trials? And from that, we can make some inferences about where the action happens, right? So whether it facilitates processing at the validly queued location or kind of hurts performance at the invalid queued location. And obviously, you could also imagine a mixture of these two, right? I'm just kind of putting up here the two most clear results. And so let me show you what we find in this paper. We find data consistent with the excitatory or enhancement account, such that the magnitude of the ACOP, here plotted on the y-axis, predicts performance only on validly queued trials with no difference on invalidly queued trials. And so this suggests that the ACOP enables more effective visual discrimination at the queued location and implies that the ACOP does not necessarily reflect suppression at the unqueued location. And so interestingly, we find exactly the same pattern of data for our alpha amplitude. So here alpha and ACOP really seem to kind of go together. We don't always find that in all of our experiments, but here it really seems to be the case that lateralist alpha predicts performance, again, only for valid trials, possibly suggesting that these alpha changes reflect facilitation at the attended location with no suppression at the unattended location. And so I think this is, while this is rather indirect evidence, it at least points to the fact that possibly these two neural markers are really related to kind of enhancement of neural processing at the location of the sound. Okay, so now lastly, I want to provide one more piece of evidence where we try to experimentally isolate this neural enhancement and suppression, an experiment that was just recently finished by my graduate student, Jonathan Keefe. And so in this task, we're going back to our lovely Gabor patches here with the same task essentially that I showed you at the very, very beginning of the talk. So participants performing a discrimination on these oriented gradings. We're presenting our kind of standard left and right sounds, but now we're also adding a central sound. And so the central sound is basically supposed to serve kind of as a baseline here. Really, the idea is that the sound is matched in all low-level features and also has the same kind of temporal predictability that these left and right sounds have. But the only difference is that it does not cause any spatial orienting response, right? So the sound comes from the center. It's very clearly kind of, you don't orient left or rightward. It should not cause any lateralized changes in visual cortex, but it might still cause changes just due, and many studies have shown just even central sounds activate or can elicit activity in visual cortex. And so really want to get kind of at this general excitatory or whatever response that these central sounds have, but then compare it to these spatially peripheral sounds where the orienting response happens. Just like in all other tasks, the sounds are not predictive at all of where the visual targets are about to appear or when they're appearing. We actually have here different types of SOAs. So either the visual target appears right after the visual cue, the auditory sound, sorry, and that allows us to look again at behavior as usual, but then we also have trials where there is a rather long delay, around 600 milliseconds between the sound and the visual target. And we also, again, have trials where there's no target at all. And so these latter trials, the long SOA and target, we can use to look at the EEG activity without the contamination of the visual target. And so that's what I'm showing you here. So you're fairly familiar now with these waveforms. We're again time locking to these peripheral noise bursts. And here what I'm just showing you is again the waveforms to the left and the right sounds. So we find again here in this highlighted region the ACOP, similar distribution as I've shown you before when we look at the topographical map. So really the main question is now, where does this central sound fall, right? Where does the waveform of the central sound fall? So if the contralateral waveform differs from the waveform elicited by the central sound, for example, it is more positive than the central sound one, this would be consistent with the contralateral cortex getting enhanced relative to that baseline. And likewise, if the ipsilateral waveform differed from the central tone or central sound, for example, it's more negative, relatively speaking, this would suggest a role of suppression, so less activity ipsilateral relative to this baseline, to the central sound. And so what we find is that really the waveform to the central sound kind of lies on top of the waveform to the ipsilateral sound. It's even a little bit hard to see. Here I'm plotting to make it a little easier the mean amplitude for our three conditions, so contralateral and red on the left, followed by ipsilateral, and for the central tone. And so this really suggests that kind of all the action happens, all the difference happens in the hemisphere contralateral to where the sound was, with no additional kind of costs ipsilateral. And so this is broadly, again, consistent with this idea that the sounds primarily enhances visual processing at its location, but does not have any kind of costs or suppressive or inhibitory effects in the opposite hemisphere. And so this pattern of neural data that we find here very nicely matches our behavioral data in this task, which I'm now plotting here. So again, this is just proportion correct for our three trial types, valid, invalid, and central. And we can also see that when we're just kind of looking at the cost benefits of these sounds on visual performances, what we see is that there's really just benefits for the validly cued trials relative to invalidly, and then our baseline, the central sound. So consistent with our interpretation of the neural data, that really the sounds are primarily facilitating visual processing and not hurting visual processing at the unintended location. And so kind of looking at these different, right, so in the last part now I showed you that this like stronger activity contralateral relative to ipsilateral, and the central sound is also consistent with enhancement. So I think we kind of are getting pieces of evidence looking at the data in different ways and looking at different types of experiments, suggesting that these biases that we see in visual cortical areas are primarily driven by enhancement, neural enhancement in visual areas. So to sum up, I started with showing you that sounds influence visual perception, in particular by increasing visual discrimination accuracy. That's the main task that I talked about today. And then I showed you a series of studies where we found that salient sounds very robustly bias visual cortical activity, and that these biases even occur when there's no visual targets presented ever. So when participants are in completely auditory contexts. And the kind of last part of my talk, I showed you some evidence suggesting that the sound-induced effects on visual performance are largely driven by excitation or enhanced neural processing at the sound's location, with no effect really at the unattended location. And I think this might have important implications when we kind of more broadly think about spatial processing across modalities, but also if we kind of compare effects of exogenous attention, which I think is basically what we're studying here in a cross-modal context, and endogenous attention. In endogenous attention, many studies have shown, and there's still currently a lot of ongoing work showing that endogenous attention enhances and at the same time suppresses information processing at unattended location. So there might be a really interesting difference here of how these two types of attention operate in enhancing visual perception. Okay, so I want to thank the wonderful people that helped with all this work, and particular shout out to Jonathan Keefe, my graduate students, who's been working a lot on these projects, and also all the other co-authors of the work that I presented today. I want to thank my lab at UCSD and at Dartmouth, and I want to thank you all for listening.
Great, thank you very much. Wish that the entire audience could clap, of course, but this is...
Thanks to you for clapping.
Yes, that's fascinating. The first question that I have, and then we can get to some questions from the audience as well. So are people hearing this, and maybe you mentioned this, through speakers in front of them or through headphones?
Yeah, I think I mentioned this very, very briefly. So it's all through speakers that we mount to the sides of the monitor. We have not done this with headphones. I think my intuition is that it's actually important that these speakers are kind of an external space, so that you really kind of get this. I think it's very important that you kind of get this like external space perception. Of course, you can titrate it with headphones. So I think it could work with headphones, but we actually haven't tried it.
Yeah, how selective do you think the spatial location is? Like, in other words, how important is it that like the sound source is directly there versus if you had like a speaker like perpendicular?
So we know from many pilot experiments in the lab, and just kind of like trying things out differently, that it's actually really important that the visual target location overlaps very precisely with the sound location. I think that might vary by where you are in the visual field. So in all of those cases, and maybe I didn't emphasize that enough, all these sounds and the visual stimuli are really far out in the periphery. So we're talking about like a visual angle of like a minimum of 24 degrees to get these really clear and reliable spatial effects. So when you move closer, the behavioral effects, at least we know, kind of attenuate. And so there could be something about really this like peripheral part of the visual system that I think interacts maybe with like the auditory system basically particularly well, possibly because visual perception is kind of more poor, right, in the periphery. But in these like peripheral locations, it's really important that we have very close mapping. So we're actually adjusting the sounds. We have the speaker kind of next to the monitor, but we're actually adjusting the sound to be perceived exactly to where the target is by adjusting the volume between the two speakers. And that seems to be important to kind of get the most reliable behavioral effects, at least. So there seems to be a pretty close mapping, maybe surprisingly close.
Have you tried it with using, like, for example, the ILD instead of like a speaker?
No, we've only done it with speakers. We haven't tried any of the other ones. And I think, yeah, I think it could work if you, yeah, I don't know. I'm really not sure, actually. I'm always skeptical, so that's why we never tried it, I guess.
Yeah. Another question about the potentials. I noticed the very first EEG you showed, the ERPs rather, there was not a large positivity associated with the auditory stimulus, whereas in the later ones, there was this very sort of clear positivity at around 200 milliseconds. And I was wondering if you think that that is somewhat equivalent to like a visual P3, but occurring earlier. I know that everything in the auditory domains kind of shifted a little bit earlier.
Oh, I see. So early, you mean like early in time in the ERP waveform, right?
Yeah.
Yeah. So, I mean, we see when we look over auditory cortex, we see the lateralized effect, a small, much smaller one, but we see a lateralized effect as you would expect in the auditory N1, right, around 100 milliseconds. So when we look over auditory cortex, we see a small contralateral increase in the auditory N1 component, and then really nothing happening afterwards. So then it's completely just over occipital scalp areas. Right. I don't think this is a visual P3 because it's not related to a visual.
Right. So I was thinking like the sort of analog of.
That's the right way. I mean, we think it's like a new component that we found, basically, and it's not really related to like the visual P3, but it's true that it's rather late, but it's not triggered by visual stimulus, it's triggered by a sound stimulus. And so it also, like, the pattern looks pretty different. It's kind of longer lasting, right? It's really like 200 to sometimes 300 milliseconds long. So it's almost slower than any type of P3 component.
Oh, sure. Yeah. So the ACOP, right? That's the sort of bilateral difference. I was thinking of it looks like there's also a, not difference wave, but like a bilateral.
Oh.
That big, huge peak, right? Which is bilateral.
Got it.
That kind of might be the equivalent of sort of the P3 to a visual event.
Maybe the P3, I think we've been thinking about more because we see this really interesting difference that I just very briefly, I think, pointed out between when the sounds actually carry predictive value versus they don't carry predictive value. And we see that the overall waveform shifts when they carry predictive value about the location, that it becomes more negative overall. So we have this kind of like upward shift of the entire waveforms. And I think that might be related to kind of like sustaining attention maybe at that location, right? Because they actually participants are trying to use the sounds. And so it could be, I don't know if it's like the P3 or maybe more something like a sustained activation negativity that kind of tags that particular location and kind of sustains there over time. And is really related to more these attentional, kind of endogenous attentional processing. That makes sense. But I think we would have to do more manipulations like that to actually kind of get at the overall shape of the waveform, which we just haven't done a lot. We really just have this one comparison where kind of popped out to us that the predictability of the cue actually shifts these waveforms around.
Yep. Well, let's dig into some of the questions here. The first one is, could the ACOP be a general spatial expectancy signal in the parietal cortex, I guess, relative to something that's specific to the auditory?
Yes, and I think that's actually likely the case. I would add that we don't think it's just the parietal cortex. We really think, actually, in the really early studies that we did, we always found mostly occipital activity. We also did like some distributed source modeling and dipole modeling. Of course, these are all just models, but it always really fell into visual cortex in particular. But we often kind of see this additional parietal component. So it might be a mixture of both parietal and visual, I think, components that are inside the ACOP. And we actually ran a study just looking at visual cues, and we find essentially the the visual counterpart to the ACOP, which we call the VCOP, and it looks really strikingly similar. It's much more difficult to interpret because, of course, when you look at activity elicited by the visual peripheral cue, you're going to get early difference in the waveforms just triggered by the visual response itself, right? So you have big difference in the P1 component, N1 component, but all of that is followed by a kind of slow wave, contralateral larger than ipsilateral, exactly resembling kind of the results of the ACOP. And we have a recent paper kind of comparing these two components, suggesting that this is a general kind of marker of exogenous orienting. So I think that's true. I think it's just much, much harder to interpret in the visual domain, basically.
Yeah, it's nice that you have the auditory stimulus, makes it a lot easier.
Yeah, that's right. That's one big disadvantage of auditory stimuli.
Yep, that's cool. Next question from in the Q&A is by Jan Hartung, and he asks, he says, cool talk, thanks a lot. So you said that the sound might prepare visual cortex because in this specific context, the sound is associated with a visual stimulus most of the time. If you decrease the proportion of trials including a sound, does the effect on visual cortex disappear at some point? So I guess like in the extreme, you might just have like one trial. Like, do you think it would work in that case, I guess?
So if we just decrease the proportion of sounds overall, is that the question?
Right. So that the sounds were very rare.
So I actually think it would probably increase the ACOP because I do think it has a lot to do with just kind of how salient these sounds are. We try to make them so salient that they basically stay salient throughout the whole experiment. Even though people have been in there for an hour, they still kind of capture attention and are salient enough to trigger the ACOP. My suspicion would be if we made them even more rare, that actually the ACOP would increase because they kind of engage, they like basically capture attention more, right? Because I really think it's an attentional effect. And we have not done, of course, we have the data to actually look at kind of like trial by trial variation of the ACOP to see if potentially if we happen to have 3 left sounds in a row, does the third one actually produce a smaller or bigger ACOP, right? To kind of look at these sequential effects. Jonathan is actually currently working on that, but we don't know the answer to it yet. We haven't really done it systematically. And so we're kind of looking at the data that we currently have to see if we find effects that I think could get at that question. But anyways, I think my intuition would be the other way around. I think actually more rare sounds are going to produce a larger ACOP because they engage kind of attention more, essentially.
Yeah, no, I completely agree with that. I think that they would just sort of pop out.
Yeah.
Someone asks directly in the chat, someone was wondering if the same component would be there without any audiovisual integration. And I think that you showed that. Is that true?
Yeah, I think yes, yeah, because we have these tasks where participants do never see a visual target. And so we even then see the ACOP, right? So if they just hear this random stream of sound, I think that's kind of the strongest experiment that we have showing that, where these sounds are just all in random order, participants never see visual stimulus, and we get basically the same sized ACOP that we find in the other experiments. So I think this is basically suggesting that there's a direct kind of interactions in spatial processing between audition, the auditory system, and the visual system. We don't need the visual target, right? So it really gets, that's probably what the question is about, like, do the broad multisensory integration question, do we have cross-modal interactions even without kind of the second modality being present? And I think we do have evidence that that's the case, that the visual target or visual stimulus does not need to be present for these interactions to show.
Right. We have a question from YouTube. Aparna Pandey asks, do you think that the facilitation on visual stimuli would be possible for more meaningful auditory sounds or related sounds? So like, for example, a case where the auditory sound is related in some way to the visual stimulus?
Yeah. So, I mean, there's a bunch of work kind of on using different, how like features, right? So for example, like high pitch sounds are associated with, I don't know, brighter stimuli. So maybe the question is getting a little bit like, that's not really meaningful, but there's a bunch of research kind of showing that there's like these correspondences where also behavioral performance is facilitated. Yeah. And one of my grad students, Jenna, she's really interested in these correspondences, and she also re-replicated these behavioral effects. I think that people are finding, but I think those are fundamentally different mechanisms. I really think this here, these spatial connections, I think are really robust, possibly even hardwired. And I think as soon as you kind of move into maybe looking at feature correspondences, they're probably happening at, I think, like at a higher level essentially, right? And I mean, we are doing a little bit of this work, so looking at different features, and another grad student of mine is working on really high level stimulus, so really looking at how kind of auditory, the sound of an auditory scene, for example, right? Like listening to a football stadium or something when you walk in a football stadium, how that affects the visual discrimination of real world objects. And behaviorally, we find associations between all of them, but I really think that that happens at a like completely different level, right? Those are like semantic associations probably that are really well learned. And so I don't know exactly, I think at this point, how to compare them. I do think those associations definitely exist, but I think here we're looking at something like really low level of kind of looking at like spatial maps across sensory modalities, which I think is just a more possibly hardwired mechanism. I don't know if
that makes a lot of sense.
Yeah.
I was wondering also if you've tested how specifically in time the relationship holds, right? So normally, like with exogenous cueing, you see a sort of very brief window in which it's effective on the visual domain, right? How does SOA affect here?
So we've the ACOP component and also the alpha modulations, they really basically always start exactly at the same time. I mean, more or less around 200 to 240 milliseconds. They do, so when we do manipulations such that the sound, for example, becomes predictive of the target location, they basically can last longer. Actually, the ACOP not at all, but alpha. So alpha activity seems to be modulated by kind of the top-down goals in addition to this reflexive oriented response, whereas the ACOP really seems to be primarily driven by just kind of this exogenous part. Mm-hmm. And also behavioral, we find actually really similar types of effects in terms of timing as you would for visual cues. So I think this kind of exogenous attention is probably like very broad across these modalities. Mm-hmm. Something that I didn't point out is, but the ACOP time window corresponds really, really nicely to this time interval of kind of broad exogenous cueing effects, right? That we usually kind of say peak at around 100 milliseconds or last up to 150 milliseconds. And so the ACOP, it's a little bit complicated because you actually have to like subtract how long it takes to reach visual cortex for a target stimulus, right? Yeah. But it really falls if you present the sound and 100 milliseconds later you present a visual stimulus, basically it takes another 100 milliseconds or 80 or so.
Yeah,
so that visual stimulus is actually in visual cortex, and that's exactly when the ACOP starts. And then it lasts around 200 milliseconds. So it's really in that time window that many, many visual exogenous studies have shown where the peak is of exogenous attention. And so that's also right why this link, I think, to spatial attention and exogenous attention more broadly makes a lot of sense.
Right. So we've only got like perhaps another minute or so, and we're going to cut off probably pretty abruptly. But let's ask one more question here. Which is whether you think that these findings translate in some way to blind or visually impaired people.
Oh yeah, we actually have a study on that that was conducted by Mori's lab at the IIT in Italy. And so they tested blind subjects, and basically similar paradigms that I showed you today. And so interestingly, in congenitally blind people, so those that never have no visual experience whatsoever, we find that the ACOP exists. If anything, it's bigger than in non-blind people. Really interesting finding, really suggesting that there's some really like hardwired, interesting relationship, I think, between auditory processing and the visual cortex that doesn't need the visual experience for that wiring to occur. And so yeah, we have those results. They're published. People can look it up. Monica Gori is the last author. Yeah, I think it's really interesting and really suggests that there's a kind of very robust wiring between these two systems in terms of spatial processing.
Is that Gorey as in Gorey?
Yes.
All right. Okay. Well, thanks again.
Thank you.
It was excellent.
Thanks for having me.
And thanks also to the audience.
Yeah.
Thanks
everyone for listening. Have a
Bye,
good
everyone.
day.
Bye, Viola.
Bye-bye.
Nice to see you again.
Remind you that if you have any thoughts or comments, to feel free to tweet about the conference and to use the hashtag NMC3. Okay. So we have a fantastic lineup of speakers today. We're going to be learning a lot about object recognition and visual search. This is an interactive session, so the talks will be short. The speakers will take about 5 minutes to introduce their research, and then there'll be plenty of time for questions. So you can use the chat to send messages to everyone, to share links to papers or other resources. that are relevant to the session. And if you want to ask questions, to please use the Q&A tool. Because it's an interactive session, you can ask your questions on screen. So if you'd like to ask a question on screen, then please just type on screen at the end of your question, and I'll be able to move you onto the panel. But if you prefer not to, then I'm very happy to read out your questions for you. So please ask lots of questions. We'd like to get a really good discussion going today. Okay. I'll start the session today then by introducing our first speaker. The first speaker is Thomas Cherian. Thomas is a PhD student in the Center for Neuroscience at the Indian Institute of Science, and he works in the Vision Lab with SP Arun. Thomas's background is in psychology, and he's interested in understanding how complex visual stimuli are encoded in visual cortex. And today he's going to talk about amodal statistical completion of occluded objects. Okay, Thomas, if you'd like to start.
Yeah, I'll just share the screen. Can you see my slides? Okay, so yeah, hi everyone. I'm really pleased to be able to share my work regarding the representation of occluded objects. Excuse me? Shall I go ahead? Okay.
Did someone say something?
I think David was unmuted, so just muted him. So everything's fine.
Okay.
My fault, sorry.
Yeah. Okay, so if you see occluded displays like this, let me just get my pointer here. So if you see occluded displays like this, we don't think that this is basically a cut object placed next to the occluder. And this is kind of what previous studies have already shown. This is kind of obvious if you consider the fact that the cut object has extra features, which is this cut contour, and which obviously makes the this display different from the occluded display. Now, we are asking a different question. We are asking which of these two objects do we think is behind the occluder? And we felt that this object with the curved corner is more likely than this object with the sharp corner. This is because the occluded display, actually, the visible portions of the occluded display also have curved corners. And similarly for other displays with sharp corners. Now, it doesn't make sense to make a direct comparison between the occluded displays and the separated displays because a lot of features have changed here. Instead, we thought it best to compare the arrangement of multiple shapes in your perceptual space. Now, to get the measurement of objects in perceptual space, we measured the perceptual distance between pairs of stimuli using visual searches like these, where the task of the subject's task is basically to identify the oddworld target and indicate its position with respect to the vertical red line. Now, we can simply ask if the pairwise distances between occluded objects matches better with the pairwise distance from likely or unlikely displays. And we hypothesize that the match between likely and occluded arrangements will be more than the match between unlikely and occluded arrangements. And we can see that the occluded dissimilarities correlate better, or the likely dissimilarities correlate better with the occluded dissimilarities as compared to the unlikely dissimilarities. And furthermore, if we use the likely dissimilarities to predict the occluded dissimilarities, we get a significantly smaller error than when using the unlikely dissimilarities to predict the occluded dissimilarity. Now, the difference in the correlation doesn't seem that large. So to visualize this difference better, we took the dissimilarities between shapes and scaled them down to 2 dimensions using multidimensional scaling to get a visualization of the perceptual space. Now, the objects are just marked by numbers here, but we can see that the arrangement of these objects or these occluded objects matches better with the arrangement in the of the likely objects as compared to the unlikely objects. And similarly, the same can be observed for all stimuli in my experiment. So from experiment 1, we can conclude that the representation of occluded displays matches better with likely displays because of the consistent curvature or lack thereof. Now we went a step ahead and wondered if the number of protrusions of occluded displays like these is represented in a more likely way, where the number, frequency, or the frequency of the protrusions is dependent or is determined by the number of protrusions of the visible portion of occluded displays. So we did the same kind of experiment as before. And again, we can see that the likely dissimilarities correlate much better to the occluded dissimilarities than unlikely dissimilarities. And again, the likely dissimilarities are a better predictor of occluded dissimilarities. And the same way, if you look at the arrangement of these objects in perceptual space, we see that the arrangement of the occluded shapes matches much better to the arrangement of the likely shapes than compared to the arrangement of unlikely shapes. So from experiment 2 also, we can conclude that the representation of occluded displays matches better with the likely displays, again, because they have consistent features. And in conclusion, this means that amodal completion is not simply the extrapolation of occluded edges. It is amodal completion actually extrapolates the statistical properties of these occluded objects. Thank you.
Thanks very much, Thomas. That was a great talk. So I'll wait and see if any questions come through. So please.
Sure.
Please remember to ask your questions, and if you'd like to ask them on screen, just to write on screen beside your question. So could you explain a little bit more about the perceptual distance metric?
So the perceptual distance, we're basically we do a visual search basically, and there's an oddball target in this visual search surrounded by distractors, which are all the same object. Now the idea here is, if the target object is represented in perception very dissimilar or differently from the distractors, that means that these two objects are very dissimilar, and hence you will have a very short reaction time. Conversely, if they are very similar in perception, you will have a large reaction time. And we just see that taking the reciprocal of the reaction time is a much more consistent measure of this dissimilarity.
Thank you.
Yeah.
Okay, I think we have a couple of questions. Okay, this is from Ilyan Budno. Sorry about that. These properties seem to be based on shape and less so on texture. Would you expect the same if varied texture properties like color?
So when we're working with color, I think the bigger problem, so if you're going to vary the color, you kind of change some aspect of the object itself. But even...
Sorry, she also added or roughness or hue, if that helps.
Okay. Roughness is in some sense similar to what I've done with the numerosity set. So I've only shown a few objects here, but we have objects which have like random protrusions. So they seem like very spiky, rough, spiky balls, and we're just changing the number of spikes, which are like not in any pattern per se. So roughness is kind of what we've done in the numerosity set. But I'm not sure how color or hue would behave here, because there's a good possibility that your perceptual system just ignores that, or maybe the shape perception might just or like the shape properties might overpower the color properties or something like that, because we've only used grayscale stimuli here. So yeah, I'm not sure if that answers the question exactly.
Thank you.
I think there's a question regarding, so the results are not from brain scans here. This is just behavior actually. And yeah, in the future, I do plan to look into neural responses from behaving monkeys. And that's the upcoming plan, where we're going to look at trying to see if monkeys can actually tell me what they see behind the occluder. So I'm currently training monkeys to do tasks. And basically, we will record neural responses from the IT cortex and see like if neural activity predicts behavior or vice versa.
Wow, that sounds really exciting.
Yeah.
I was just wondering, how large is your stimulus set?
So I have 16 shapes across in each of these experiments. but each shape is presented either as occluded or as a likely and unlikely completion. So each of these, so it's like essentially I would have 16 into 3 shapes here.
Okay.
Yeah.
Can you say a little bit about what you might be expecting to see when you record from IT? Are you thinking that will sort of line up with the perceptual space that you're seeing now?
So I've not really thought about like what response or what activity in the from the neural space I want to look at. But primarily right now, my aim is to look at behavior, and we do see some promising results where monkeys, without being trained, at least for us, they seem to have some preference for completed displays or the likely representation. But I guess, so this response is not very consistent, as in they don't say occluded displays are similar to likely displays all the time. But it is definitely, they don't say that occluded displays are equal to unlikely displays. So the idea, what I think would be interesting is to look at, okay, there is a pattern of behavior here which is not entirely consistent, as in they don't always say occluded displays are likely. So I'm guessing there's a, there's some neural representation of this behavior, right? So that's probably something I might be interested in looking at. And of course, I can look at how each object or how multiple objects are represented in the neural space also, and possibly I might get the similar kind of arrangement that we saw. And instead of perceptual, it will be in like neural space or so. Yeah. Does that answer your question?
Yeah, definitely. I'm really looking forward to those results. I think it's...
Yeah, me too. I think Mohammed has a question here.
We have one more question here. Have you tried non-occlusion control, as in 2 disjoint objects that fit perfectly?
Could you elaborate on that question, Mohammed, a bit? I'm not quite sure what you mean by 2 disjoint objects that fit perfectly. If you mean like cut displays where I have the occluder and like the object just shifted with like a cut edge, that has already been done by many people, but we feel that that's not exactly the correct way to do this experiment, because if you look closely, there are certain issues in that kind of experiment design where you directly compare, I guess, disjoint objects, or as Mohammed refers to here. Yeah.
Great. Well, we'll thank Thomas again for a fantastic talk and a really interesting discussion.
Yeah.
So our next speaker today is Dr. David Nicholson, and he's a postdoctoral fellow at Emory University working in the laboratory of Astrid Prins. And his talk is titled, Neural Network Models of Object Recognition Can Also Account for Visual Search Behavior.
Thanks, Elaine. Let me go ahead and share. Are you all seeing that okay?
Yep.
Cool, thank you. Not that. Let me click on the right button. Okay. Right, so I'm gonna tell you about some work we've done in the Prince lab using neural network models of object recognition to account for visual search behavior. And when I say visual search, you might be thinking of some behavior out in the real world where you're seeing natural scenes and you're trying to answer some question like, is there a person there? And you have all these context clues, like there's a bench, so I'll look there. I won't look in the sky because I don't expect people to be in the sky. So we know people perform this task really efficiently using a lot of this contextual information, but still there are limits on our ability to perform that task. And for a long time, people have asked, like, if I examine what factors limit performance, does that tell me how the brain solves this task? And as Thomas has just done a great job of showing, helping me introduce this because we have zero time to provide nuance, people have historically used these really simplified stimuli to investigate visual search in lab tasks. And there's a lot of good reasons to do this if you want to ask questions about, like, does occlusion matter? If you want to ask these really controlled questions. And so I don't have time to get into the nuance of these stimuli or the results obtained with them or the models that are based on those results. But the main issue that we want to address is that the way that the prevailing models are specified assumes that the inputs will be some discrete set of items where those items are described by human-defined features like color, luminance, orientation. And it's just unclear how you would extend those dominant models to natural images where you can't just easily extract some description in terms of color, orientation, number of objects that's there. So that's one reason we want to use artificial neural network models, which are image computable. So you can give them any image and get some answer, which at least gives us a hope of reconciling results across all these different tasks that use different stimuli. And the other reason that we want to use artificial neural networks, we're motivated by a proposed computation untangling, which we think could account for visual search behavior across these different stimuli. So let me give you my attempt to define untangling in like 20 seconds. So this is proposed by people studying object recognition. If you think about the low-level features that your retina extracts, you have this high-dimensional, low-level feature space. And the sort of idea is that your ventral visual stream performs some series of nonlinear transformations such that you map this low-level feature space to some neural space, like inferior temporal cortex space, where your brain can find some separating hyperplane that lets you discriminate between two objects. And there's good reason, based on previous literature, to think that this might possibly account for visual search behavior. So I promise we didn't just pull this out of thin air, but again, I have zero time to touch on that actual literature. But let me just tell you what we've done. So we take these AND models of object recognition, hold all the parameters constant, except for the last layer, this sort of decoding output layer, which we train with something called transfer learning on these simplified stimuli that have historically been used. And when we do that, we see what are called set size effects. That's a technical term from the literature. So you see that as the number of targets and distractors increases, that's the set size, there's a decrease in accuracy. So you know, this is a hallmark effect in the literature. So if untangling can provide this effect, then that suggests that it could account for this behavior. In contrast, if we take the same models, but without pre-training them for image classification, which is what makes them object recognition models, instead we just train from scratch on randomly initialized weights, we see that they can, in some cases, find near perfect, they get near perfect accuracy. They can find some solution that lets them perform almost perfectly. Not always, but this is like an existence proof. There is a solution. They can find it if they're not pre-trained for object recognition. And this holds across various types of models. If you train them on a bunch of stimuli, it's not perfect, but this is like a proof of concept that untangling could account for set size effects seen in these experiments. But you know, there's more work to do. This is just the beginning of a story. But more what we care about is what I alluded to in the beginning. We want to account for effects across these different stimuli types. So to get at that, we took this visual search difficulty data set, which assigns difficulty scores to images from Pascal VOC, which is like a benchmark image classification data set. And the scores are derived from human reaction times, people searching these images. So what we did is we performed the same transfer learning experiment where we just hold the parameters constant and then just train the last layer to do this task. And when we do that, we measure performance on the test set where we binned the images by the difficulty score, and we measure the accuracy within that bin. And what you can see is we find this inverse correlation, whereas the difficulty score increases, the accuracy decreases. So again, this is just suggesting that this untangling could account for aspects of, limitations on visual search performance. And one last thing really quick, what's nice about this data set is there's multiple targets labeled, we did multi-label classification, so we can ask for the same image, are multiple targets detected? This is not a solved problem in the same way that single label classification is, but for models that perform well on the test set, we can again see that there's this inverse correlation between difficulty score and accuracy. So based on those results, we think that so far, untangling does have the potential to account for these limitations on visual search performance across stimuli types. I'm on Twitter as Nickel Dave, so I'll tweet out these slides later if you want to replicate our findings in the safety of your own home while social distancing. I'll tweet out these links as well. And then Elaine asked to put up some discussion points. Thank you, Elaine, for preparing us. Maybe if I read the emails, then I do a better job of preparing myself. But I do have some points here to talk about, like, as I alluded to, if we're optimizing for object recognition, is that what limits our ability to perform visual search? Can think about this within this sort of deep learning framework for neuroscience. Or you might argue that we just haven't figured out the right way to optimize our networks for visual search. You might talk about other aspects of visual search behavior that people measure, such as reaction times, and how we would account for those. And then you might point out that the last task I looked at is effectively the same as an object recognition task. So what does it mean that those tasks that we use in the lab are very similar, even though we're trying to get at what we think of as different behaviors out there in the real world? And then you might ask why I completely avoided using the term selective visual attention. And I'm happy to talk about that as well. Let the questioning begin. Thank you.
Thanks so much, David. That was really interesting.
Thanks.
So yeah,
could I ask a question?
Yeah,
sure.
Yeah. So I think I missed exactly, and it's kind of on that last point that you said of like the task at the end seemed very similar to just an object recognition task. And so I was unclear just literally on how you were going from like a VGG or some deep network to doing a search task. Like what exactly, how did you make that linking?
Right. So in the case of the first task with the simplified stimuli, there's two output units. So it can either say target present or target absent. So it's classifying the image as target present or target absent. In the case of the Pascal VOC data set, I'm really just doing image classification. So if it, and we trained it two ways. For single label classification, we tried doing either just train it to report the largest image, which you can do using the bounding box, the largest object, or we tried using just some random object during training time. And then you can just go back and ask which ones does it report as is present. And if you only look at the images which have only a single label, which is like a majority of the data set, then you can get the accuracy for just those images. Or you can do the multi-label classification, which I won't nerd out too much about the details, but then you can ask like which ones does it report as present.
So, and I guess more broadly, like what is the difference between visual search and object recognition or object detection? Like I'm not familiar with like visual search literature that well. Like is there, yeah, how are they the same? How are they different? Or is part of the point of your work they're kind of very similar in some way? Like I don't know. Like if you could talk on that, I'd be curious.
Yeah, I mean, my take right now from the literature is that the way these tasks are performed in the lab, they are effectively the same, right? So especially in the case that I'm able to address with the current models where you can ask about accuracy. So the motivations are different. The people that measure accuracy when they're doing a visual search task, they, just like people that are looking at object recognition, it's a time limited setting. So I show you the stimulus for like 100 milliseconds. The reason to do that for people studying object recognition, there's a host of reasons, but the way that they've operationalized object recognition, they want to look at first some number of seconds of neural activity, and they want to, and they know that performance is effectively as good as it's going to get within those first 100 milliseconds. For people studying visual search, there's a question of like, do I want to allow eye movements? And one way to control for that is just show the image for 100 milliseconds. So again, make it time limited, but in that case, you don't have reaction time, depending on how you do the task. So if you just measure accuracy in this time limited setting, you end up sort of by accident with the same task, but you're saying that it's visual search. But this is sort of where there's this mixing of the task and what it is you're trying to get at, right? If you're trying to get at visual search, if you're using visual search to try to understand attention, then maybe you think that all you need to do is this time limited setting where you don't move your eyes.
Okay, we have a great question here from Katharina Ducker.
Right, I see, yeah. Can untangling account for serial and parallel properties of search? Yeah. And then she also asks, assuming sorry, I've heard neural networks can only classify one object at a time, which would make me think untangling would mainly reflect serial search. It's a really good question. And I'm trying to give a clear answer. So these neural network models are feed forward. They're okay, yeah. So a little bit of background really quick. There's sort of two competing models based on these simple stimuli. There's a serial model, which argues that limitations arise from some serial processing step like binding the features together into an item. Then there's a separate family of models, which are usually specified in terms of signal detection theory that argue that the limitations are from parallel noisy processing. So a neural network's massively parallel, but it's not noisy, right? It's deterministic. If I show it an image and I don't change that image, I always get the same answer, unless I have it still in training mode and I have something like dropout that makes it behave stochastically. So I can't get an answer if that's my model about whether it's parallel noisy effects. I also can't really ask a question about serial processing because there's no explicit representation of time in these models. So there's different ways you can get at this. So as Elaine will know, because she's working on this, right? You could use like a diffusion drift mechanism to try to get at this. So people have done this, adding this diffusion drift type mechanism to these parallel noisy models to sort of pit it directly against the serial models. So you could do something like that with an AND model, an artificial neural network, and try to sort of pit these two parallel versus serial models against each other that way. And anyone else who knows more about this than me, please feel free to jump in and correct me.
That was great. So there's just one other quick question that looks like it might be quick. Do you train the network for search task on each object you are searching for? Okay.
Right. So in the first case with the simplified stimuli, the training set is all 10 stimuli types at the same time. And we do see, like, basically as you increase the number of types, there is sort of an effect there, which we don't expect to be the case for human search. So that's one sort of place where you could say that there's a caveat here. But and the same is true for the with natural images, right? We're training on the whole data set all at once. So yeah.
Okay, great. I guess we'll move on then. We're just about at time. So our next speaker is Dr. Alex Kell. Alex is a postdoctoral fellow in Elias Issa's lab in the Zuckerman Institute at Columbia University. For his PhD, Alex did computational auditory work with Josh McDermott at MIT. And today he's going to be talking about conserved core visual object recognition across simian primates. Marmoset image-by-image behavior mirrors that of humans and macaques. Is Alex there? Have we lost Alex?
Bringing him back on now.
Sorry about that. Can everyone hear me?
Yeah.
Okay, yeah, sorry, my Zoom quit at the worst time possible. But I am back. All right, so let me share the slides and make sure everyone can see those. Here we go. All right, can you see my slides? Can someone nod or give me a thumbs up? Great, cool. All right, cool. Yeah, I'm Alex Kell. I'm a postdoc with Elias Issa at Columbia. And today I'm going to tell you about some of the stuff we've been up to. And before I begin, I just want to introduce you to kind of the main character of my talk, which is the marmoset. So this is a marmoset. It's a 1-pound monkey. And obviously that's pretty small for a monkey, and that's about the size of a rat. And that's part of the appeal of the marmoset, is that it's one of the smallest of the simian primates. And those are the primates that are most closely related to humans. So the monkeys and the apes. In addition to being small, the marmoset has a pretty flat cortex, which is what you can see here. So you can see, I'm showing you a lateral view of a marmoset brain, a macaque brain, and a rat brain. And there are two things to note. First is that the marmoset cortex is relatively flatter than the macaque brain. You can see that there's a lot more folds. And the second thing to note is that the marmoset brain is much larger than the rat brain. So marmosets are about the size of a rat, but their brain is about 6 times larger than an equivalently sized rat. And a flat cortex is appealing methodologically for a variety of reasons, both in terms of electrophysiology, but also in terms of maybe even more so in terms of imaging. So marmosets are increasingly popular and are seeing a large investment both from Japan, but also from the NIH. But there still remains some kind of genuine concerns about marmosets' behavioral abilities, and that's really limiting their broader adoption. And one of the biggest questions is is marmoset behavior basically rodent-like, or are there capacities that marmosets have that rodents don't? And so that's what we're going to kind of address here. So we wanted to put some numbers on marmoset abilities. So we wanted to quantitatively and rigorously benchmark marmosets to alternative animal models. So we compared marmoset behavior with rats, with macaques, and with humans. And we did this in a quintessential domain of systems neuroscience, visual perception. So to evaluate these species on different footing, we replicated experiments previously done on other animals in marmosets. So the first one that we did was a study that was done on rats, where they saw these two objects at different scales and rotations, which you can see on the top right of the screen. And the rats had to classify which one they saw. And so here are the results from the previous work. The authors found that rats performed relatively well, about 85% at this view that they had the most experience with, this kind of canonical view. But the performance, as you can see, quickly dropped off. But nonetheless, the fact that there was any kind of generalization off this kind of medial axis where they had a lot of training was the key finding of this previous paper, and they were pretty excited about it. So what do we see when we run marmosets on this exact same stimulus set in this exact same paradigm? We see a pretty different pattern of results. Rather than kind of seeing this quick drop off, we see the sea of blue, which is to say that marmosets were basically unaffected by these rotations in scale, where rats were dramatically affected, marmosets were simply invariant. And this result demonstrates that at least in the domain of visual perception, marmosets are clearly capable of things that rats simply are not. Okay, so we next compared marmosets with macaques and humans. And we did this on a task that's relatively hard, that humans aren't at ceiling at. And it's this kind of core, quote unquote, invariant object recognition task, where you'd see different objects, in this case, camels, wrenches, rhinos, and legs, at different rotations and poses and scales, at different on these rich naturalistic backgrounds. And so you just see a flash of an image, as kind of David was talking about before, of you just get this quick image, and then they would have to determine what they saw. And so we measured performance for marmosets and for humans for each of 400 different images. And so the kind of the takeaway from each animal is going to be this 400-dimensional vector that's with each element indicating how hard each image is. And you can basically think of this as a barcode of each visual system. So to evaluate performance across these different animals, we measured these fingerprints on each, and we just simply correlated them to compare them. replicating previous work, we found that macaques and humans were relatively correlated with an R just under 0.8. So that's pretty good. The critical question is how similar are marmosets to humans? And given that this is a pretty high bar, we were just kind of hoping that they were off floor here. But what we ended up seeing was that marmosets were pretty strongly correlated with humans, and not as correlated as macaques, but pretty darn close. And notably, this kind of behavioral signature seems to be somewhat non-trivial in the fact that deep neural networks, like the kind that David was talking about, fail to show this kind of similarity, the same degree of similarity. Even though they're performing this task at human levels, they exhibit a non-human-like behavioral signature. So what we see here is for high-level visual behavior, marmosets clearly outperform rats, both in terms of their overall performance, but also critically in terms of their generalization and the nature of their generalization. And then secondly, we see that marmosets are quite human-like, and basically as human-like as are macaques, at least in this one domain. And we see these results as a kind of demonstrating marmosets have been a bit underestimated, and that they can perform these challenging high-level tasks, and they can do them in a pretty human-like manner. So we tend to think that marmosets seem to be a pretty powerful small model organism for visual neuroscience, and maybe even beyond. So finally, I just want to point out that there's a preprint of this work up on bioRxiv that would be great to check out. And yeah, thanks. I'm looking forward to kind of talking about this.
Really interesting work, Alex.
Thanks.
The first question that jumped to mind for me was whether you'd looked at response times across the species as well, and whether they were.
Yeah, so we haven't dug into those that carefully because reaction times just tend to be a lot less reliable than errors in this paradigm. So we can get really reliable, like those 400-dimensional signatures can be correlated across across animals at like an R of like 0.9 or higher. But we don't. But the reaction times are much less correlated, so we haven't really dug into it because we haven't really been able to get reliable measures there, or as reliable. Yeah.
Okay. I think we have a question. The question is, do we have a mapping for marmoset brain anatomy?
Yeah, so there's been a lot of big work kind of developing some broader infrastructure for the marmosets in the last few years, including like there's recently, I think last year in Nature Neuroscience, this big MR-based, both the diffusion, myelination, those kinds of things, atlas of marmoset brain. There was also like set architecture atlases, maybe from like 10 years ago that was developed then. And there's also tracer studies. And there's been various, I'm not sure exactly the state of what's out and what's not out on that stuff, but there's some folks who are kind of developing that stuff. So a lot of these, like, because I think an appealing thing about marmoset, like as a, or one of the appealing things is there's a lot of kind of energy around them. And so a lot of this kind of core infrastructure is being built up. Rather than just kind of picking one species and you're kind of the only lab doing it. It's nice that there's kind of this broader community.
Alex, if I may ask.
Yeah.
So the performance of the rats kind of drops down when they move away from the canonical orientation on things like that, right? So like, are the images presented to the rats like at the center of their or at least each eye's receptive visual field? Is that the case?
Yeah.
Or
so
like at the front of the head or something like that.
Yeah. So the images for the rats. So rats obviously have pretty low visual acuity. Like rats don't have a fovea, as I'm sure you know. Their visual acuity is like 1 cycle per degree, whereas like humans are like 60 cycles per degree, and marmosets are like 30 cycles per degree. So these images actually have been blown up to a huge degree. They're like 30 degrees of visual angle, which I don't know, for a visual neuroscientist is kind of strikingly huge. And they are presented like, to the extent that there's like, they don't have a fovea, but they are presented centrally for the rats. So I don't think it's, and moreover, in that case, they're not flashed. And so they have plenty of time to kind of move their eyes around. So I don't think that they're like,
okay.
Yeah. I don't think that's driving the effects here.
Okay, and there's a question here from Katharina Ducker again. Is there an advantage of testing marmosets instead of macaques in neuroscientific studies, for example, ethically?
Yeah, well, I mean, I'm not an ethicist. I don't want to speak to that. Certainly methodologically, as a neuroscientist, I think there are major benefits. That's part of the appeal of them, is that they're a pound rather than being like, I don't know, a macaque is like 20, 30 pounds. So they're much easier to work with. But methodologically, there are big advantages in terms of like they have the slack cortex that you can use calcium imaging, either 2P or wide field. You can, there are transgenics that are being developed. I think there are a bunch of, those are the main advantages or benefits that I kind of think of when it comes to them over macaques, but there may also be ethical ones.
Yeah, it's great that you, like you pointed out, it sounds like there's a really established research community. I wonder, can you say a little bit more about like maybe what differences you might be able to look at in behavior? Like you've shown really well, I think, that they are able to perform object recognition like humans. Is there maybe some insight you can get by seeing where they're not able to perform object recognition in the same way?
Yeah, no, I think that's interesting. I don't think a marmoset's a human, obviously. So there are definitely behaviors that humans do that marmosets don't do. The more obvious ones for where I would look for would be domains outside of like the visual system, where if you just like look at the relative expansion of different areas across the cortical sheet from going from like a marmoset to a human. A lot of these like kind of perceptual areas, like including high level perceptual areas like IT, have expanded among the least amount. So it seems like they're relatively conserved. Whereas like if you look at like prefrontal or posterior parietal or like kind of these maybe usual suspects, you get the most expansion there. So I'd expect that you'd get probably, if you're the most salient differences in behavior, I suspect would be in behaviors that underlie those kinds of, or behaviors that are underlain by those brain areas. Within the visual system, I don't know, but I imagine there would be some, and maybe looking to like the kind of neuroethology of like exactly the kinds of behaviors that marmosets do versus like our more kind of typical humans might be one hint of where to start looking for that. But I'm not sure. Nothing immediately off the top of my head as to what you would look for there.
Yeah, it makes sense. I totally agree. Like, and then maybe getting a little into the details of the task, like when humans do it, they're not rewarded in any way, right?
Yeah, well, so I mean, like I was a subject for this, and there certainly is like I'm not getting it. There's no juice tube, but I am like I did get some reward, like social reward of like, oh, I got that right. Like it's hard to kind of avoid that, but there's not juice, obviously.
Can you? How do I get a juice tube? No, sorry, that's not my question. But yeah. And then I think someone might be asking a similar question about like, are they performing the experiment in place where the animal is, about asking questions about stress? But I'm guessing they're like super habituated by the time that they've.
Yeah, so I mean, we actually do this in their home environment. So we just attached these like nest boxes on to their home environment, and they work there. We don't even bring them into the lab. Cool. And there are probably there might be ethical benefits to that if they're able to hang out with because they're pair housed and group housed. They're able to hang out with their family and friends. I don't know, housemates. But there are also methodological advantages of it that allows you to parallelize across many different animals and collect a lot of data like much more quickly than if you had to bring each one serially into the lab.
Sure. Yeah. Mechanical marmoset instead of mechanical Turk.
Yeah, no, that's what we call our software, MK Turk, Monkey Turk rather than M Turk.
Perfect.
Yeah.
Okay, great. Well, thank you all. Thanks very much to all of our panelists once again. There were really fantastic talks and really interesting discussion.
Thanks for
really enjoyed the session. Thank you.
Yeah.
See you.
Thanks.
Okay, hello everyone. I'm Valentina Borghesani, and together with Arash, our amazing behind-the-scene helper, we bring to you two great speakers for this session. Regina Zaghi from the Istituto de Neuroscienza de Alicante, and Jeffrey Mitchell from the Department of Psychology of the University of Bristol. And please remember to write your question for the speakers in the Q&A section at the bottom of your Zoom window. And you can start typing during their talk, and I'm gonna have the pleasure of read them right after. But also check out the chat, because there is where we will put some nice information about the speakers, some link on where to find them online and offline. Our speakers will talk for about 12 minutes each, presenting their work, and then there will be 3 minutes to ask them questions. So it's my pleasure to introduce the first one, Regina. Regina works at the Istituto de Neuroscienza de Alicante in the lab of Alejandro Gomez Martin, and she'll show us how to play magic tricks to deep neural networks, untangling human deception. Please, Regina, take it away.
Thank you. Can you see my screen? Okay, perfect. So, well, as she said, my name is Regina Zaghi. I'm a first-year PhD student here in Spain. And the goal of my PhD is kind of finding ways to study human cognition outside of the strains of lab and in the real world and in real world scenarios. So I've been working with these wonderful people for some time, and we've been playing magic tricks to deep neural networks to try and untangle human deception. So basically what we're doing is we're using magic as a tool to study human cognition. Magic seems like a great source of information to study cognition because there are a lot of processes and biases in our brain that allows us to get fooled by magicians, but also we are enjoying it while we're getting fooled. So it's kind of like a great experience to have. So I am not doing the magic, right? We're working with a professional magician, Miguel Angel Gea. And we started actually studying the magicians themselves and the tricks that they were doing. And we started using Deep Lab Cut as a tracking tool to see the fingers, to see the coins. We started seeing the movement, how fast they were, if we compare with other magicians. So there's a lot of things you can study from a magician, and we're already working with that. And then we also focus on the public. What is the public seeing? How is it reacting? The public is really important for our magicians because magicians cannot trick themselves. So they need a public to trick someone else. Now, the public, they're gonna work as groups and they're going to react also as individuals. So there's a lot of things to see here as spectators. The thing is that while we were doing the tracking, we realized that there might be another subject that we can be using to study or to compare cognitive maybe ideas. And that's when we realized that there were maybe 3 subjects that we want to put attention in our research. So we have the magician that I put a yellow color, like the spotlight of the magician, the human spectator that they have gray matter on their brain, so that's why they're green, and the blue cold machines, right? That's the blue ones. So we have these 3, and we were already working with the first 2, but we wanted to focus a little bit in what to do with the machine. So the Deep Lab Cut, it's a software for tracking with deep neural networks. It also has the convolutional layers that work with score maps that create likelihood of maps to find the location of the labels that you created. And it's also trained with ResNets that have that they come from this bank of images that give a reference to the machine. So knowing this, what we want to do was basically turn this deep neural network tracking system into an artificial spectator. Now, to do that, we have to tone down a little bit of the whole magic experience, right? So we chose some magic tricks that they were only visual and that they have some things that they could be tracked by the machine. So we had 5 magic tricks with coins. So we made a video and we made a lot of repetitions with the magician. We trained the Deep Lab Cut and then we test with the rest of the repetitions. But the training that we did, we showed the machine that there was a coin we also showed that when you close the fist or when you're not seeing the coin, you can still see it. I mean, we tried to show some object permanence, something that we learned when we were little. And we were training it as a naive spectator. That means that we were training it as someone that does not know exactly what's going on, that's not the trick, right? Now, with the humans, Miguel Angel has been working for a long time and he knows that these tricks work, right? He knows how to fool us. But we have some weird results with the deep lab cut, because there's sometimes that it's getting full, sometimes that it's not getting full, right? So for us getting full in humans, it means that the trick worked, but for the machine, it means that the prediction that it makes, is showing the reality of what's happening, the thing that the magician knows. So there are some times in which you can see that the machine can do it and sometimes that it cannot. Now, if we go to think about all the cognitive processes that are involved here, there are a lot. And this is a paper that summarizes some of these most important cognitive process involved as a spectator, right? Because the magician is going to play with lots of things. He's going to play with the input of images or to our senses, right? It also knows that we want things to continue the way we already know. And if they don't continue, they're going to create a big contrast. And in those contrasts, it's gonna grab our attention, so it's kind of misdirecting us. It also knows that we have perception, and in that perception, we kind of fill blanks with concepts we already have, and also with the memory of events that we learned in the past or that we learned in that moment. The magician is always creating subliminal messages, and it's kind of forcing us to make decisions so the tricks kind of work. So that's what we know that it's happening to us in our brains, right? So we want to see if we can compare it somehow to the machine. So right now I'm going to show you the tricks, but you're gonna have to try to pay attention. They're gonna be a little bit fast. First you see the trick, then I explain the trick, and then we see what's going on with the magician. Just remember that this is not the same experience that's being in the theater with the magician, so be kind of gentle with that. So let's see the first trick. Okay, so I hope you can see that it's throwing 2 coins and then suddenly the coin is already in the hand, right? So what's happening here is that instead of throwing the 2 coins, it's just throwing one of the coins, and there's another hidden coin that he's launching really fast with a lot of talent, because I've seen it in real life, and you just can't notice this, right? So this is what's happening with the machine. The machine is already tracking that there are 2 coins and both of them are also falling. So it's kind of not knowing what the magician is doing. And it's also not seeing the fast coin that's going on over there. So for us, we know that there's some postural inference that the magician is playing with how we think things are going to continue. And maybe it's doing the same to the machine. Let's go to trick number 2. I really hope you don't have that much lag, but if you can try to understand what's going on. So the magician is showing the coin and then he's tossing it to the other hand and then it's magically disappearing. So what he's doing is that he's doing a false deposit. He's actually not throwing the coin and it's in the same hand and then it's just putting it back in the pocket, right? So this is what happens with the machine. It sees the coins, it thinks it's going to the other hand, but it's actually not going to the other hand. So it's gonna got tricked a little bit too over there. So yeah, again, the magician is working with occlusion. We assume that the object permanence is still in the hand, but with the speed, he's kind of tricking us and the machine. So let's see trick number 3. So, okay, lots of things are happening here. There's 2 coins in one hand, there's 1 coin in the other hand, and a lot of things are happening really fast. What's actually happened is that there are 4 coins and he's always dragging 1 coin, right? So For us, that's a lot of information happening really fast. But the machine, it's also a tracking software, right? It's not a problem to see which one is dragging and which one is not dragging. And so it's not going to fool the machine because it knows that there's something there. It doesn't feel that information overload that we have, right? Let's see trick number 4. Now this trick, it works better when you're in person, right? Because he's putting the 4 coins, but the 4th coin, you're not supposed to see it in that moment because he's kind of putting number 3 and 4. But when he's doing that, he's showing the other coin in the other hand, and it's also seeing the coin in the other hand. So when you're there with him, you see where he's seeing, right? So there's a misdirection. Now with the machine has no problem. It really doesn't care where the magician is looking at. It's just finding the coins, and that's the task, and he's doing it. So it's not getting fooled by that. No misdirection of attention. For us there is, but not for the machine. And in trick number 5, okay, let's start again. So what's going on here is that we assume that he's grabbing 1 coin in each hand, but he's actually grabbing no coins in one hand and 2 coins in the other one. So it's a really simple trick to do, just knowing that we are going to assume something really simple. The machine does not have those simple assumptions, right? So it's not going to get fooled. It's just going to track what it sees, no matter where the magician is putting the hands, right? So it doesn't have that modal perception or that trying to fill the blanks, right? So when we start trying to see actually what's going on, there's a lot of things happening inside of each of these subjects. And there are a lot of things that are different for them. Some they might be comparable, some might not. For example, the human knows that there are some coins. It doesn't know exactly the number, but the magician and the machine both know exactly the number of coins. Nevertheless, sometimes the deep lab cut gets tricked and even knowing the number of coins. Now, there are other things that humans know, like what's a coin, what's the concept, how does it work with other things, how does it weight, it's not going to simply disappear. The machine does not have this information. It has the reference from ImageNet, right? As humans, we have object recognition. We know it's a coin, but deep lab cut, maybe it's recognizing features. We not really know, right? Also, deep lab cut works with frames independently. So you can put the video on reverse and it will still find the coins. And for us, that wouldn't work, right? So there are a lot of things happening inside of each of them that are constraints sometimes, and sometimes they're just opportunity for you to compare and think what makes the human experience as a cognitive being experience a magic trick, what makes a machine, or what is the machine actually trying to do, right? We cannot say it's thinking something, but something is definitely happening, and how the magician is just using all of those biases that we have to, instead of manipulating us, entertain us, right? So as researchers, we just want to focus in these 3 aspects, on these 3 subjects. And I'm going to finish really fast. So we're just going to, with the machine, we're keep doing some things. We're showing the machine what happens if it knows the trick, for example. And we're also trying to get a little bit inside and see what are the score maps. We can see that it's getting confused because it's not always the point where it shows the final results. Sometimes it's getting confused with the forehead because it looks like a hand or something. And the deep dream, we know that it sees something, but we're not sure how. So this is all opportunities for improvement. In research cognition, maybe to focus in new different ways of making research. Also improving the tracking AI, showing this object permanence physics, so the tracking is easier for these neural networks. It can make possibilities of making better magic tricks, maybe simpler, or maybe magicians are going to have to entertain the machines in the future, so they should be already ready. And some new directions that we can have. How can we accommodate these different constraints we have so we can make comparable experiments between all of these intelligence beings? And what will happen if we tell the number of coins to a human? Will that change the experience, or will it get tricked? So just here's the YouTube with all the videos if you want to see it. And thank you to all the people that's been involved in this project. So thank you.
Thank you, Regina. Really interesting avenue of research. And I see we already have questions, so I'm gonna go straight to it. Jan Ostrovsky is asking, the video material of a magician doing trick is already very complex, as you explained. How does AI work with simple visual illusion? Can they be a naive onlooker in those cases? Have you tried like simpler kind of visual illusion? Because it's true that these are already very high level ones, right?
Yes, we haven't personally, but I just saw a study of other Spanish investigators that they're doing research just by image contrast, and the machine, it still can get full, but it can see like the high contrast. It's just a really simple experiment, just showing the high contrast between images, and like those kind of illusions in which this is the same color of gray, but if you put it in white, it looks, yeah. So that kind of thing, it also kind of fools the machine in somehow. So we're not doing it, but I know there's other investigators doing that research. Yeah.
Well, thank you very much. Really interesting. If there will be other questions in the Q&A or in the chat, we can try to circle back at it at the end, or we can send them to you, worst case. And we shared your contact information so people will be able to look you up online. And now we can move to our second speaker. Thank you very much again, Regina. So our second speaker who is joining me on screen is Jeffrey Mitchell from the Department of Psychology of the University of Bristol in the lab of Jeffrey Bauer. And he'll present us his work on spatial symmetry and its relation to grammatical structures. So please, Jeffrey, you can share.
Thank you. Yeah, so this is a talk about the relationship between spatial structures and grammatical structures. So the linking idea here is symmetry. So spatial symmetry is an important constraint in neural architectures, particularly to do with vision. It's been enormously useful in helping us recognize pictures of dogs, mainly. And so the important thing about spatial symmetry is that given a dog in one position in the training set, we are able to generalize to the same picture of a dog anywhere else at test time. So this has been an enormously important constraint on the architectures that people have looked at in deep learning. But what I want to suggest is that this is also enormously useful in thinking about symbolic systems. In particular, we can apply spatial symmetries to the memory structures that symbolic systems work with. So not only will spatial symmetry help us deal with external input in the form of images, but it also help us structure our internal representations in terms of memory. So just to motivate this, I've got an example sentence. The barmaid that John's extremely obnoxious friend, offended, refuses to serve us. So when you're processing this sentence, what you want to pick out of the sentence is who did what to who offended who, and who refuses to serve us. So this is a relationship syntactically between nouns and verbs. And recently, neural language models have become extremely effective at processing such sentences and extracting this who did what to whom information from them. Lecretz et al. Looked at how these syntactic dependencies between nouns and verbs were represented within a neural language model. And what they found was that short range of dependencies, such as between friend and offended, were represented in a separate unit from the long-range dependencies, say between barmaid and refusees. So this is a problem because really these are the same dependency between, syntactically they're the same between a noun and a verb, and you want to represent them in the same way. So what we're going to do is suggest that you can use a symmetry to connect these short-range and long-range dependencies. Just as you can learn that a dog is the same wherever it is in an image, you can use symmetry to learn that a short-range dependency is the same as a long-range dependency. So just thinking in a bit more detail about that sentence and how you process it psychologically, what would happen is that you hear the barmaid and you think, well, what did the barmaid do? So you need a verb phrase. Then you hear that, so you know there's some extra information about the barmaid. There's a relative clause. The barmaid that did something else. So you need two verb phrases. But now you get John's. You need to know what belongs to John. Is it a hat or a leg? And instead of a noun, you now get an adverb. So you need an adjective to go with that adverb. So extremely tall or extremely short. So you need an adjective next. So we've built up these expectations about what we expect to happen in the sentence. And what's going to happen next is that we've fulfilled these expectations in reverse order. So we're going to get a last in, first out structure. So the last expectation was adjective, and that's going to come first. So we say it's extremely obnoxious. We then say it's John's friend. What did the friend do? He offended the barmaid. What did the barmaid do? She refuses to serve. And that's why we get these short range and long range dependencies, because they become nested as we do this backwards recall. So linguists have thought about what sort of computational machinery you need to process this. And in particular, they've also been interested in what sort of constraints do you want to impose on your computing machinery in order to account for natural languages. And generally speaking, they've gone for context-free languages. There is a hierarchy with Turing machines at the top and finite automata at the bottom. But generally speaking, when we talk about language, we're thinking about context-free grammars and push-down automata. So here's an example, a really simple example of a context-free language. We've got 5 symbols, A, B, C, D, and O. And each sentence in the language is a palindrome. It's the same forward as it is backwards, with an O in the middle. And so for each A in the first half of the sentence, there's a matching A in the second half. The same with B, D, C, and A. And so we get these long-range dependencies and also short-range dependencies, and they behave very like the dependencies between nouns and verbs. So we're going to use this toy grammar to explore context-free grammars rather than the full grammar of English, because that's a lot more complex. So in terms of a symbolic machinery for processing such a language, this would typically be a push-down automata. And what this is, it's a controller that reads and writes from the input, and it has a push-down stack, which is a last-in, first-out memory structure, which it writes to. So it's here in the middle of the palindrome, and it's read ACDBA, and it's written that to its stack. And it's now going to read it out in reverse in much the same way as we saw with the barmaid example. So ABDCA. So that's a symbolic architecture, but I'm more interested in the neural architectures. So neural architectures are very similar. The main difference being that the memory here has no structure. It's just a set of hidden units that the neural language model can use however it pleases. And in particular, one of the popular architectures is an LSTM. And basically that has 3 types of gate: an input gate that writes to the hidden units, an output gate that reads from the hidden units, and a forget gate that essentially controls the recurrence. It controls what is held in memory and what is forgotten. So if we take this LSTM, we can train it on some language data. Here's some data from that palindrome language. We have medium length training sequences, and then we have some out of domain test with much longer range dependencies in them. So if we train our LSTM on that data and test in domain, that is on sequences of the same length, we'll get 100% generalization to that in domain test set. If we now try to generalize to much longer sequences where the dependencies are much longer, we haven't seen that before, our performance falls off very sharply. We no longer generalize. And like I say, this is because there isn't a connection between short range and long range dependencies. And so what we want to do is to make that connection via asymmetry. So in much the same way that you say a dog on the left is the same as a dog on the right, doesn't matter which position it's in, we want to say the same thing about these dependencies. It doesn't matter if it's a short range one or a long range one. So we'll take our neural memory, our neural language model, our LSTM, and in order to structure those hidden units, what we want to do is impose a symmetry so that every range of dependencies within those hidden units is treated in the same way. It turns out we can do that simply by replacing the forget gate with a convolution. So the same convolution that we use in image processing, we can impose on our hidden units, and it turns the hidden units into a push-down stack. They now have a spatial structure, they have an adjacency, and we can push things up and down the stack in exactly the way we need. And so it's not surprising that when we train such a model on the language, you now get perfect generalization to the long range dependencies. So that points out that there is this connection, there is some sort of connection between spatial symmetry in image processing and context-free grammars. So the constraints that people have been looking at in terms of neural architectures turn out to be very similar to the constraints that people have talked about in terms of context-free grammars. So this work is related to a number of other investigations. On the symbolic side, a number of people who are interested in the grammar of languages have looked at the relation between planning and navigation and symbolic grammars. People who are interested in how planning and navigation happens in the hippocampus have also been interested in looking at how those spatial representations relate to more abstract conceptual knowledge. So our work is kind of sitting in the middle saying, you know, the bridging connection possibly is this idea of symmetry. So thanks for listening. I look forward to some feedback.
Thank you very much. Very interesting. And personally, I study like semantics, like high-level language. So my mind was immediately going towards the grid light code for concepts. Like it resonated with me. Maybe sort of a naive question just to start. Do you have any data or any insight, or are you planning to explore how this would actually work with English, like with a real language, with grammar in a real language?
Yeah, so that's a good question. I mean, so I'm not the only person looking at these stack-like recurrent models, and people have found that there may be advantages in terms of uncovering the syntactic structure, the syntactic trees within languages like English using a stack structure. The trouble is that natural languages are really complex, you know. There's all sorts of things beyond context-free grammars. Even beyond syntax, there are, you know, semantics is a very important, and that introduces dependencies that can be very diverse and tangled. There are parts of language that are not compositional. They are just, they don't really have an internal structure. So all these things conspire to make these models difficult to train. There is, of course, the issue that maybe back propagation isn't the best way to train these models anyway. So even on this simple toy grammar, the training is quite often non-robust. It quite often goes off and does something crazy that I don't want it to do.
Right, right. There are always too many things that are covariate in natural languages, right? Like to disentangle one thing from another then becomes virtually impossible. So what is the direction you're thinking of taking? Like, what's the next step? Do you think there's another principle, another spatial principle on top of symmetry that you could inject in the model, or?
There's various things I've been thinking about. So making the training of these stack architectures a bit more stable, that would be useful. Bringing more semantics into it. So at the moment, it's really just a connection between context-free grammars and symmetry. And as you mentioned, semantics is an important part. So we have to think about how that fits into the model. And in particular, how it fits into the model without deteriorating the syntactic part of the model, which is complicated in itself. So that there's a few directions to go in.
Absolutely, very interesting. I don't know if we want to have both speakers back online, if you guys have questions for each other. I'm still monitoring the chat and the Q&A in case anyone writes now. But otherwise, I'm just gonna thank you very much for your time. Thanks all the attendee, and possibly good night for you that are in Europe. Thank you very much.
Yeah, thank you.
Yes, thanks.
Bye.
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 So we're still in the practice room.
Okay,
so I'll click this blue button up here.
So when we go, I will start. Like it should be in 1 minute or so, right?
Yeah, I would give people a minute or two to filter in.
Okay.
You'll see the participants number increasing.
I can see it in the attendees in the chat, right? How many people we have?
Yeah, in the participants window.
Okay.
All right, I'm going to disappear my video. Okay, I'm going to go ahead and go live. We should be there in about 10 seconds.
I see attendees are joining. So we will wait a minute or so. So I can see that the number of attendees are rising. So we wait a couple of minutes just to let people to join.
Okay, let's start. Hi, everyone. I hope you enjoy NeuroMatch conference so far. Hereby, I would like to welcome you to this session. I am Yalda Mohsenzadeh, the MC of this session. And it is with my great pleasure to welcome Dr. Dwight Kravitz, a professor of cognitive neuroscience at George Washington University today. And Dwight's research expands a multitude of cognitive neuroscience domains, including visual object recognition, word recognition, concepts and scenes, mental imagery, and visual working memory. I'm personally a fan of one of his papers published recently in the connection of perception and visual working memory. And like you, I am excited to hear his talk today on predicting functional organization and its effects on behavior. With that, I welcome Dwight on the stage to start, and also remind you that you can use the Q&A panel to ask questions, and I will ask them at the end of the session. Okay, thank you.
Well, I want to thank everybody for coming, and thanks to Brad and Yalda for inviting me and pulling all this together. I know it's an incredible amount of work, particularly in the midst of a pandemic. So to jump right into it, this talk is really sort of a whirlwind tour of a bunch of different studies that I've done over the last 10 or so years that we're looking at specifically bridging the divide between cognitive science, the science of behavior, right, and neuroscience, which was and still is producing a number of fundamental insights about how the neural substrate behaves, how it can support complex behavior. But between the two, there's a vast gulf of complexity, right? We really do not have a firm handle on complex circuit behavior, and the entirety of systems neuroscience, certainly from a cognitive perspective, ultimately relies on trying to find aspects of the physiology which are correlated with behavioral performance, and then in theory also related to our sort of putative cognitive mechanisms. But in fact, the problem is really very difficult. The river itself, right, is constantly changing. It changes from person to person. There's no reason to expect that the solution of a neural network to a problem in one person will necessarily be the same in another. And as we'll see towards the end of the talk, it changes from trial to trial, where the relationship between the physiology and the adaptiveness of individuals' behavior is going to change in reference to the particular task context. And this, certainly in my career, has been a limiting factor in how tightly we can actually bridge this divide. The typical approach, I think, is we have some sort of putative mechanism, right? A theory that produces a hypothesis that we test in some particular instantiation. And then we go ahead and look and see, while the subjects are performing this task, what the activation of their brain is like, right? At least within the context of human neuro. And then I think oftentimes the temptation is to look at the pattern of activation we see and start ascribing particular functions, right, related to our putative models to the activation that we see. So we point at a particular blob of activity and say, aha, this is, working memory, or perhaps the central executive or the sketch pad or whatever it is. I think in practice, this approach, as the field has started to really acknowledge, has its limitations. What we're doing there is looking for bits of the variance in the physiological response that agree with what we thought there should be. And when we find agreement of any sort, really, we're happy enough to go ahead and report it. This is a good sort of first order approach, but I'm going to advocate for doing something a little bit different. So the approach that I've taken, along with a number of others, I think in this field, has been to try to take our physiological observations and then look in detail at the things that we know, right? So let's take, for example, the case of localization. So we have some area of the brain that's active in a particular task, but now rather than leaving it at that, let's instead dive into the known physiological properties of that region of the brain, see whether or not any of those properties then make some kind of a behavioral prediction. Right? So rather than just noting that there's some variance that seems to agree with us, see whether or not the consequences of the engagement of a system are reflected in actual ongoing behavior. And ideally, that should be a prediction that's novel, something that you couldn't have made without the physiology. And that helps to bridge this divide, right? It helps to shoot the arrow from the other side of the chasm, right? Having managed to find its mark. And then eventually to work our way back to our original models and update them. Right? Not just to sort of wholesale reject or accept them on the basis of the data, but to try to bring them into closer alignment with the known properties of the physiology. So I'm going to take you through a set of studies across 3 different domains where we attempted to do this, looking again at localization and category selectivity, the relationship between working memory and perception, and then finally at a set of domain general mechanisms and how those are going to play out in the context of attention and learning. So to begin with category selectivity, it's helpful to kind of set the stage. Going back to the late 70s or early 80s, our understanding of the primate visual system has been a bifurcation, with a dorsal going pathway responsible for spatial processing and a ventral pathway that's responsible for, say, recognition or identification. And the core data that established this dissociation, right, is the inability of monkeys with lesions in the dorsal pathway to learn tasks based on spatial adjacency, but they are unimpaired at learning tasks on the basis of recognition, and vice versa is true with ventral temporal lesions. This basic idea, right, of certainly the ventral pathway as a staged hierarchy, where what you're doing is proceeding from early visual cortex down the length of the temporal lobe through a series of processing stages, each one of which is getting more computationally complex and building more and more complex representation from the aggregation of the simpler representations rather, that are feeding into those areas, has proved to be very powerful, right? It's a relatively simple idea. It's elegant. There's a fair amount of data that sort of directly supports this characterization, where you have an increase in, for example, the latency of the first spikes, and an increase in receptive field sizes and the complexity of representations along the hierarchy. When we take this basic idea, which is really a sort of neuroanatomical extension of Hubel and Wiesel's ideas, and then we instantiate those into computational models, we get really powerful results. Models are quite capable at this point of doing really complex object recognition, even amongst complex scenes. That story, right, is therefore quite compelling, and it's largely an inductive story coming out of neuroscience, with some observations coming from monkey neuroanatomy and monkey lesion work. But we run into a problem when we then look at the human data. So human fMRI, particularly sort of the early studies looking at category selectivity, were coming from the other direction, where they were looking for neural correlates of the sort of specialized processing for particular categories that have been observed behaviorally. And what they find is actually quite astonishing, right? It's a set of regions which are in stereotypical locations across participants, right? And each one of these regions appears to be selective or more strongly responsive for one category than for many, if not all, of the others tested. And the fact that these regions are also showing up in stereotypical locations, right, suggests an importance to this organization, right? It's not a totally arbitrary organization. But there's no way to go from the sort of staged hierarchy overarching model, right, of the visual system to clustered selectivity that's always occurring in the same location. And as a result, some proposals have emerged where this organization is proposed to be actually arbitrary relative to the neuroanatomy, which is to say, here in a paper from Edward DeHaan and Alan Cowie, right, they conceptualize the extrastriate cortex as a series of specifically evolved modules, each one of which comes into being in order to support processing of a particular type, and then is densely connected to other modules which share that sort of processing, but only loosely connected to ones which don't. This is a conceptualization that certainly fits with the fMRI data, but it produces a problem, right? The first is it has no underlying relationship to the non-human primate literature at all. You know, beyond just now running the same studies in primates and seeing if they also have these areas. And the other problem is that it opens up a sort of infinite quest for new regions, right? We can go out and now try to tessellate the entirety of the inferior temporal cortex with sort of object selective modules, but it's not entirely clear, right, that we're going to get any of a deeper sort of theoretical understanding of why those modules are there, right? In fact, this conceptualization sort of cuts that discussion off. So I'm going to argue actually that implicit in the data that already exists is the answer to this conundrum. The first hint is the VWFA, which is this really, again, very surprising region that appears to be selective for visual word form, right, over consonant strings or pseudo words or digits. It's also in a consistent location across individuals. There is a associated disorder that results from lesions in and around this area of the brain. But unlike the other areas, which are all sort of biologically relevant categories, right, this cannot possibly be the result of evolutionary pressure. Literacy, right, in the general populace in the United States is, you know, charitably really 200 years old. And so there's not really been time for some sort of a mutation event that's going to enable the recognition of orthography. And so in order to resolve this and a number of other related conundrums, I undertook with a bunch of collaborators who you see down here at the bottom. And throughout the talk, there will be pictures of my collaborators, without whom absolutely this work is impossible. Here in particular, I have to call out Salim Khattar-Bacha on the far left, who is simply encyclopedic of his knowledge of macaque neuroanatomy. And so with his help, we undertook to look in greater detail at the neuroanatomy of both the ventral and the dorsal pathways relative to the last 30 years of neuroanatomical tracer work that had been done since these pathways were originally characterized. And as we'll see in the details of this neuroanatomy, we begin to see hints of why this stereotypical functional organization emerges. So here is a sort of summary plot of that neuroanatomy. Don't try to take it all in. It's of course hideously complicated. In addition to the complexity that's internal to, in this case, the ventral visual pathway, there are also a complex set of relationships and projections in and out of it coming from external systems. Like the medial temporal lobe, the orbitofrontal, the basal ganglia, etc. And within this specific set of connectivity, we begin to see a reason why we might observe stereotypical structure even amongst high-level visual representations. And so in order to make this link, the first thing that we have to do is to recognize that the models that we're building, right, are models of a computational principle, right, an algorithmic approach to the problem of vision. What we're testing when we look at our deep belief or DNN models is whether or not a sort of staged hierarchy with these placed nonlinearities is capable of solving some of the problems of vision. But this is not really a model of the actual detailed neuroanatomy. And as a result, those models are not going to naturally produce the same functional organization that we see in humans. So the argument that I'm going to make here in this section is that natural selection, right, the evolutionary events occur at the level of the gross connectivity of the brain, right? They are in response to the problem of packing this many neurons into a person's head, which necessitates that only some connectivity is going to be possible. And that in turn is going to mean that information is only available, right, in certain parts of the brain about certain aspects of the visual world. Right. As a result, when you go ahead and now begin to interact with faces and scenes and body parts and so forth, there are certain parts of the extrastriate cortex that have better access to the information you need in order to do that sort of processing. And as a result, you end up with an experience-driven development of category selectivity, right, which nonetheless will still have a stereotypical location defined by the gross neuroanatomy. And so we set out to look at this experimentally, and moreover, to look at it in a domain where we could really take a data-driven approach that would allow us to see whether the predictions from the neuroanatomy really hold true without necessarily having to have a biased paradigm design, right, where it can only show us that we're right or not. And so we chose the domain of scenes. Scenes are great because they are exceedingly complex. right? When you look at this, this room, for example, you have undoubtedly across the entire cortex responses to a bunch of different aspects of this scene, right? So you could be responding to the objects, to the category of scene. This is a hotel room, to the geometry of that scene, right? How navigable is it? How do I get from one side of it to the other? You could be grappling with the complexity that this is an enclosed space that has windows to the outside world, and that you're a fair deal off the ground. Right. In addition to being able to handle all of those different forms of complexity, there's also a huge amount of heterogeneity, right? Different scenes look very different from each other, and yet the underlying physiological response has to be capable of capturing, right, aspects of all these different aspects of the scenes and across very different scenes as well. So what we decided to do was look at part of the ventral visual pathway that is known to be highly scene selective, the parahippocampal place area. You can see its location here in a schematic, and then also in raw 770 EPI slices, right? This sort of blue swath here. And what we're going to try to do is say, well, we know that PPA is responsive to scenes, right, selective for them, but what we can do is now try to predict exactly what aspect of scenes PPA is going to be picking up on the basis of its connectivity, or the connectivity of the homologous area in the macaque. Right, so if we go ahead and now look at the macaque, our homologous area is, not surprisingly, the parahippocampal gyrus, which in macaque includes these two areas, TFO and TFTH. You can see here that they get the majority of their input from V4V. Not shown is that in detail, they're getting the majority of their input from the peripheral portions of the V4 ventral map. Okay, so that gives us some clue that they're going to be, these areas are primarily getting their information about the peripheral portions of space. We can also see some evidence of that in the human data, looking at these sort of classic studies from Rafi Malik's lab, where he shows that there is a center versus periphery bias with the medial aspects of the temporal lobe representing the periphery. We can also look at the descending connectivity coming into this area from the dorsal stream. It turns out that there is an evolutionarily ancient pathway, highly sort of studied in rodent, but also present in primate, that goes from the caudal portions of the inferior parietal lobule down through the retrosplenial posterior cingulate into TFO, TFTH. And you can also see the dense interconnections between those areas and the hippocampus. Okay, again, there is some evidence for this connectivity in the human, looking at functional connectivity. Here, a schematic from a study by Margulis and colleagues, showing in blue a large amount of functional connectivity between the IPL and the medial temporal lobe. And so in our study, what we're going to do then is predict that PPA should be really responsive to the spatial aspects of scenes. It's receiving sort of descending information coming from the parietal, deeply integrated with the navigation system, right? And now what we're going to do is present scenes, but in a way that is unbiased, right? So what we're going to do is present as many scenes as we could. At the time, that was 96. And we draw them from a wide variety of different categories, some of them enclosed spaces, some of them open. And what we're trying to do here is to just see what aspect of the scenes the region ends up representing. What we can do is use multivariate pattern analysis on each individual scene now, look and see how similarly the region's response is treating every pair of scenes, visualize that in this configuration where adjacency is reflective of similarity. Some of you can already see what this structure actually is. It's a little easier if I turn it into symbolic. So here's the same structure now with the scenes colored by whether they're man-made or natural, near or far, and closed versus open. And so very clearly there's a distinction here, right, that the region is holding between these enclosed versus open scenes, right, consistent with its connectivity. What we can do then is now make the pivot I was talking about earlier and try to make a behavioral prediction. So if in fact this region is responsible for representing this distinction, then we should see its representation reflected in participants' behavior. And so what we did was to take a separate group of participants and arrange a series of matches where they were presented with two scenes and simply had to judge which one of them was more open, right? And then using the same math that they used to rank chess experts, what we can do is derive a linear ranking of how open or closed a scene is on the basis of this separate group of participants' responses. We can derive a similar metric, right, from the neuroimaging data where we see sort of how closely correlated the scene is with other open or closed scenes. And then we can directly correlate the two, and the relationship is quite strong. It's always good to have a control region. So here's early visual cortex, which doesn't show the same distinction and shows no relationship to the behavior either. Okay, so in this, what we've done is a data-driven approach to understanding scene representation, and we've shown that it's consistent with its neuroanatomy, but there are other aspects of this organization to category selectivity that we can explain. And so here's a study where, actually a set of studies where we were looking in particular at why these regions tend to come in pairs, right? At first glance, this seems highly redundant and not terribly efficient. The sort of existing accounts of this, the data has always been a little bit equivocal, but the idea is that perhaps the lateral regions are lower level than the ventral regions and are somehow accomplishing, say, an earlier stage of processing that then feeds into the ventral regions that have a higher order kind. If we look at the neuroanatomy, though, from the macaque, we see a different potential reason. So if we look at the lateral surface, we can see that it derives the majority of its input from the dorsal portions of the visual field maps. That is, of course, the lower visual field. The ventral surface derives the majority of its input from the ventral portions or the upper visual field. And so another reason why you might have these apparently redundant regions is because they have been shaped by experience to deal with these categories of objects when they occur in different locations on the retina, right? Namely the upper and lower contralateral visual fields. We can test that directly simply by localizing the regions and then running population receptive field mapping. And we see that this is in fact a difference which holds. What we see is that in the PPA on the ventral surface, there is a bias to the upper visual field, and TOS a bias to the lower visual field. This sort of argues against as well the hierarchical interpretation in that it's difficult for regions to be hierarchically organized if they're responding to different portions of the visual world, at least outside of fovea. All right, so that's my whirlwind tour of category selectivity. I want to sort of try to play the same game now, but in the domain of working memory. So the classic accounts of working memory very much hold working memory to be a sort of specialized system which exists distinct from the circuitry that's responsible for perceptual processing, right? So you have an executive, it has control of these slave systems that deal with specific kinds of information, but it's a sort of distinct process. And the neural models reflect that distinction. So they propose that initial perceptual processing is occurring in these perceptual circuits, but after that, information is going to be recoded and transferred into a frontoparietal circuit, which is specialized for maintaining state, right? We set out to kind of investigate an alternative possibility suggested by the sensory motor recruitment framework, which is sort of actually a generalization of that, which is the idea that actually you maintain information not solely within the frontoparietal circuit, but within whatever circuit is most appropriate for holding that form of information. And so we take a very simple paradigm here adapted from the paradigm used by Frank Tong rather. And what we have here, right, is two samples that come from two different categories of visual object, followed by a cue, which tells the subject which one they should be holding on to during this delay period. Okay, and in both of the two different types of runs of this task, this sequence is exactly the same, right? What differs is simply the task demand of whether following the delay, participants have to identify whether a fragment of an object was part of the item they were holding in memory, or whether an object is from the same particular category, right, as the object that they are holding in memory. And so in the visual task, right, you are required to maintain visual detail, whereas in the non-visual task, you're required only to maintain some abstract category label. And our prediction is that we should see, using again multivariate methods, information during the delay within perceptual cortices during the visual task and within the prefrontal cortices during the more verbal task. Despite the fact that the lead up, right, to the delay and the delay itself is identical. And this is exactly what we find. So here in a object selective cortex, we see significant classification performance of the item held in memory during the visual but not visual runs, and then the nice crossover interaction with the opposite being true in the prefrontal cortex. Right, so this is good evidence that it's not the case that there is some specific system, right, that has been adapted in order to maintain state. In fact, any system is capable of, at the very least, reflecting, right, maintained information. However, what we would like to see, right, is a cleaner behavioral correlate of that as well, right? Is it the case that this information is really indicative, right? The presence of this information is really indicative of that circuit's involvement. And so let's consider then the behavioral predictions that emerge from that result. If in fact working memory and perceptual processing are occurring within the same circuits, then we expect there to be bidirectional interference between the two of them, right? What you are holding in mind should alter what you see, right? And what you are currently seeing should alter what you're holding in mind. And moreover, the amount of interference should be commensurate with the overlap in the neuronal populations that are engaged. And since we know a fair amount about the tuning of these areas for at least low-level phenomenon, we can predict, right, that there should be more interference the more sort of shared cells, and therefore more similar the stimuli are, at least according to these tuning curves. So I'm going to show you that prediction holds across 3 separate experiments. So the first thing I'm going to show you is that ongoing perception is going to bias visual working memory in a way that actually directly recapitulates these tuning curves. And so then it's a very simple experiment. Participants are presented with a memory cue. They have to hold on to it. And then during this delay period, before they have to report the item they're holding in memory, they're going to do an RSVP task where a series of letters is going to appear. And whenever one of those letters appears in white, they have to press a button. Behind this RSVP stream, and totally irrelevant to the task, is another colored disk, right? A distractor. And what we're going to do is systematically manipulate the similarity of that distractor to the memory cue with the anticipation that we should introduce a greater distortion or bias, right, in the working memory report, the more similar the distracting cue is. Okay, and then following this, they're going to do this continuous memory report. Right, what I'm going to plot for you now is the amount of bias that's introduced by the cue as a function of how dissimilar it is. And we get, of course, exactly what we expect, which is this nice monotonic decrease with similarity. This is this game played out in color. We can play out the exact same game in orientation. And again, we see pretty much the exact same result. Okay, this shows us that, in fact, the tuning curves are reflected in this interference between perception and working memory. Now I want to show you the other direction, that working memory is going to alter ongoing perception. Okay, so in order to do that, we're going to test for a distortion introduced into perceptual discrimination thresholds by holding an item in memory. And so I apologize in advance, it's rather involved, but we'll get through it. So participants will be shown a colored Gabor, right? And that Gabor has both an orientation and a color. They are tasked with holding in mind one aspect of it, either the color or the orientation. Following a delay, they will then be asked to do a color or orientation discrimination, where again, they are presented with 2 colored Gabors, and they have to say whether they are the same or different on the basis of one of those features. Following that, they do a continuous report, again, for either color or orientation, depending on what their actual task was. What we're going to do is systematically manipulate the similarity between the discrimination stimuli and try to derive the discrimination thresholds as sort of an extremely low-level perceptual task. And then we're going to see whether or not we observe a predicted effect, right, of what you're holding in memory on those discrimination thresholds. Moreover, as is my habit, I look for crossover interactions and I design for them. And so what we're also going to do is now systematically manipulate whether the item you're holding in memory, the feature you're holding in memory, is going to match the feature upon which you're doing the discrimination or not, right? And so if you're holding color in memory and doing color discrimination, that's match. Orientation is match, but you can also have these non-match conditions. And in the non-match conditions, we predict that there shouldn't be, right, nearly as strong an effect. In fact, it should be largely absent because you're not actively engaging the same population of cells, which you're then going to use to do the perceptual task. Okay, and so how are we going to test for this effect? Well, we're going to take advantage of our previous result, which shows that the amount of interference is going to be proportional to the similarity between the items held in memory and the items you're currently perceiving. And so we're going to have two conditions. In the middle condition, right, in both conditions, we will present the same discrimination stimuli represented by these sticks here on this color spectrum, right? In the middle condition, the item you're holding in memory is going to be between the two. And what that's going to do, right, is draw the representation, right, of the two discrimination stimuli towards itself. In so doing, the actual distance is going to actually be larger than the perceived distance. Then we can contrast that to the case where you're holding an item in memory that's off to one side of the discrimination stimuli, in which case there should be a greater shift, right, for one of these stimuli, one of the discrimination stimuli than the other, and thus the perceived distance will actually be greater. And so in this case, we predict higher discrimination thresholds because it's harder to distinguish the stimuli, and here lower discrimination thresholds. And I'll accelerate a little bit and tell you that that is exactly what we find. So here are the raw psychophysics curves for the middle and side conditions, middle in blue, side in green. You can see that side has a significantly lower threshold, right, than does middle. This is for color memory. When we do the non-match condition, there's no difference between them. Okay, when we do now we look for orientation memory. Again, we see the difference when there's a match condition, and the difference is absent when you're in the non-match condition. We can plot that as the crossover interaction, and you can see very clearly what this effect is, right? So ongoing perception is going to affect working memory, introducing a bias, and it is also going to be affected by working memory, causing slight distortions to be introduced. And this is all, of course, commensurate with the co-localization of these processes, right, to the same areas. I'm also going to show you that these effects persist across delays. So in the recoding hypothesis, over time, you're moving information out of these perceptual circuits. And so over time, there should be weaker effects, right? Whereas with sensory motor recruitment, right, you're using the same circuit. And so over time, the only thing that's going to happen is the working memory representation is going to decay, right? But you'll still get these interference effects throughout. So for the threshold effects, right, we're going to expect both of these models to predict that the effect should reduce over time, though recoding does predict that they will eventually extinguish totally. But the bias should actually increase according to the sensory motor recruitment hypothesis, because you have a decay of this working memory representation. Whereas the recoding approach would predict that the bias should also be extinguishing over time. We can test this very simply by adding a variable ISI to the experiment that we've already run between the orientation and the memory and discrimination task. And we get exactly what we would predict from the sensory motor recruitment story. So over time, the color discrimination effect reduces while the bias effect increases. This is a summary plot of those two things on the same axis. And so you can see that even though there's a reduction in the threshold effect, it stabilizes and it is always significantly above zero. We have run versions of this task out to 6, 8, 10 seconds, and the result holds. Okay, so in total then, what we've done is taken a co-localization that fell out of fMRI data, and we've used it to make a novel behavioral prediction, right, that holds up across a number of different experiments. And it reveals what I think is a really novel and profound interaction, right, between these two processes that we would not have been able to discover were it not for the physiological data, right? And then I think just very quickly, no, there's really not going to be enough time to do justice. So I will leave this last section, and I will only sort of précis it by saying there is a paper which is deposited on the SciArvix, for which there is a link in the chat window that describes this work. In summary, what it is about is showing that the low-level consequences, right, of synaptic response, where what you have is necessarily as a population of cells is firing, a longer term, well, sort of a short-term potentiation of those synapses due to calcium kinetics, but then also a long-term sort of Hebbian learning, which is obliged to always occur. As a result of those, what you can see when you look at big data, right, and you have enough data to look for these subtle effects, is that over time, subjects are about every single feature of the display, actually, ones that are both task relevant and task irrelevant, they are accumulating evidence, right, for the presence of a certain feature. And their behavior on a current trial is going to be profoundly influenced by the history of trials that came before, in a way that is actually strikingly consistent with basic metrics of statistical evidence, and the time course of which is also consistent with known mechanisms of synaptic plasticity. But beyond that high-level summary, there's not really enough time, but we can go into it during the discussion section if we want to. I'll skip, oops, sorry about that. And so what I want to leave with is really just this sort of summary statement. What we've got now is a huge amount of data and a lot of really careful experimentation across the entire field, where what we're looking for are correlates of our sort of putative behavioral mechanisms. Where we've run into limitations has been really with the sheer complexity, right, of the physiological data that we're able to collect. The data is itself a very low dimensional cut through very complicated brain response. It's colored by the particular paradigms that we use. And it is also malleable, right? It changes from moment to moment, as does the behavior of the subjects in our experiment. And so my approach has been, and I think I'm not unique in this, to search for those aspects of the physiology that make specific predictions, right? That are going to hold true even over all of that variability across individual trials and across individual people. But I think that we will need to increasingly, as we did in this final project that I ended up having to skip over, I think we will increasingly have to grapple with that variability directly. We will need to understand what is actually changing from trial to trial and model it specifically to understand what aspects of the physiology are related to stable cognitive mechanism versus what aspects of that response are really capturing more implicit learning, right? That's automatically happening at a local circuit level. And in general, what we need to do is test these really specific behavioral predictions that fall out of our physiological work in order to really bridge this divide much more effectively. And so with that, I want to thank everyone for their attention, and I'm happy to take questions. And if I don't get to a question, just shoot me an email. Thanks.
Thank you so much, Dwight, for the interesting talk. Now we can open the to questions. There is one question on Q&A that I see Hazel Jackson asked. So the question is, I may have like basically missed like the macaques or human studies.
Sorry. So yes, I see it. The macaque work is really all neuroanatomy. The rest of the empirical work is all done in humans. So the work that comes from macaque is really an aggregation of 30 to 40 years of neuroanatomical tracer work, but all of the neuroimaging and the behavior, that's all humans.
Mm-hmm. Okay, thank you. So if you have, like, I'm talking to our audience, if you have any questions, please feel free to send them on Q&A. I will read them. Meanwhile, I have a question, right? So you had some very interesting results on organization of like category regions, and its connection with maybe even topology of visual field, right? If I got it correctly. So I want to know more your insight on that. Like, for example, at one point you said TOS has a more higher visual field bias compared to PPA, which has a like lower visual field bias. That's a very interesting finding. Can you comment on more on that?
Sure. So one way of thinking about this in general, right, is that we want to have an inductive account, right, that stems from the known properties, right, and the better understood properties of low-level visual cortex, and nonetheless makes a prediction, right, about the functional organization that we're going to observe as we move sort of higher up in this visual hierarchy. And we kind of began this actually looking at retinotopy and how sort of position invariant object recognition actually was. And in fact, we found that there was not very strong evidence for that position invariance. Certainly, particularly when you looked at tasks that were more visual versus semantic. And that led us to this sort of long project where we were looking at the potential relationship of retinotopic maps, right, to category selectivity. And when you think about the categories, they come with a bunch of sort of stereotypical retinotopic locations, right? So when you're looking at, for example, bodies, right? If you're looking at a person, you typically fixate their face, which places their body in the lower visual field. And then you look at, say, body selectivity, and the lateral body area is much more reliable and robust, representing the lower visual field than the ventral body area. Likewise, right, the left side of the body ends up in one visual field and the right side in the other. We ran another multivariate study looking at information for body parts and found there was greater information when they were in their stereotypical low locations. And so to my mind, I think that the sort of category organization that we see in the extra striate is reflective, right, of these lower level constraints, right? In essence, the banks of the river. So even though the river changes from moment to moment, and it probably changes certainly at a micro level from individual to individual, at a broad scale, gross connectivity defines the banks, right? There is only certain areas which are going to have access to the right kinds of information. Combine that with experience and frankly, a task demand to deal with this stimuli in the real world, and you end up building clusters, right, which are going to be all processing similar things because they have access to similar kinds of information, and they've been shaped by experience to do that. And so, you know, pretty much every time we've gone looking, be it in behavior, fMRI, neuroanatomy, we see this sort of a linkage. And in fact, as we got more involved in developing imaging methods, right, as the data became cleaner and we got better views of this, of the ventral surface in particular from our 7T data, the more this appeared to sort of hold up, right? The less and less they look like isolated islands, sort of arbitrarily placed, and the more they look like processing streams, where what you've got is sort of a peripheral processing stream that is somewhat specialized for scenes, because that's primarily what you do in your periphery, is process these spatial aspects of scenes, right? And then sort of foveal stream that's devoted to words in one hemisphere and faces in the other.
Okay, thank you. We have a couple of more questions in Q&A. Can this bias considered as a confabulation mechanism related to poor memory, or is it all due to perceptual mechanisms?
So, I mean, I think if you're talking about it at a pure behavioral level, right? If you just look at the behavioral paradigm, then the answer to that question is ambiguous, right? It's difficult to say whether or not you have some sort of a distinct memory representation, right, which is degrading and it's poor and it's being impinged upon by ongoing perception, or whether what you have is the sort of, as proposed, this mutual engagement of the perceptual circuitry. I think ultimately the argument rests on a combination of the behavior and the imaging result. So I think the imaging result says, you know, look, we are equally capable because of this crossover interaction of detecting information in either of these systems, but we really only detect it in this perceptual area during the perceptual task. Then we make this set of behavioral predictions that falls out of the sort of tuning curve properties of those perceptual areas. And so the sort of simplest account, I think, is the one which I gave. If you sort of want to maintain the notion of a distinct system, then I think you have to grapple with, you know, not only why is it that ongoing perception is impinging on that system at all, but then also why is it that the adjacency rules that come from the perceptual tuning are also in this system. And so I think this is one of those cases where, you know, as you know, not one of those cases, as is always the case, there's no silver bullet result, right? And I wouldn't want to just take the behavior and make that argument. But I think in combination with a bunch of imaging work now, I'm more comfortable with that.
Okay, thank you. So the next question is by Hajar, and the question is, in the world of big data, it will be harder to conduct very controlled experiments. How do you think we could incorporate your arguments during this talk with population-based neuroimaging data?
So I think, all right, there's two parts to the question. I'll do the second part first. So in terms of using population-based neuroimaging data, that's actually what effectively, too far. Sorry, there we go. That's effectively what this prediction is, right? What we have here is across a population of subjects in fMRI. This is the average response of the PPA, right? This is on average how similar, how open or closed it treats any individual scene from amongst the 96. And then in a separate population of people, right, again, in a controlled laboratory experiment, what we can do is test and see whether or not the specific representation we derived from the population fMRI holds in the behavior. And so I think there are opportunities, right, to do that sort of work where what you're looking for are going to be aspects of the population level physiological response that do make these particular behavioral predictions. Now, for the former part of the question, you're absolutely right that in the world of big data, you're not going to have laboratory levels of control. That's not going to happen. I think, though, that when we think about big data, we need to think of it as a complementary kind of mechanism by which we can study this. So the advantages are the sheer volume of data, which allows you to overcome some of the issues of a lack of control with the sheer power, right, of the sample that you can collect. So to give sort of an example of that and sneak in a little bit of detail on this final paradigm that I didn't get a chance to get to. Too many animations. There we go. So what we're doing here, right, in this plot is we can look at now behavior on every trial, because we have in this plot 50,000 people, in some other plots, right, 1 million people who have done trial 1, or 15 million people who have done trial 1. And so in the context of big data, I think there are opportunities to look at trial by trial effects, to look at in general effects with a level of fidelity that we've not been able to do before. Where we run into the issues, right, are we want to then see, right, from the big data, a set of predictions that do then hold in our more controlled laboratory experiments as well, right? What we don't want is to have big data sort of spin off into its own subdomain and then have the two camps begin to ignore each other on the basis of either a lack of fidelity or a lack of control, right? Ideally, what we're looking for is going to be results that fall out of the big data that then lead to predictions in laboratory experiments and vice versa. And this project, of which again there is a preprint deposited on the archive, is a start at doing exactly that.
Okay, thank you so much. I think we are like done with the time, like 4 minutes. At the end, I would like to again to thank you, Dr. Dwight Kravitz, for joining us today and very interesting talk. And thanks the audience for the great questions and attending this session. So that's it. Bye, everyone. Have a great day.
Thanks, everybody, for coming.
Us about quantification of active and passive defensive behavior in half-fixed mice. And the floor is yours.
Great. And you can see the full screen, right?
Yes.
Okay, cool. So, yeah, so animals show complex defensive responses in aversive conditions. And here's an example of what I mean by complex response. So as this mouse is moving around in its shelter, a light comes on at the opening, at which time the mouse arrests movement briefly and then eventually may flee. In this case, the mouse has learned to associate the light with perhaps the owner of the apartment coming home. And therefore, the light is the conditioned stimulus, and the freeze and the flee is part of a complex response. So to study neural mechanisms that underlie these conditioned behaviors, neuroscientists use typically Pavlovian fear conditioning. It's the most commonly used behavioral paradigm in neuroscience. And the basic protocol goes like this. A mouse in a box is presented with a cue, most commonly a tone, but it could be a visual stimulus or a neutral odor that initially does not elicit any response. But then the tone is paired several times with an aversive stimulus, like a foot shock. And after this pairing, subsequent presentations of the tone alone leads to a freezing behavior. So while freezing is a clear measure of aversive learning, there are other behavioral components, as we just talked about, to a condition response that are often missed. And this limits the measurements of condition aversion that we can look at when we're trying to study the neural circuitry underlying defense responses. So as Meng-Sen mentioned in my introduction, my goal is to understand encoding of emotional stimuli in the amygdala. And so I record neural activity in the amygdala while mice are engaging with a variety of stimuli, both aversive but also appetitive over time. And while I'm not going to go over the cellular imaging data today, the important thing to know is that we are using 2-photon imaging of GCAM through a grid lens to do this. And we like the 2-photon imaging because we can better convince ourselves that we are holding the same cells over time. But this requires head fixation. And so we wanted a behavioral paradigm that would allow us to do this, but while giving us the ability to measure various behaviors. So the virtual burrow assay was invented by Andrew Fink and Carl Schoonover in Richard Axe's lab at Columbia. In the virtual burrow, a head-fixed mouse has the ability to ingress or move an enclosure over itself. This setup accommodates a 2-photon objective to be placed above the brain for simultaneous neuronal imaging. And meanwhile, the linear displacement of the burrow is measured with high spatial and temporal resolution using a laser displacement sensor. Now, in their original paper, our collaborators extensively characterized behaviors in this apparatus, demonstrating that mice treat this admittedly contrived scenario as an enclosure that they can retreat into if they wanted to escape. They also emerge or egress when they're given stimuli signaling that rewards are possible in the environment. So one such behavior that was replicated in the burrow was escape from a looming stimulus. And so mice that are presented a looming stimulus that's going to be on the top will tend to escape to a shelter if there is one available. So this mouse is exploring, and then the stimulus comes on, runs into the burrow. Okay. So the behavioral paradigm that we used involves 4 odor stimuli. In a separate chamber, 2 of these odors have been paired with a foot shock, so the CS +1 and +2, while the other 2 were not paired with the shock, so CS -1 and -2. And the CS- is control for the specificity of the learned response. And we're using 2 of each CS type to be able to distinguish odor identity from the meaning of the odors. So, and mice largely responded similarly to odors of the same meaning, so I'll group them for the rest of the talk. And we test the ability of the mice to discriminate between the 2 odors, or the 4 odors, in the virtual burrow assay the next day. And here's an example of trials from the testing day of the CS- and the CS+. So this is a CS-, and the odor stimulus comes on here, it's indicated by this white box. So the animal kind of moves the burrow a little bit, not too much. This is the response to the CS+. Stimulus again comes on in the white box. When the stimulus comes on the mouse, you see the squinching of the eyes and this pulling of the enclosure over itself. All right, so this is the displacement data from this laser sensor that we have on the side of the burrow. And this is displacement data from one example trial. And the displacement of the burrow is plotted on the y-axis times on the x-axis. The odor stimulus comes on at 0 seconds and lasts for 8 seconds. Now, the beauty of the burrow assay is that it allows us to capture fine-grained details about the animal's movements. And if we take a look at other example trials, we see that there's a diversity of movement patterns. Sometimes there's not a response. This is all to the CS+. Sometimes there's not a response. Sometimes the response kind of goes up, then comes back down again. Sometimes we have these beautiful sustained responses. But other times we have, a response that kind of got up late in the trial and is somewhat sustained, but not very. So we wanted to quantify this ingress response, which we think is akin to the escape response in freely moving animals. And so first we compare the ingress on each trial to the max possible displacement in that mouse. So it's an idealized step function displacement of the burrow. And we calculate an ingress score, which is the ratio of the actual ingress on that trial to this ideal ingress. And so on this particular trial, the ingress score is quite high, it was 0.9. 1 is this perfect ingress, and 0 is no ingress. These are a few other example trials. So we get a variety. This one in the center is a mouse starts to ingress and then is kind of shaky and then actually ingresses to this max displacement. So from the active ingress response, we have a couple of features, the onset time of the ingress, and then also this ingress score or magnitude. But we also then quantified another behavior that had been noted by our colleagues in the original paper presenting the virtual burrow. And this is this slight oscillatory movement of the burrow. Hopefully you can see my pointer, of the burrow that they called tremble. And we think this is akin to a freezing behavior. And to quantify the tremble, we computed the continuous wavelet transformation of the displacement in this period of time, which yielded a 4 to 10 hertz oscillation of varying magnitude. And here are a couple examples, trials showing both ingress on the top and the tremble. The tremble quantification on the bottom. And note that you can have a trial where there is tremble, actually quite nice tremble response, but no ingress. And there were also some, not shown here, that had ingress but no tremble, right? So another feature that we looked for tremble was the, in addition to the magnitude, was the duration of the tremble response. So overall, and this is all the animals across all the trials for the CS plus on the left and the CS minus on the right, we're looking at ingress responses on the top and the tremble below. We observed that for both tremble and ingress, both of them were more pronounced during the CS plus as compared to the CS minus. And these are the same data, all mice, all trials, but now looking at the various features that I had talked about for both ingress and tremble. And so we observed that ingress magnitude is higher in the CS plus, so this extra hump in the distribution, than the CS minus. You also have shorter latency to ingress in the CS plus and the CS minus. And similar to for the tremble, a longer duration of tremble, higher magnitude of tremble to the CS plus and the CS minus. And not showing here, but both tremble and ingress actually behaviors extinguish over multiple deliveries of the CS plus. So there are similarities between both active and passive responses. Are they just completely related to each other? And I already showed you an example where this wasn't true, where we saw a tremble but no ingress. But what about overall? So if we plot tremble against the ingress, we see that across trials are actually not related. This is also the case across all mice. So this is an average across all trials for each mouse. This is for CS pluses only, where there's no correlation. So the best fit line is in this gray here. And this all indicates that there may be something distinct about these two behaviors. And if we cluster the trials based on features, we see different combinations. So we see that there is, on some trials, strong ingress, very little tremble. Other times there's both strong ingress and tremble. Sometimes there's just strong tremble. And then you get a lot of non-responses, probably during the later trials of the CS plus. And in general, not too many responses to the CS plus. Interestingly, when they do respond to the sorry, I said CS minus. When they do respond to the CS minus, they actually more likely respond with a tremble rather than this more active ingress. And then finally, the amygdala, the brain region that I'm imaging in, it's been shown to be involved in both active and passive responses in freely moving animals. And so we did a bilateral inactivation using dreads and looked at the effect on both the ingress and the tremble in the burrow. And we see that both are impaired. So you get this reduction in the magnitude of the ingress and the tremble. And so in conclusion, the virtual burrow mice display both amygdala dependent, passive or tremble, and active ingress responses. And these behavior responses have some similarities, but they're also distinct and perhaps reflect the mouse, the more complex response that we might see in freely moving animals, despite the reduced preparation that we're using. And I guess I can take questions. Hopefully I'm okay on time. I didn't get a alarm from you, Mengsen.
You're very on time.
Great.
You still have 35 seconds for your talk, so I'm gonna pause this. And, okay, great talk. I think it's very interesting. I already learned a little bit about fear response, tremble, and ingress. A lot of these are very new terms for me. I'm going to chit chat with you while we're waiting to see if we have any questions coming in.
Okay.
For me, it's kind of interesting because I study rhythmic behavior. So when you show tremble, for me, that's rhythmic behavior. I think that's quite interesting. But for me, I would probably look at a different variables than you are. I may not necessarily look at the duration as the most interesting. I may look at the phase and frequency. I don't know how this behavior, how complex the details of the behavior is of the tremble, and if that's going to predict the ingress behavior or something.
Yeah, so we're trying to get into that kind of analysis now. It's early days in that analysis, but one thing that you said that was that reminded me that I wanted to mention this is that something else that we're doing is to look at more kind of autonomic responses that might be related to what this like oscillatory, movement is in the burrow. And so we're now recording the breathing, the respiration, to see if these responses might be, and people have described that you have these changes in these kinds of responses in the freely moving animal when they're freezing. So, but yeah, both kind of behaviorally and then also analytically, we're trying to tease out, what the distinction is between both the tremble and ingress, but then also more details about each of the behaviors.
Yep, thank you. We have a question coming in. Jennifer asked that, what do you think is driving the occasional tremble response to the CS minus? Is it an error in discrimination or something else?
Yeah, I mean, that's an interesting question. I mean, it could be. And so, we haven't carefully looked at when in the trial progression that we're seeing this tremble to the CS minus. So perhaps in the beginning when an animal is more in like a generally anxious state, right? When they're first getting the CS pluses, which are of course interleaved with the CS minus, they might be more likely to mistake a CS minus for a CS plus, and then after they've gone through the extinction, less likely. So yeah, you're right. I mean, it could be that it's an error.
All right, thank you very much. We're just on time to move on to Alexander, and I will introduce Alexander while he's putting up the slides. And Alexander Heimel, and he's a group leader at Netherlands Institute for Neuroscience in Amsterdam. And he's been studying visual system of rodents, and he's recorded from a visual cortex, thalamus, and the eye of mice and squirrels. So recently, he's been focusing on investigating the role of superior colliculus and innate visual behaviors of mice. And please remember to ask questions in the Q&A, and I will moderate them after his talk. All right, Alexander, the floor is yours.
Okay, thanks a lot, Mengsen, for the introduction. I'm talking to you all from the Netherlands, so it's 10:30 here this evening. So I'm discussing work done in my lab by mainly by Azadeh Tafreshia, that you see here, together with Sven van den Burg, Kato Smits, and Laila Bleumer. And it's work building on previous work by these famous two ethologists, Nico Tinbergen and Konrad Lorenz. And they were some of the earliest researchers working on visual innate behavior. And that's what I'm going to talk about. But while my talk will be on visual innate behavior in mice, they worked on many animals, but much of their most famous work is done on birds. In particular, I'm talking a bit about the experiment that they did inspired by Oscar Heinroth's proposal that birds respond to birds of prey in the sky. So what they did is an experiment just before the in the 1930s, where they had cardboard silhouettes. They drew over in the sky over turkey chicks, and then they would respond with alarm calls. And then Timberken and Lawrence noted that they would raise alarm calls to silhouettes of birds of prey, like you see here and here, but they would not respond to geese or seagulls or non-birds of prey. So especially Nico Timberken then hypothesized that there's an innate template, a genetic, that they wouldn't have used the word genetic at that time perhaps, but that there would be a template for short-necked birds of prey. So I'm leaving you with this question for a little while. I'll come back to it later. So similar experiments now started to be done, just like we saw in the last talk, where there were looming stimuli. Also the lab of Sam Solomon and Franceschi, they showed stimuli like this, where you see the mouse just exploring a new environment, is happily exploring, and then you'll see a disk appear here. And if you noticed, so the video will repeat, initially the mouse freezes, and then when actually the stimulus is passed, it runs, escapes to safety to the nest. So here again, you see it freeze and it runs away. So here it's a slightly different setting. Now you see it from below, so it has a transparent floor. You see a hawk silhouette moving across the sky, and you see the mouse freeze or pause. So here it's on repeat. So you see the silhouette, it freezes and then continues what he's doing. So we see that mice, just like these turkey chicks 80 years ago, respond to these birds of prey or silhouettes, but actually already we see now that mice do not have an innate template for raptors or for birds of prey, because they would freeze to the hawk, like we saw in the last video, but they would also freeze to discs, which do not very much look like a hawk. So and this is they freeze about half of the sessions for both of these stimuli. So, but what you notice if you do this repeatedly, so in the first session of 20 stimuli, you see a lot of freezing. So the freezing is indicated here by a period of red. The time is on the x-axis. So the first appearance actually didn't freeze. The second one it froze. But you see over time, over 100 trials, you see less and less freezing. So they habituate to these stimuli. And that was for a single example mice. Here for each color is a different mouse. So you see it's a very generic behavior. So some mice freeze a lot, but over sessions they start freezing less. Some mice already initially freeze a bit less, but again they start freezing less and less. So they really habituate in this freezing behavior to overlap stimuli. And this is to the disc stimulus, and you could think, oh, the disc stimulus, they know this is not a bird of prey, so the habituation is specifically for non-bird stimuli. But you see exactly the same pattern. Initially, they freeze a lot to the hawk silhouette, but then they freeze less and less over different sessions. So already we know that these mice do not have a template for predator silhouettes. But now the interesting thing is, if you've shown this hawk now for 5 sessions, and then in the 6th session you suddenly also start showing a disc, which is novel to these mice, and suddenly they freeze a lot again. And then over the next sessions, they start freezing more, freezing less. So they really habituate specifically to this hawk in this case. Vice versa, also, if you would have started with the disc, you would get a lot of freezing in the first session, indicated here on the right column. But after 5 sessions, they've habituated. But then in the novel stimulus, in the 6th session, when you give another stimulus, they would respond again. So the habituation is really stimulus specific. And actually, later on after the original experiment by Timberken and And Lawrence also, people showed that for these turkey chicks, there was no innate template. But what had happened there, that these were not naive turkey chicks. These turkey chicks that they used were chicks that had been out on the Bavaria farmlands, where they had seen a lot of geese passing over, while there were much fewer birds of prey. So what we see here is not a genetic template, but stimulus-specific habituation. So, but this is still very interesting because it can help us now to probe. So the resolving power of the visual system. So how can a mouse or a chick tell these stimuli apart? But it also helps us to understand how scale invariance and translation and rotation invariance come about, because they haven't seen the stimulus in every position in every possible way. So can they generalize? Can they generalize over location? Do they need to see it on the left side? And then do they only habituate to the stimuli on the left side? Or will they also have habituated to stimuli on the right side? So the first question, so is this translation invariance? Not going into much detail here, but here you see over 5 sessions. So you see where it's actually you saw the stimulus. So we're looking at the mouse from above, and the gray points are where there was a stimulus, and the red points is where it's freezing. So it looks sort of covering the whole sky. If it's perfectly, if it's only habituating to specific locations, then you would not see crosses, and then you would really nicely cover the whole area. But we don't see any location specific habituation. So it really seems there's a lot of translation invariance. So it suggests it's quite deeper in the visual system where you get the habituation. So this is quantified by, if we look at the number of free starts that happen in a sector in the sky where it already froze to before, it's really not different from the real data from when we just shuffle all the data. I can go into a bit more detail later if there's a question about it. What I wanted to go to now is, so what makes these stimuli different for these mice? So of course, these were the 2 stimuli that we initially used. So quite a large disk, and so these are both in scale, and this is the hawk. So of course, there's a lot of detail in the hawk that's not in the disk. So is it the wing tips and the tail, or is it the surface area? Just the disk is much larger, how we can distinguish. Or is it the more the coarse shape? So the disk is, of course, perfectly the same dimensions in both X and Y, while the hawk has more an eccentricity. So this we started investigating. So the first, what we checked is, so now if we don't take the detailed shape, but just take something roughly of the same eccentricity and surface area of the hawk, can the mice tell these apart? And actually, they cannot tell them apart, at least not in this task. So they habituate to both, but then, and when you start giving a novel stimulus, they still don't freeze. So they don't seem to notice that there's a novel stimulus appearing, at least not in this freezing behavior. This is perhaps not so surprising, because now if you look at the stimulus that we gave, this hawk, and then if you look at a grating at the visual acuity limit of a mouse. So now if you sort of blur this image, like the mouse eye would have blurred it, or at least how the mouse acuity, we blur it to the limit that it just wouldn't see this grating anymore. You also see that the hawk, there's something left definitely, which looks a bit like an oval blob. So it's not so surprising that it doesn't distinguish this ellipse from this hawk. And then we repeated the original experiment with this disk and the ellipse, and then indeed you still see that the mice distinguish this disk from this ellipse. But then these are still different in 2 dimensions. So both the aspect ratio of this ellipse versus this disk, the aspect ratio is different, or the eccentricity, but also the surface area is different. So is it just one of these dimensions that the mice use to distinguish stimuli? So first, let's check the surface area. So if we show both small and large disks, do they see the difference? So actually they don't. So here we either started with the large disk or we started with a small disk. We habituated, but then when we exchange the disk for the large for the small, or the small for the large, they don't seem to see the difference. So there's really a size invariance here. They've seen only one size, but they still do not respond to the other size. So then we thought, oh, it must be the aspect ratio. So let's check the aspect ratio. So keep the surface area the same and change the eccentricity. What happens then? Actually, again, the mice don't distinguish. So they habituate to either stimulus. You introduce the other stimulus, and they still don't respond. So the slightly unsatisfying answer is that this generalization over surface area and over aspect ratio, and indeed a disk does, if you present it somewhere else, not directly above you, would be like an oval. So on the retina, they would have seen the different shapes. Even if you showed the disk, it would have seen an ellipse and vice versa. But it's the combination of the two features in this case, or that makes the different stimuli distinct enough to be told apart by the mice. So what I'm not going into now, but what would be very interesting to know is, so what's the circuitry? We know a bit from previous research that the circuitry will start with the retina, obviously, then probably the superior colliculus makes the first response. You directly gets the input from the retina, and perhaps there is where these shapes are being distinguished. From the superior colliculus, there's a path to the perirectal gray, and then it starts freezing. But there's a lot of other brain areas, specifically the amygdala that we just heard about, that may influence this process, and also the cerebral cortex. And with this, I want to acknowledge again Azade's work, and I'm open for questions.
Thank you so much for the talk. Let's see if there are any questions. Let's see if there's no new questions. I can ask something. I'm learning a lot now about it's so interesting. What I was thinking about is that, of course, the aspect ratio appears to be most important, but I think the natural moving birds of prey will have some other features. For example, I think one that might be scary for the mouse would be looming, or if the size of the circle is actually
definitely, yeah,
that would be scarier than passing over.
No, that's definitely true. And so much of this recent work on visual innate behavior and defensive behavior in mice started with the work of Yilmaz and Meister, specifically on these looming stimuli. Looming stimuli have been used a lot, and they're very effective in getting defensive behavior. Actually, much more effective than these overhead stimuli. But with this looming, it seems to be much more hardwired. So I don't know if there will be much stimulus-specific habituation there. So there is habituation. People see that even for these looming stimuli, if continuously something comes towards you, at some point you just start ignoring it. And also the mice start ignoring it. It might be at their own peril, but there's habituation. But where this habituation is, and if we could use this for asking these questions about scale and translation invariant, it might be more difficult. But it's definitely also an interesting stimulus to look at.
Nice. We have a question coming in. Is there an habituation to the speed of the stimulus?
Yeah, so whether that's specific or not. So that's definitely a good question. We don't know yet. We haven't tested it. I think there's probably is going to be some specificity there, but then probably also again in a range. So, but we haven't tested it here. And for the looming, also there the habituation has not been tested. For the looming, it is definitely specific speeds of looming will get the best response. But if you then also specifically could habituate to one of those speeds, it would be interesting to test. But still, because all these experiments take quite a lot of repetitions, because you think this is really hardwired. You just get the stimulus flying over, and these mice, they freeze, but you notice that they don't freeze 100% of the time. So we don't actually know what's going on there. Do they really miss it? Because sometimes we can see it's in their plain view, and they don't have phobia, so they don't really have to look at it. It just pass. They should see it, but still they don't respond to it. So this variability is very interesting, and there's probably lots of the rest of the brain interacting in this behavior. but it makes the experiments much longer because you have to do more experiments.
That's so cool. We are on time. We still have 4 minutes before we're getting booted out of the room. I'm going to see if anyone else wants to ask a question. I will ask one myself. As I said earlier, I'm interested in rhythmic behavior. So the whether they will respond or remember the for example, frequency of the flapping of the prey, I bet they will be very different across different species. For the
different birds.
Yeah.
Yeah, that's true. Of course, the mice acuity is really poor, and most of the birds of prey, of course, they when they're far high up in the sky, I don't think even we could tell very well how much they flap, and then in the end they circle. So I don't know if they can tell the difference. But there's definitely also innate behavior. There seems to be like itching for, like, if you're interested in this rhythmic behavior. So if they see apparently if they see other mice scratching, they also it's contagious. Just like we see scratching, we also start feeling itchy, or even talking about itches, right? Makes you itch. But mice have this as well, and then it also is by them perhaps seeing some rhythmic motion there done by other mice.
Very interesting. All right, thank you so much for the wonderful talks, and I thank the participants for asking questions. And now we're going to give them a round of applause. You can only hear me, but I'm loud enough. And thank you so much for coming. I hope you enjoy the rest of the conference. And we're going to end here today.
Thanks for hosting.
Yeah, thanks, Meng-Sen. That was great.
All right. See you later.
Bye-bye. Thanks.
Youngning is connecting his audio. Hello, Dina. Can you hear me?
Yeah, I can hear you.
Your name is
I've got video.
Hey, hi, nice to meet you.
Hi.
So it's Dina Goldblatt, right?
Yeah, yep, that's it.
Okay, great.
Cool, thanks.
Hello, everyone.
Hi. So
Hello.
your name should be pronounced Yangning Lu.
Yes, that's me. Thank you.
Did we meet at Cold Spring Harbor?
Yeah, I think so.
The neural connectivity. I think we were in a breakout room.
A few months ago, yeah.
Yeah.
Nice to meet you again. Great.
And one of you can try to put on your slides and share your slides and see if everything goes well.
Okay, so Dina already tried that and it was fine. Yeah.
Okay, good.
So, like share screen?
Yes.
Okay.
Share your slides to check.
Hope you see the screen now?
Yes.
Okay. No. Is it on?
Yeah.
Everything is going?
Yep.
Okay, cool. Should be good. Can you see the animal moving?
Yes.
Okay.
That's
good.
very important.
Okay.
To
great.
see the sea alligator moving.
So it's working.
Awesome. We have one more person. Let me just email her really quick. I was in the last session and the first speaker remembered it on the wrong date. And I was.
Oh my God.
Yeah. And Tess, I'm going to be a no-show. So I have to entertain people for an extended period of time. Let me just email her. All right. I hope everyone knows where their presentation, when their presentation is.
Yeah. It's a little bit hard to keep track on things. There's so many things going on, and yeah.
Yes, the other speaker was like, "Oh, I thought it's Thursday." It's like, okay, fine. So, okay, and if in the rare case of, unfortunate case, if the person doesn't show up, we will keep the original order. So, because people may come in at the precise time that you're presenting just for your talk. So we're gonna keep your schedules constant. And if this person doesn't show up, I will, and we will have a panel discussion. No, so you're on the same boat with me. You have to help me.
Nice.
Great.
That's great. So you will have 12 minutes for your talk, and there are not much buffer. So you have 3 minutes Q&A following your 12 minutes talk. And I will hold up my hand like this. And when you have 5 minutes left in your talk, and this when you have 2 minutes left in your talk. And if I don't see you on your concluding slides in the last minute, I may interrupt you at the end. But hopefully, you know, if I see you're wrapping up, I wouldn't interrupt you at the end. I will let you finish. So that's about it. And let's hope people are, yeah, I have a bad feeling about this.
Who's the other one?
That's Ewan Wang.
Okay, nice.
From University of Chicago.
Joseph Love.
Yes, I thought.
So we organized by talking about lava of different animals.
Yeah, and what is the best way to describe the nature of this this session?
Lava, I think.
That sounds promising.
Yeah.
Neural connectivity.
Okay, so I'm going to start the webinar so that people have a couple of minutes to enter.
Okay.
Okay.
Oh.
Yeah. Arrived.
Hi, Evan.
Sorry. Sorry for this. Yeah. Hi. Hi everyone. Yeah.
We still have 1 minute, and then I will let you set up your slides while I introduce you.
Okay.
We're going to start on time.
Okay. I'm so nervous right now.
Okay.
I like your background.
Thank you. Official one. And I also have some light on my face. I hope it's not bothering anyone. Have we started the webinar?
We have, yes.
Okay.
Still waiting for people to join.
Yeah,
we should begin at 3 o'clock.
Okay, cool. Oh, even I will remind you, you have 12 minutes for your talk and 3 minutes for your Q&A, and I will remind you with my hands gesture when you have 5 and 2 minutes left. So I won't keep my camera on so you can see me. Oh, I have attendees trickling in.
Should we start? It's 3 o'clock.
Okay. I am waiting for more attendees to show up. I'll give us 1 minute.
Okay, yeah.
All right. I think we have some people. Okay, it's 3. I should get started. So hello, everyone. Welcome to this session. And we have really interesting talks lining up today. And I will remind you to keep posting questions in the Q&A session in the bottom of your screen. And you can vote for your questions in the Q&A session. And I will read your questions to the speaker at the end of their talk. And yes, and you can also chit chat in the chat box and to pass on different information about the talk. So our first speaker is Evan Wang. She is a postdoc at University of Chicago, and she is obsessed with understanding nature. And her current research is to decipher how stem cells dictate synaptic wiring decisions of their progenies. And Even you can take it away and tell us about your work.
Okay, yeah, thanks a lot for the introduction. Yeah, so as a neuroscience, I'm very interested in the development point of how the stem cell actually contribute to the partnership of a circuit. So, stem cell, neuron stem cell, we are knowing this is a developmental unit. Neuron stem cell is actually undergoing self-renewal and also the differentiation to produce a mature neuron. They have a dendrite and axon, and is also capable for neuron transmission. So it will have the input into its target. So the connection between the input and also its target is called the synapses. And the number of synapses actually determine the intensity of the neuron transmission signal. However, there's very little known about how the stem cell lineage actually affect this synaptic partner selection. On the other hand, there are a lot of knowledge about how the neuron stem cell are able to generate neuron with wider diversity. It's through the spatial cues and temporal cues. So for spatial cues, we are knowing about these actually used to specify the identity of individual neuron stem cell. So here the diagram shows the lateral view of Drosophila embryo during the neurogenesis. The ventral nerve core here are showing the green bar. And here shows the individual neurogenic unit, which is composed of 30 neuron stem cells. In Drosophila, this is called a neuroblast. So each neurogenic cluster actually undergo a left-right asymmetry and also the segmented pattern along the ventral nerve core. So within this neurogenic unit, the stem cell actually specified by both the column, by the spatial factor that define the column and also define each row. So the combination of these spatial factors actually determine individual stem cell lineage. The lineage here is all the descendant generated from a single neuron stem cell. On the other hand, So on the other hand, the temporal cue further specify the individual lineage. So here is the sample of the neuroblast NB33 in the A1 abdominal segment. So this is our model lineage. So as you can see here, the lineage progression actually generates 2 neurons and then 11 EL, which stands for the event skip lateral interneuron. So the lineage progression is actually coinciding with this temporal transcription factor switch. So the combination of a temporal transcription factor actually specify individual neural identity. So together we use the spatial cues to illustrate the we use a spatial cue to illustrate the stem cell lineage. And also we use the temporal cue to illustrate it by the neural birth timing. So in our lab, we are actually interested in how the spatial and temporal cue actually contribute to the synaptic connectivity in the level of a stem cell. Again, we use a neural NB33 as the lineage model. And first, we characterize all the neuron of NB33 lineage. And then we define the birth timing by the genetic methodology, twin biomarker. So after we characterize each NB33 neuron, it will help to reveal all the synaptic input onto the NB33 neuron by using the connectomics analysis. So the connectomics analysis actually show there are two major inputs. One is the sensory input, and the further characterization of this sensory input actually show the NB33 can be further divided into two different temporal cohorts. The temporal cohort is defined by a set of neurons. They are lineage related, means they are actually coming from the 33 lineage, and they are continuous born. So there are no separation from each other, and they actually have a similar circuit function. Here, it actually means the different sensory input. So the early born temporal cohort is shown in pink here. They only got the cototonal sensory input, while the late born temporal cohort in the purple, they only got the proprioceptive input. So this actually put the temporal cohort are very important, suggesting, so the temporal cohort actually suggesting that temporal regulation is actually important. Okay, on the other hand, the other major input onto the EL is actually ventral nerve core interneuron. And we are actually interested in the ventral nerve core interneuron onto the early born EL only. So the pie chart here shows the composition of the input. It includes the ladders, the early born EL, as well as the basins. The next step, we're actually trying to characterize this neuron and to study their connectivity and also the developmental origin. So for example, here shows the ladder. We actually show the ladder actually from the midline neuroblast. We characterize this by using the ladder D as shown in the green, and also and find out it actually colocalized with a midline neuroblast as shown in the red. It actually suggests the ladders are coming from the midline neuroblast. Previously, there are some initial data about the neuroblast, midline neuroblast lineage progression. So it suggests at the very beginning, it produces a motor neuron, and then continuous born 6 to 7 interneuron. By connectomics data, we know the ladders are actually coming from the same lineage, and also we also find out there are 6 ladders only from A to F. It's actually put the ladders into these spots. So it's likely that the ladders are actually continuous born. So we know the ladders are from the midline neuroblast altogether. It's actually from the same lineage, and they are actually continuous born. So we are interested in this question is whether they are actually coming from the same circuit, whether they are actually contribute to the same circuit. So the assay we are using is we actually study the input of them, and we actually find out there are only cototonal input, but not others. So together, this data suggests the ladders are a temporal cohort of a midline neuroblast lineage. Okay, so the next thing is we're actually interested in how the temporal cohort act. How does the temporal cohort direct the connectivity? How does the temporal cohort interact with each other? So in this case, we actually study further for the connectivity. So the ladders are shown in the orange here from different segments. And then we found out it actually gave an input for the early born ELs in the pink in the A1. So this is a very complicated connectivity data, but we can just simplify by this cartoon. So the temporal cohort of ladders actually wiring to a temporal cohort for early born EL. Okay, so this actually put a temporal cohort this actually put a temporal cohort neural connection unit. In addition, we are actually interested in their developmental origin. By knowing that they are midline neuroblasts, the next question we're asking is whether their birth timing. So one of the two major input for the letters onto the early born ear is D and F. Because we have the availability of the letter D for genetic analysis. So we decide to determine the birth timing of the letter D. The first thing is we actually align the midline neuroblast lineage and also the NB3 lineage. And then the idea is we infer the letter D birth timing from the early born and also the late born ear clonal generation time. So the strategy is we do a short time period of collection sample, and then we incubate for various durations. So it will lead to different neurogenesis stage before the clonal induction. The idea is we can only get a clone if and only if the neuron is actually burned within the duration or after the duration of the clonal induction. So the picture, the data showing here is if we do the incubation time around 7 hours. So if we do the incubation time around 7 hours, we can get the early born EL, late born EL, as well as a later, suggesting this time period is a very early neurogenesis stage. On the other hand, when we incubate to 9 hours, we can only detect the early born EL. We can only detect the late born EL among the EL population, suggesting the early born EL actually, suggesting this time period actually pays the birth timing of the early born EL. And very interesting, till the 10 hours, we can still detect the letter D. So this actually suggesting the letter D is actually born within the duration of late born EL or after the late born EL, as shown in the yellow rectangle here. Okay, so the final point for this slide is actually we find out the input is actually born after the target. Okay, so in addition to it later, we also study the early born EL from other segments as well as the basins to study their connectivity and developmental origin. And here's a summary of the data we have. So we found out together they generate 70% of synapses onto the early born EL. And by the lineage analysis, we found out they are coming from the midline neuroblast, NB3 and NB3 respectively. Because the major input actually coming from these three lineage, it shows the lineage are not equivalent, showing the spatial cue is important. In addition, we also show that all of these three neurons are actually forming a temporal cohort within each lineage. So here, the rectangle bar here is actually showing the neurogenic period, and the temporal cohort showing in the filled box here is actually the time period they spend within this lineage. In addition to the letter D, we know it's actually born after its target, the early born EL. We also find that basin is also actually born after the target, showing maybe the birth timing of input is actually later than the target. It's maybe a general phenomena for all the NB3 lineage. Okay, so together give a conclusion. We think the integration of a spatial and temporal cue actually contribute to the synthetic partnership selection. Yeah, with all this, I will acknowledge my lab and also the McLean lab in our school for the computational analysis, although I didn't show it today. And also, thank you for your attendance, and I'm willing to take any question you may have.
Thank you so much. Very fascinating talk. Let's wait for a minute to see if there's any questions coming in. This is very interesting to me that I didn't know any of this, that it's just so sensitive in space and time. It made me wonder, what do we, how much we don't know about how the brain is actually grown.
Yeah, I think the previous knowledge, the temporal cue is important, and so spatial cue is important, and to specify diversity and also to specify the projection way. But I think our study, is really, I think it's one of the first study actually really study every neuron of the lineage to see their connectivity.
It's really fascinating. I really look forward to see what we can learn more from this. The more the merrier. All right, we are at just on time for the next speaker. Thank you so much, Ewan.
Yeah, thank you.
I will let Adina set up her slides while I introduce her. So Dina Goblad, she's a fourth-year PhD candidate at NYU, the Neuroscience Institute. Her work investigates how to bring examples of complete sensory motor circuit. And her current focus on the mechanisms that specify excitatory vestibular interneuron identity in the larval zebrafish. So Dina, take it away.
Hi, thanks for the introduction. Thanks for coming, everyone. So I'm really excited to share some of my thesis work today on mechanisms that specify excitatory vestibular interneuron identity in the larval zebrafish. So again, I'm really interested in how the brain assembles a complete sensory motor circuit. Now, the problem is relatively straightforward in a 2-neuron circuit when you have just one sensory neuron and motor neuron partner. But of course, it becomes much more complicated as you add more neurons to the circuit, and considerably more so if you have many subtypes of cells that each have to connect to the right sensory neuron and motor neuron partners for proper behavior. And of course, wiring subtypes of cells to the correct partners is a very general developmental problem. And principles by which sensory neurons and motor neurons and either stochastic or interneuron intrinsic signals instruct identity have been well described for just one subtype of cells or wiring across one synapse, but how these mechanisms come together to build a complete circuit has been much more difficult to define. So a very simple example of this general problem is the gaze stabilization reflex. It's highly conserved and it's used by all vertebrates to maintain balance. And of course, it's present in the zebrafish, where we have easy genetic, optical, and developmental access. So the circuit uses just 2 subtypes of cells to transform nose up pitch tilts into downwards eye rotations and vice versa. And it uses just 3 neurons to do this. There's a population of excitatory vestibular interneurons that's indispensable for the behavior. So here I'm first just showing you the nose up subtype. Now these interneurons relay sensed destabilization from afferents and sensory neurons contralaterally to motor neurons that then rotate the eyes down. And of course, there's the other subtype that does the opposite. It relays downwards eye rotation, downwards nose rotations to eyes up motor neurons. And so you can appreciate here, there's 2 developmental problems. The circuit needs to specify up and down interneuron identity, but then it also needs to wire interneurons to the correct up sensors and down motor neurons. And of course, if you have developmental miswiring where up goes to up instead of to down, it would be very hard to maintain your balance. So how does the gaze stabilization circuit come together? Before you can wire 3 neurons, you first need to define nose up and nose down vestibular interneuron subtypes. Now, in most studies, identity is defined molecularly, but because we don't have molecular markers that define excitatory vestibular interneuron subtypes, I needed a functional assay to measure the identity of these cells and to test mechanisms that could instruct their identity. We do have an enhancer trap line that labels these cells. So this is a sagittal view. There's about 45 neurons in each hemisphere, and the entire nucleus is only about 40 microns in width and depth. And of course, I can express a calcium indicator in these neurons to measure their functional identity in response to pitch tilts. Now, previous studies have only recorded from single vestibular neurons at a time, but I wanted to record from the entire nucleus. And to do this, I needed a volumetric imaging paradigm. And so to do this, I've been using a single objective light sheet microscope called SCAPE, which we've built in our lab with the help of Elizabeth Hillman's lab at Columbia. Now, I won't get into the technical features, but very simply, it can perform volumetric imaging at very high speeds. And so I can mount a fish on a galvo under the SCAPE objective, deliver a pitch tilt vestibular stimulus, and then measure the functional responses of the entire nucleus simultaneously. Okay, so I needed a stimulus that would allow me to elicit reliable directional responses to probe nose up and nose down identity. And to do this, with the help of Kyla, another grad student in the lab, we designed a simple step stimulus. So we start by imaging the fish at a horizontal baseline position. We then step the fish down in a 19-degree tilt, and we hold it there for 15 seconds to allow the cells to reach steady state, and then back up into the imaging plane. And of course, I can repeat this for a nose up tilt to see the nose up response. Now, because this happens so quickly, I can measure the calcium responses to their previous orientations, and you can see this here in the shaded bars. Now, there are some limitations to our stimulus, but for the purpose of differentiating interneurons that respond to up and down sensations, the step stimulus is a simple and appropriate way of doing so. So these are some example traces of responses from a nose up and a nose down cell. I'm showing you the mean DFF response over the course of the entire trial. I've highlighted in pink and green the time of the nose down and the nose up stimuli in shaded bars, and I've clipped in yellow here the time the fish is tilted away. There are some really interesting properties that we see from these responses that I don't have time to get into, but I do want to highlight here that our stimulus is successful. We can see a response from the cell, the response is stable, and we can clearly identify its preferred direction. I can then pseudo-color cells according to their binary nose up or nose down preference and overlay that with an anatomy scan to generate a spatial map of up and down cells. So here I'm showing you a maximum intensity projection in an axial view, and what you can see here is a very striking sandwich organization along the rostrocaudal axis. So you can see that nose down cells are on the extreme ends in magenta, and nose up cells in green are in the middle. Now, when I rotate this into a sagittal view, the organization becomes even more apparent along both the rostrocaudal and the dorsoventral axes. And so the skate predicts that there's essentially microtopography, which we define as an orderly arrangement of cells at length scales roughly comparable to an individual cells. Again, the whole nucleus is only about 40 microns. And this is really exciting because functional subtype organization has never previously been shown in a vestibular brainstem nucleus. Now, to test this prediction with finer cellular resolution, I took a multiphoton imaging approach. And I was actually able to replicate the same rostrocaudal and dorsoventral microtopography. So I'm showing you here, this is a heat map representing the probability of observing a nose up or a nose down cell in each grid space. And again, this is the same sagittal view. Now, what you'll see is the borders are a little fuzzy, but the microtopography is still very clear. I also want to note we're still working on a statistical analysis to measure how fuzzy or not fuzzy the borders are, and I'd welcome any ideas for how we can go about this. So to summarize this first part, we see microtopographic organization that suggests a possible role for spatiotemporal identity programs. And this hypothesis is further supported from preliminary data I don't have time to show today, but what I found is that early born cells are exclusively nose up, and this suggests that developmental subtype segregation is in both space and time. So there's one other hypothesis that I needed to address, and it's the role of motor neurons in specifying identity. Of course, it's preposterous to think that motor neurons could play a role given how much we know from sensory activity deprivation studies, but in our circuit, the leading hypothesis is that motor neurons are active specifiers of identity based on observations that interneurons wait to synapse onto motor neurons until motor neurons synapse onto muscles. So I wanted to test this role of motor neurons, and to do so, I replicated a previously published mutation in the FOX2A gene, which is selectively expressed in these motor neurons. So here I targeted guide RNA fairly upstream in the FOX2A gene, and I have two mutations, a 2 base pair insertion and a 22 base pair deletion, and they both cause identical truncated proteins. Now, these fish don't inflate their swim bladders, that's this white arrow here, and the swim bladders are necessary for buoyancy, and because of this, the homozygous mutants only live for about a week, but otherwise, the fish are grossly morphologically normal, and the mutation isn't embryologically lethal. So FOX2A is upstream of the reporter islet 1, which is a general marker for motor neurons. And so I can measure the extent of FOX2A knockouts on the motor neurons using an islet 1 GFP reporter. And I'm showing you here an image of the entire nucleus in a wild type or a sibling fish. And this is what it looks like in a FOX2A knockout. You'll notice there are a few cells remaining. It's an incomplete effect. Our best guess is that these cells are FOX2B positive. But I can quantify the number of remaining cells in each fish. And you can see that it's about 95% of islet 1 positive motor neurons that are eliminated in my mutant. And I do occasionally see a complete knockout effect. So now I can take my same calcium imaging setup, I can give the same stimulus I've shown was effective before, and I can analyze the properties of this population. If there's no difference, it argues against a role for FOX2A in specifying functional nose up and nose down identity. So I first asked whether the ratio of up and down cells had changed, which would of course affect the motor neuron's ability to interpret pitch tilt destabilization. So in sibling controls, I see just about a 1-to-1 ratio of up to down cells. And I see that that ratio is largely preserved in mutants, suggesting that at the very least, motor neurons aren't necessary to specify a binary up or down identity. But I wanted to ask, well, is it possible the mutation changed something about the intrinsic properties that allow these neurons to convey destabilization? So I'll highlight just 2 metrics here that I tested. The first thing I wanted to ask was whether these neurons retain their sensitivity to destabilization. And so I looked at the distribution of peak response amplitudes. And I see no difference between my sibling controls here in black and my FOX2A mutants in red, suggesting that these cells are just as sensitive to pitch tilt sensations. So I then wanted to ask whether there's a change in the temporal dynamics of the cell's response. And so here I fit a 2-term exponential to the response and asked whether the decay coefficients changed. And again, I saw no change in the temporal dynamics of the response, the decay of the response. And so I'll lastly note that while my knockout only eliminates 95% of cells, I did have 2 fish included here that had a complete knockout, and their functional responses looked no different. So in conclusion, my data largely suggests that motor neurons are not required to specify vestibular interneuron identity. And so I can eliminate the leading hypothesis in the vestibular field that a retrograde motor neuron-derived cue assembles this circuit. So I'm really excited to follow up in the next part of my project on a spatial factor that might specify identity. And I'm going to do this with both a single cell and a bulk sequencing approach. I'll sequence the interneurons at time points I've identified for identity specification. And this will be a really unbiased approach to identifying transcription factors that define subtype identity. And of course, I'm really excited to test the role of sensory neurons. I'm taking a similar constitutive knockout and silencing approach to test the role. And I'm designing a similar comparable optogenetic assay to measure changes in interneuron and motor neuron identity, because obviously I can't use my same calcium imaging setup if I don't have sensory neurons to convey pitch tilt information. And with these experiments, I'm really excited to further probe genetic mechanisms that specify identity, as well as downstream molecular factors for multi-synaptic wiring specificity. And so I just want to say thank you to both the Chopek lab and the NAGLE lab, especially Kyla, Paige, and David, who put a lot of work into designing the stimulus setup. The Hillman lab, who's helped us build our scape here at NYU, my thesis committee, and of course my funding. And thank you for your attention.
Thank you for the talk. It's really nice. Let's see if we have questions coming in. Please ask questions. Please post your questions to the Q&A and vote for them. While we're waiting for the questions, I will ask you something.
Sure.
So you were showing us that the distribution of these nose up and down cells. So I think it was the nose down cells were sort of have 2 blobs and the nose up one blob. I'm wondering why is that there is such a topographic organization?
Yeah. So my best guess, there's been evidence for rostrocaudal subtype organization in other circuits, such as spinal circuits. My current hypothesis is that it's Hox derived, some kind of Hox transcription factor or a code of Hox factors organizes cells along this gradient. And that could explain why we see these clusters of down cells on the extreme rostrocaudal ends and then up in the middle. And there is also evidence for microtopography in some Drosophila circuits.
That's pretty cool. I'm slightly disappointed that the motor neurons didn't matter because I study motor coordination. Yeah. But I will have to leave with it.
Yeah.
Okay, I think it's time. If there's no questions, it's time for us to move on to the next speaker. thanks, Dina, again. I will clap for everyone. And I will let Yangning set up his slides. So Yangning Liu is a postdoc at MIT and Howard Hughes Medical Institute. And during his doctoral studies, he worked with C. elegans neurocircuitry and showed the importance of extrasynaptic transmission and neural multitasking in circuit function. That's what he will be showing us today. And now he is a postdoc. He is developing novel tools that will facilitate the study of neurocircuits in general. So the floor is yours, Yangning.
Thank you, Monson, for the nice introduction, and really appreciate this very nice conference to have me. So what I'm going to talk about today is how symmetric behavior is generated from asymmetric wiring. So in most animals, particularly bilaterians, there are distinct locomotion forms, mostly composed of undulation or a mixture of undulation plus limb movements or simply limb movements. And one of the key hallmarks of these behaviors is the left-right symmetry. Underlying the left-right symmetry is the left-right symmetry of the neurons that control this behavior. Take, for example, in the limb-free locomotor center, there are excitatory motor neurons that activate left and right sides equally. In between them, there are commissure neurons that relay the cross inhibition. So that these two left-right locomotor centers can coordinate antagonizing each other and at the same time generate an equal level of locomotor behaviors on left and right sides. In the first lobal stage of C. elegans, which is the youngest lobal stage of C. elegans, it's generating this very balanced sinusoidal movement on its dorsal and ventral sides. So in C. elegans, the animal cross on its lateral sides, and by generating bends on dorsal and ventral muscles. So basically dorsal and ventral sides in C. elegans can be considered as left and right in most bilaterians. This is not very surprising and very normal. However, if you look at its locomotor center, it's very surprising that it's composed of a highly polarized dorsal-ventral wiring system. Specifically, it's composed of excitatory premotor interneurons that synapse onto excitatory motor neurons, which is acetylcholine, a releasing neuron, that only activate synapses onto the dorsal muscle. At the same time, it innervates the GABAergic neuron that only innervates the ventral muscle. So basically with this wiring diagram, you don't expect the behavior to be dorsal and ventral symmetric at all. You expect the dorsal muscle to be contracting all the time, whereas the ventral muscle relaxes all the time. However, as you just saw, this is absolutely not the case. So curious about how the muscles actually behave during the L1, the first lobal stage C. elegans locomotion. We used the genetically encoded calcium indicator GCAMP and expressed it in the muscles with an RFP as the ratiometric indicator. The ratio of these two would tell us how the muscles change its activity during the locomotion. So we developed segmentation algorithms to divide the animal into all these segments, and then we can accurately quantify how the activity is behaving during the locomotion. So remember, the motor circuit that I just mentioned only controls the posterior body wall muscle, which is the 65% of its body on the posterior side. The head muscle is controlled by a totally different system, which is symmetric. And when we quantify the muscle activity propagations, we can see clearly that the dorsal muscle, as well as the ventral muscles, they output roughly equal amount of total activity for the body wall muscles. It's only the pattern that overlaps, that antagonize each other. The overall level of output is completely equal. And that's consistent with what we saw from our naked eyes, that yes, it's producing a sinusoidal movement with no bias. So the question becomes, how does a structurally asymmetric circuit generate symmetric locomotor output? So to answer this question, people in my previous lab developed high throughput electron microscopy reconstruction strategies, and they actually identified additional cells that were previously uncharacterized from the first EM study done in 1978. They actually identified additional excitatory neurons that innervate dorsal and ventral muscles, as well as another cell called HMC, which is not a neuron, but also has gap junctions with dorsal and ventral muscles. So together, this is the wiring diagram we have, but still it doesn't look very symmetric either. And we want to address how the symmetric behavior activity comes from. Over the past 40 years, actually, people have generated various hypotheses regarding this question. For example, many people suggested that the ventral muscle on L1 C elements might be intrinsically active, and it's more has more activity over the dorsal side. Or maybe the ventral muscle is proprioceptive and can be activated by sensing dorsal bending. Or that the GABA released on the ventral side might be excitatory actually to the ventral muscle. Or that the GABAergic motor neurons can co-release unknown excitatory neurotransmitters. Or that there are simply more excitatory motor neurons, which our colleagues discovered from the EM reconstruction. Or that based on the new EM reconstruction, the HMC cell might relay the dorsal muscle activity to the ventral muscles, or maybe some other, which we don't know about. So to tackle all these hypotheses, we established two scenarios where we first established a correlation between these cell activities with respect to the locomotion. And second, we established the causal relationship between these cells' activities and muscle activities. For the first approach, we simply used calcium imaging during free moving animals. And take, for example, where in this particular animal, we label one type of motor neurons. And this type of motor neurons are activating only during the backward locomotion, which is highlighted in the right panels. And because of their anatomical wirings with respect to the body curvature change, we can establish that this particular type of cells correlate with dorsal bending during backward locomotions. So this is the correlation that, strategy that we used. For the causal relationship establishment, we used the simultaneous manipulation and monitor of the circuits. So to do that, we used optogenetics, which is widely used nowadays, and couple it with calcium imaging. One of the well-known problems with this optical interrogation approaches is that there are spectral overlaps between opsins as well as the G-camps, for example. That means that when you turn on the light, you're already activating the cells, and you have no reference as to how the G-camp looks like before the activation takes place. So people have largely used two approaches to address this problem. One is to use spectrally shifted opsins and couple it with G-camps, for example. And another is to use spatially patterned light to basically specifically target cells of interest, and then without touching cells on outside of the cells of interest. However, these two approaches are both requiring technical challenges, and we probably want to make new animals if you want to do that. To take advantage of the fact that the G-camp actually has a delay with respect to the onset of the cell activity, we reasoned that we don't need anything complicated like that. So imagine how the cell looks like pre-stimulation, during stimulation, post-stimulation. Ideally, it looks like this. What the G-camp will look like would be something like this, the red line. That means by the time you turn on the cell, you activate the cell, the G-camp doesn't have enough time to reach its peak value. It takes about 200 milliseconds to reach its full length, full peak value. And therefore, by only recording and illuminating at the same time, activating the opsin at the same time, the first data point you acquired is reflecting what happened before stimulation. And at the end of the simulation, you're going to see what the cell looks like after the simulation. And therefore, we only need to record and illuminate the opsons at the same time. And by capturing the first few seconds or a few hundred milliseconds, you have a reference point already. And that approach is very inclusive. That approach doesn't care which particular opson, which particular wavelengths you use at all. So using this approach, we basically spent 3 years and concluded nicely that none of these hypotheses are valid. And it's including the fact that the muscles may be autonomously active. We also excluded this hypothesis either. Okay, so this is very unfortunate. Moreover, there's one thing that's left, which we never touched, which is the excitatory premotor interneurons. Okay, this type of premotor interneurons, in C. elegans, there are only 5 of them. And 2 of them control the forward locomotion, 3 of them control the backward locomotion. And interestingly, all these premotor interneurons have this long process that lies along the ventral nerve cord. It's actually in proximity towards the ventral muscle cells. And all of them are cholinergic. All right, so this actually set up the realm of our hypothesis that actually maybe, just a maybe, that these premotor interneurons might be underlying the ventral muscle excitability. So to test this hypothesis, we used the optical interrogation strategy, which we described, and expressed general opsin in one of those premotor interneurons. And to avoid the GABA synapse releasing that might be complicating our results, we used a mutant that doesn't release GABA, that doesn't synthesize GABA, so that we can actually purely see what would happen when we activate the premotor interneurons. So to our surprise, yes, it's actually activating the ventral muscles by simply activating the premotor interneurons. And as you can see on the ventral side, the activities are increased upon activation of one of those premotor interneurons. And this is highly satisfying, that this is the first time we can actually get a neuron that's activating only the ventral side. And on contrary, when we optogenetically silenced the premotor interneurons altogether and recruited the muscle cells, we saw that, yes, the ventral, as well as actually the dorsal cell muscles, are inhibited. So this is sufficiency and necessity. However, both of these approaches are optical interrogations. And we wanted to validate our results by other approaches. So we simply used a light-sensitive protein that when activated can ablate cells where it's expressed. And using this, after we ablate all the premotor interneurons, what we saw is actually very satisfying, where that we can only see the remaining of the dorsal activity, but the ventral muscle activity is pretty much gone. And the fact that we can see the dorsal activity is consistent with our previous findings, where upon ablation of premotor interneurons, the excitatory motor neurons are central pattern generators, where they can generate activities that activates the dorsal side. So this is consistent with our previous finding for the central pattern generators. What's really unexpected is that we saw a loss of ventral muscle activity in this approach. And when we saw
you have to wrap up now.
Okay, very fast. So when you look at the behavior, you can see that the dorsal side is always quiet. So in conclusion, so additional experiment where we spread out the calling, we actually saw the muscle activity being expanded only on the ventral side. And this is highly satisfying as well. So in conclusion, we found that the premotor interneurons have dual roles, that it's not only the premotor interneurons, actually motor neuron by themselves, and that extracellular transmission compensates this structural asymmetry. And by additional stories, we also found that the GABA is also acting upon the excitatory motor neurons, so that we have a balanced outputs on both sides. In conclusion, we have basically identified the extracellular transmission that was previously unknown from the premotor interneurons that activates the ventral muscle cells in L1C elegans, and that by negative feedback, you can form a highly symmetric but structurally complementary shape of dorsal and ventral muscle cells activity patterns, which is what you can actually achieve by a symmetric wiring, but they take advantage of the asymmetry here. So with that, I'd like to thank all the lab members from my previous PhD study and all the funding sources, collaborators, committees. And with that, I'd like to take questions. Thank you.
Thank you so much for the amazing talk. Let's see if there's any questions. This session, encourage people to ask questions. For me, this is quite amazing. I was wondering if I guess you're still in the stage. This field is still in the stage of figuring out what are the neurons or how they're connected. I was thinking I'm a computational neuroscientist, so I think more in terms of if anyone has done the modeling of this stuff. Even with very simple neurons, their behavior could be quite complicated, and they will be able to generate rhythmic pattern despite there may not be very complicated wiring. So what are the major perspective on that?
Yeah, it's a very nice question. Yeah, so actually, we are trying to model how the system works, and but of course there are a lot of parameters which we would have to fit to actually get the results similar to what we saw. But yes, there are efforts currently being done to try to simulate this the entire system, and it looks like this system is more complicated than we thought. But the fact that it's actually using fewer, only about half of the neurons where the adult C. elegans uses, it's still able to generate similar behavior, and this is really inspiring to us because it looks like it is possible that even with a loss as much as half of the neurons, it is still possible for you to converge onto the behavior that that's also implying that the loss of motor neurons in some of those diseases, neurodegenerative diseases, those models, it is possible for those systems to adapt similar strategies to achieve functional outputs at the similar to the wild type. Yeah, but like your question is, yes, the efforts are being done, but we haven't actually finished this non-trivial task. Which
looking forward to any upcoming studies.
Yes.
So it looks like we're at the end of this session, and I want to thank all the speakers, and I want to thank myself for asking questions, and thank you so much for presenting amazing work. I hope you enjoy the rest of the conference, and I hope to see you around at the conference. And we're good.
Perfect. Okay, there we are. Okay, everyone. Welcome back, and I'd like to take this opportunity to announce an excellent keynote presentation by a former lab mate of mine, Lisa Giacomo, who she worked in the same lab that I did back with Mike Hasselmo a long time ago. And she went on then to work in the Mosier lab, where she learned quite a lot more about grid cells and place cells. And she is now running her own lab, has been for a while at Stanford University. And so she's been doing a lot of really fantastic electrophysiological and also computational work on understanding how the rodent mind builds maps of space and other aspects of the environment. So I'm going to let her go ahead and take it away. Thanks, Lisa.
Great. Thanks, Brad. Thanks for the invitation to participate. I'm really excited to be a part of NeuroMatch this year. Let me just share my screen. Okay. So my lab studies our sense of location and how we use this sense of location to learn about and flexibly navigate through the external world. So one example, decades of research have indicated that the same brain regions that are critical for memory are also critical for navigation. And I think one of the behaviors that maybe best exemplifies this is parking your car in a parking lot, going into school or work or a store, and then later having to exit that context and navigate accurately back to your parked car. What this really requires is that the brain builds some sort of internal neural map of our external spatial world. And in order to do this, the brain has to combine multiple different complex cues. So for example, in order to accurately perform this behavior, you need to be able to bring online some sort of memory of where you parked your car, which can be a sort of multimodal or multisensory representation. As you navigate towards your car, you need to have information about the landmarks that are around you, the relative distances between those landmarks, as well as the distances between those landmarks and yourself. You need information about the direction that you're traveling. Are you moving towards your car or away from your car? And you need information about the distance that you've actually traveled. So how does the brain accomplish this? Well, over the last decade or so of work, researchers have really revealed that all of the neural substrates that are needed to build this internal map of the external world are located primarily in a brain region called the medial entorhinal cortex, which is shown here in blue in the rodent. The first of these cells are called grid cells, and these cells have been hypothesized to provide a type of universal neural metric or spatial metric that allows us to navigate through space. This is an example of a grid cell shown here. What you're looking at is the activity pattern of the grid cell from a bird's eye view after a mouse has explored a 1 by 1 meter open arena, where blue colors indicate spatial positions where the cell is not active, and red and yellow colors indicate spatial positions where the cell was highly active. You can see even just by eye that in many ways this pattern is reminiscent of a longitude and latitude coordinate system, only instead of a square grid covering a map, which is what we're used to looking at, say on Google Maps, the unit of the grid is an equilateral triangle. Now, there are two features of grid cells that make them highly efficient at encoding your position in space. The first is that different grid cells have different spatial phases. So this is just an illustration of that, where you have two different grid cells that have the same underlying geometric hexagonal structure in their firing pattern, but they're slightly shifted or offset from each other in space. And what that means is that no matter where you are in space, no matter where the rodent is in space, there's at least a subset of grid cells that are active. The second is that grid cells come in multiple different spatial resolutions. So this is a sagittal slice of a rodent brain. Entorhinal cortex is this structure here in the back. And you can see that in the dorsal part of entorhinal cortex, the distance between these grid firing nodes is small. It's about 20 or 30 centimeters. And as you move to more ventral portions of entorhinal cortex, the distance between these firing nodes increases to a meter and sometimes even several meters in the most ventral portion. What this means is that you can actually combine the activity pattern of a relatively small number of grid cells and accurately decode or estimate the position of the animal in space. So grid cells exist alongside other cells that are defined by their functional coding properties that are observed as animals navigate through space. There are border cells that are active when the animal is near an environmental boundary, as well as object vector cells that are active when the animal is at a particular distance from an object. And these two cells are thought to represent landmarks or boundaries within the environment. There are also head direction cells that fire at a high rate only when the animal faces one particular direction, such as the northeast direction in this case, and are thought to provide a type of internal neural compass. And there are speed cells that change their firing rate here on the y-axis with the running speed of the animal here on the x-axis that are thought to provide a type of neural speedometer. Together as a population of neurons, these cells are thought to support navigational behaviors, as well as provide some input to place cells in the hippocampus, which are neurons that fire only when the animal's in one particular place and are thought to provide the basis of episodic or autobiographical memory. But despite the really fascinating physiology of these cells, many questions remain. And what I really want to talk about today is how these neural maps might flexibly change to support memory or navigation. So historically, there's been a proposed association between how entorhinal cortex versus the hippocampus might support memory and navigation. And the proposal was that in entorhinal cortex, you have codes for spatial position, orientation, and the running speed of the animal that are independent of what are called non-metric cues. These are cues, for example, like the behavioral state of the animal or the presence or absence of a reward. While in the hippocampus, you have place cells, and these place cells are sensitive to metric cues like the geometry of the environment, as well as non-metric cues, such as the presence or absence of a reward or the behavioral state of the animal. This dissociation would allow the brain then, for example, to encode unique representations or unique episodes of distinct events that all occurred in the same environment. So this is all in the same concert hall. And this would be done by the hippocampus, while the entorhinal cortex would provide you with this universal and sort of stable metric for navigating around this environment, no matter what particular event was actually occurring at the time. If, however, you went to a completely new concert hall, the hippocampus would encode that unique episode, and the entorhinal cortex would also change in order to indicate that you were in a new spatial context. However, subsequent to the discovery of entorhinal grid cells and these other cells I've just described to you, work began to show that entorhinal codes are perhaps more sensitive than initially proposed. And this was sort of the first example of that, in which the researchers used the following paradigm. So they had a square arena. This is a 1 by 1 meter open box or open arena. And after the animal became very, very familiar with this environment, so it had experienced this environment multiple times, they would simply place a wall. And so now the animal can only explore this compressed environment. And what was found in rats, and subsequently we showed in mice, is that grid cells would actually rescale along the axis that was modified. So you would see a compression in the grid cell firing pattern along this axis that had been compressed. Grid cells are not the only cells that would rescale in response to changes in environmental geometry. Speed cells also change their firing rate by running speed relationships. So here I'm just showing that for an example set of cells where you can see that the slope of the firing rate versus running speed relationship actually changes in the modified environment. And many head direction cells also would shift their orientation to align with the modified axis. So here's an example of two head direction cells that did that, where you have their tuning curve in the baseline environment in dark purple, and then their tuning curve in the modified environment in light blue. And you can see that both of these tuning curves have shifted so that the peak firing rate is aligned with the modified axis. But it's not just environmental geometry, which is a metric cue. So that was perhaps a bit less surprising. But it's also, entorhinal codes are also sensitive to changes in an animal's behavior. We originally looked at this by simply considering what entorhinal codes were doing, or what the neural code was doing in entorhinal cortex when an animal was running fast, so say above 10 centimeters per second, versus when the animal was running slow, say below 10 centimeters per second. And what we found, which we were rather surprised by, was that the number of entorhinal neurons that actually encode navigational variables increase with running speed. And I'm just showing you one example of that here, where the number of neurons that encode either the position or the orientation of the animal when it's running slow is shown here in black, and when it's running fast is shown here in purple. And you can see there's actually an increase in the number of neurons that significantly encode those variables only when the animal's running fast. It turns out that this increase in coding reflected an increase in the sharpness of tuning. So I'm showing an example of that for a position encoding cell here. You can see that when the animal's running fast, the position encoding cell actually becomes much more coherent in its representation. And in some ways, you can see there's almost this increase in the signal to noise ratio when the animal's running fast versus slow. So this told us that entorhinal neurons are actually capable of dynamically varying their tuning properties with an animal's behavior. Based on this, we wanted to ask whether or not entorhinal maps might also adaptively alter under other kinds of task conditions. And one of the motivations for this study was that up until this point, many, not all, but many entorhinal studies had utilized just a single behavioral paradigm. Rodents were placed into an open arena, and then they foraged for randomly scattered food over the course of 10 to 30 minutes. However, we know that many ethologically relevant navigational strategies often employ behaviors such as goal-directed navigation to remembered reward locations. For example, animals are often engaged in behavior that isn't really well classified as random foraging, but rather they're navigating back to a remembered food source, or they're navigating back to a remembered food or safe location. And so we wanted to specifically ask, are entorhinal neurocodes potentially influenced by tasks that integrate a remembered reward location? So to look at this, we designed the following task when we had a black box and a white box. In the black box, the animal would perform this oft-used random foraging paradigm, such as randomly foraging for scattered food rewards. In the white box, the animal would switch between random foraging, and then when it heard an auditory tone, it had to go to an unmarked remembered reward location in order to receive a larger food reward, an entire Cheerio versus just Cheerio dust. And I should note that these two environments are actually in the same spatial position. So we would lift the black box up and place the white box down. So all the sort of external cues are identical between these two boxes, and the geometry of the boxes are identical. This is just an example of an animal's behavior in the two environments. Here you can see its trajectory in gray during the free foraging, and it covers the environment really nicely. And here in the spatial task condition, it's covering the environment quite nicely when it's randomly foraging. And when it hears the tone, it's taking these relatively direct paths back to the unmarked remembered reward location as indicated by these different colors. Now, importantly, we're actually going to compare all of our data in experimental animals, those that perform the spatial task, to a set of control animals that experience the black box, the white box, as well as the tone, but don't perform any sort of task. They just randomly forage in both environments. And we're then going to look at grid cells, border cells, head direction cells, and these cells that are called non-grid spatial cells. These are neurons that have some sort of spatial representation, but these representations don't have the same underlying geometry as grid or border cells. So they're a little bit more like place cells. Okay, so what happens to grid cells? This is an example of 3 different grid cells that were recorded in environment 1 and also in environment 2. And the first feature of these cells that we looked at was their spacing. And this is just the distance between the firing nodes. And what we found was that in our experimental animals, those that had to perform the task, we actually found a significant decrease in spacing between environment 1 and environment 2. And that's just shown here, where the spacing ratio between the two environments is on the x-axis, and then the number of cells is on the y-axis. And you can see the experimental animals have this small but significant shift to the left. The next feature that we looked at was orientation. So this is the tilt of the grid pattern. And here we found an even more striking effect. In the control animals, we found no change in grid orientation. Because the pattern is 60 degrees or has 60-degree symmetry, a point at 60 is essentially a point at 0. And you can see the vast majority of these points cluster at 0 or 60. Whereas in these experimental animals, there's actually quite a bit of change in the orientation of the grid pattern. So what this told us was that despite the fact that these two environments share a geometric environmental structure, grid cells show signatures of contextual remapping in the presence of the task. In other words, grid cells are changing in the same way they normally would only change if the animal went into 2 very distinct contexts, say 2 different rooms. We also found that in our experimental animals, head direction cells would change their orientation. I'm just showing 4 examples of that here, where the tuning curve of the head direction cell in environment 1 is in blue, and the tuning curve in environment 2 is in red. And you can see that many cells here shifted their orientation. We didn't see this kind of shift in orientation in our control animals. Border cells also changed. This middle example here is a particularly striking example where a border cell went from preferring the east wall to the north wall. Other border cells just changed their firing rate, but remained on the same wall. And you can see that here, in which a number of border cells didn't change their orientation, but we actually had a surprising number of border cells that rotated by 90 or even 180 degrees. Again, we didn't see these changes in the control animals. And then finally, these non-grid spatial cells showed different features of remapping, and I'll talk a little bit more specifically about that on the next slide. But you can see that here these cells are changing the spatial position in which they're active, or their underlying spatial pattern is changing between the two environments. Okay, so we next wanted to ask whether or not entorhinal maps restructure in a way that can incorporate the remembered reward location. So first we considered grid cells. And what I'm showing you here is the average firing rate of all of our grid cells here on the y-axis, plotted relative to the animal's position, plotted as a function of the animal's position relative to the reward location here on the x-axis. So here at 0, this is closest to the reward location, and at 100, this is furthest from the reward location. And what you can see is that in environment 2, there's actually this increase in the firing rate of grid cells compared to environment 1. And this gray curve is just the difference between these red and blue curves. We performed a variety of analyses that indicated that the reason we saw this increase in the firing rate was because in environment 2, whatever grid nodes happened to be closest to the unmarked remembered reward zone were increasing their firing rate, while the other grid nodes were either remaining the same or decreasing their firing rate. This effect was even more striking in our non-grid spatial cells. So here's the same plot for these non-grid spatial cells, and you can see that when the animal's near the reward zone, there's a significant increase in the firing rate compared to when the animal's further from the reward zone. And again, this is the difference between the two curves. This was due to sort of heterogeneous or mixed changes in these non-grid spatial cells. And I'm just showing an example of two of the most kind of striking changes that we saw. The first is that we found cells that had a strong a very spatially stable and coherent firing field in environment 1. And this firing field would actually move closer or overlap with the reward zone in environment 2. There was a second class of cells that had a relatively distributed or disorganized representation in environment 1, but in environment 2, they would form a much more coherent and stable representation over or near the reward zone. And these phenomena together, as well as a few other phenomena, increased the firing activity of these cells, specifically near the reward zone in environment 2. So finally, we asked how might task-associated changes in entorhinal codes impact navigation? And to do this, we estimated the animal's position from simultaneously recorded neurons. We had a few sessions in which we recorded several tens of neurons simultaneously, which allowed us to perform this analysis. We then trained a Bayesian decoder on position and speed matched data between environment 1 and environment 2. And this is an example of what we found from just an individual session. So here, this is the decoding performance on the y-axis. A lower number indicates higher decoding performance, so better accuracy compared to a larger number. And again, on the x-axis, this is the distance of the animal from the reward zone. So 0 is at the reward zone, and here's furthest from the reward zone that the animal can be. And again, you can see there's this very large difference where we can decode the position of the animal much better near the reward zone compared to far from the reward zone and compared to the same location in environment 1. This is across all of our sessions. So again, you see the same general trend where you have better decoding performance near the reward zone in environment 2 compared to environment 1. And again, this is just the difference between those two curves. So that tells us that these localized changes in both firing rate and the structure of the spatial codes support improved spatial decoding at the remembered reward location. Okay, so I've just shown you that entorhinal codes are in fact quite sensitive to changes in environmental geometry. They're sensitive to changes in the animal's running speed, and they're sensitive to changes in task demands. However, all of this previous work, as well as other previous work looking at similar types of phenomena in entorhinal cortex, have largely focused on dynamic coding properties of individual neurons, so comparing an individual grid cell across conditions. But entorhinal coding properties, particularly in the context of computational modeling, are often described as emerging from recurrently connected networks of cells in which each neuron's activity pattern actually influences that of neighboring neurons. So we wanted to ask, to what degree do dynamic coding properties actually manifest at the network level? And some of the challenges at looking at this with some of our previous experiments have been variable behavior. The animal rarely ever takes the same trajectory twice. These are temporally short sessions. They're usually 20 to 30 minutes. And there's a small number of cells that are recorded simultaneously. Our record with tetrarods was around 60 cells simultaneously, but that really getting close to that number is really quite rare, particularly in mice. So to deal with many of these challenges, we turn to using virtual reality. Virtual reality allows us to force the animal to essentially take an identical trajectory tens to many hundreds of times. And in addition, we can then use neuropixel probes, which are a silicon recording probe that allows us to record from several hundred neurons simultaneously. This is just an illustration of the type of data that we can get from this paper on bioRxiv here, in which you can see where the probe was actually located. It covered the entire dorsal ventral extent of entorhinal cortex, and we can often record over 200 neurons simultaneously, and even in some cases, we can record over 300 entorhinal neurons simultaneously. So we wanted to use this approach to then ask, do entorhinal maps show flexibility at a population level in an invariant spatial and behavioral context? And so we designed the following task in which animals would navigate one of two virtual realities, either one that had several visual cues or just two visual cues, and perform a random foraging task. And the random foraging task was essentially that on each trial, there would be a visual cue that would indicate a location where the animal could lick for reward. And this position would change on each individual trial. And this paradigm thus allowed us to get really nice consistent speed coverage across all spatial positions. So this is the data from 17 mice across 45 sessions. You can see that there's this really nice consistent speed profile across all of the different positions. In this task, we find that many cells have really nice, stable spatial firing patterns. This is an example of 3 individual neurons that we recorded from. The position of the animal is on the x-axis, and then trials are stacked on top of each other along the y-axis. And this is the overall firing rate map that's constructed from these raster plots here. And you can see that for each of these 3 cells, there's really nice spatial periodicity as well as spatial stability in how they're representing this virtual track. However, you might also notice that this is only trials 300 to 260, so 40 trials. When we actually look across many hundreds of trials, what I think you can see even by eye is that the patterns aren't actually stable in the sense that this representation really doesn't persist through all of the trials. And instead, it looks like entorhinal cortex is sort of switching between multiple different stable maps for this virtual reality. And one way we can sort of better characterize that is by constructing these trial-by-trial similarity matrices. So for these trial-by-trial similarity matrices, we just take the activity of all neurons recorded on a given trial, and we correlate that with the activity of all neurons on another trial. Trials then that have highly similar population level activity are color-coded in darker colors, and trials that have low correlation are color-coded in lighter colors. And what you can see is that in all 3 of these are different examples of sessions that were recorded. In all 3 sessions, you can see this sort of checkerboard-like representation. And what that means is that entorhinal cortex is actually switching between multiple stable maps for this invariant spatial context. And so this really tells us then that entorhinal neurons can dynamically vary their tuning properties despite the fact that the animal is in the same sensory context and performing the same behavioral task. So we next wanted to have some sort of way to quantify the number of maps that were actually observed. And so to do this, we grouped trials with similar network-wide activity by applying a k-means clustering to each session. And this is the output of just an example set of 3 neurons, where what we've identified as map 1 is shown in black, and what we've identified as map 2 is shown in a different color for each unit. And you can see here that in the rate maps, that if we just look at the entire set of trials, some cells we might actually identify as not really having that stable of spatial tuning, but in fact, the cell is highly stable. It's just flipping between 2 different map representations. In other cases, there's just a change in firing rate, while in this case, for example, the cell is actually shifting which spatial position it's most active in. These k-means labels often visibly match the checkerboard pattern. So here's the trial-by-trial similarity matrix, and you can see that this checkerboard pattern really nicely aligns with what the k-means is clustering as map 1 versus map 2. We had about 18 out of 32 sessions that were really well fit by a 2-cluster k-means model. So this means that it's identifying 2 maps, and we call these 2-map sessions. And I'm going to focus on these 2-map sessions for the last few slides of my talk. This identification of multiple maps was a really nice way then to look at how coherent these map switches are across the entire dorsal ventral axis of medial entorhinal cortex. So what I'm showing you here is just a really nice example session where we have this distance score where a negative number indicates that the population is more similar to map 1, and a positive score indicates the population is more similar to map 2. And then here, I'm showing you the activity of a little over 200 neurons that were simultaneously recorded that are ordered from dorsal to ventral entorhinal cortex. And what you can see is that there's these bands of black and then white activity, and that indicates that almost the entire dorsal ventral axis, cells along this entire axis, are actually participating in these map switches. So if remapping occurs without a change to the environment or task, what factors are actually contributing to remapping? Well, we were inspired by our previous work showing that running speed can contribute to the sharpness of entorhinal tuning. And we looked at whether or not running speed might also correlate with these remapping events. So here's an example again of an individual trial, an individual session. The trial by trial similarity matrix is here on top. And then the running speed profile of the animal is plotted here on the y-axis relative to the trial number on the x-axis. And every time there's a remap, I've put a red dotted line here. And you can see that actually, even by eye, that oftentimes there's a dip in the running speed that seems to correspond to a change in, or that seems to correspond with a remapping event. And in fact, this isn't just something that you can kind of see by eye. If we quantify this, it is the case that the running speed of the animal is slower in the trial just before a remap compared to the running speed of the animal during a stable block of trials. This was the case across multiple mice and multiple different sessions. Here you can see the speed of the animal just prior to a remap versus the speed of the animal during a stable block of trials. So this really told us that population-wide remaps are preceded by slower running speeds. So why might that be the case? Well, to consider that, how running speed might actually drive remapping, we collaborated with Alex Williams and Scott Linderman to characterize the geometry of position codes. So attractor models, which are a class of computational model that are often used to describe the coding properties of entorhinal cortex, would predict that the trajectory of network activity on this linear track should trace out a 1D ring manifold. So here's just an illustration of that, where you have the track up here, and then each position on the track is color-coded. And if we look at the activity of the neurons along the position, along the sort of track position, you should see some kind of ring manifold emerge if attractor dynamics are indeed contributing to the emergence of entorhinal coding features. So we then did this low dimensional linear embedding of each k-means cluster centroid, which revealed ring manifolds. And if we used PCA to visualize the 2 manifolds in the 2 map sessions, what we found was that in both the Q-poor and the Q-rich, so these are 2 different tracks, we found the presence of 2 ring manifolds that were very nicely aligned with each other. And what that means is that these remapping events essentially represent a jump from one of these manifolds to the other. And this is just a nice little video showing that. This is actually simulated data. But in essence, what's happening as the animal navigates through this virtual space is that the activity is jumping from one manifold to the other. And this is what is occurring when we actually observe a remapping event. So neural activity is transitioning then between these 2 geometrically aligned ring attractors. This analysis then allowed us to reveal some interesting features of the data. The first is that these 2 ring attractors are highly aligned with each other. If we stretch or sort of rotate them to see how much we have to do that in order to perfectly align them, the value is very, very small. They tend to be highly aligned. And that means that any downstream region or any decoder that's insensitive to the translation along this dimension here will still be able to decode the position of the animal despite this remapping. And we actually found that in the experimental data. I just don't have time to describe it today. In addition, we found that the variance in the manifolds were preferentially oriented in the dimensions separating the manifolds. And what this means at a high level is that anything that increases network variability, in theory, could predispose the network to remap. So with this in mind, we went back to ask whether or not slower running speeds might correlate with an increase in variability. And so to do this, we calculated how close neural activity was to the midpoint between the manifolds. And I'm showing you the data for just two individual sessions from two different mice. So here, a lower value means that the activity is closer to the midpoint between manifolds, and a higher value means that it's further away. And you can see that at lower running speeds, the activity is closer to the midpoint compared to higher running speeds. And this is the case for both of these animals and sessions. Here we've just split up our data into 5-centimeter position bins and looked at the fastest running epochs versus the slowest running epochs. And you can again see that these two peaks of activity are closer together when the animal's running slow compared to when the animal's running fast. So what this means is that if you have these two maps, when the animal's running slow, the sort of energy barrier, or how easily it is for the maps to switch between, or the neural representation to switch between these two maps, is much easier compared to when the animal is running fast. So let me just summarize what I showed you today. Entorhinal maps are in fact quite sensitive to environmental geometry and running speed. Remembered reward locations can serve as a contextual cue capable of evoking different entorhinal maps. And entorhinal maps at the population level can restructure based on features of behavioral state. And I think it's important to remember that many of the neural substrates that I showed you today, head direction cells, grid cells, border cells, have been found in a wide variety of different species, ranging from fish and flies to bats, rodents, non-human and human primates. And we think that some of these coding features that I described today can allow entorhinal cortex and these representations to integrate contextual features that then allow animals to support a wide range of different navigational strategies. And finally, most importantly, I want to thank the amazing group of people that performed all the work that I talked about today. Rob Munn, who's now a PI at the University of Ireland in Galway, did a lot of the work looking at environmental geometry and how that impacts representations in entorhinal cortex, with Caitlin Mallory, who was a graduate student in the lab and is now a postdoc at Berkeley, as well as Kia Hardcastle, who was a graduate student in the lab and is now a postdoc at Harvard. Kia also performed work looking at how running speed influences entorhinal maps of space with Neero Maheswaranathan, who is now at Google, and then Surya Ganguly, who's another PI at Stanford. And then finally, the work on how remembered reward locations influence entorhinal cortex was performed by Will Butler, who's now a data scientist at C.H. Robinson, as well as Kia Hardcastle. And the work looking at multiple maps in entorhinal cortex was performed by Isabel Lowe, who's a graduate student in the lab, with Malcolm Campbell, who's now a postdoc at Harvard, and Alex Williams, who's a postdoc with Scott Linderman, who's also at Stanford. And I just want to acknowledge my funding sources and thank everyone for their attention.
Fantastic.
And now I'll go back to the dark Zoom.
I'm sorry that we only have one applause. That was really great stuff.
Oh, no problem.
So just again, to everybody who's attending, we've got a few minutes left for questions. If you'd like to ask any, please fill them out in the Q&A section. And I guess the first question that I have is, as someone who studies behavior primarily, you showed this really fascinating result that the ability to decode location, I believe, increased in the proximity of the reward location. Does that mean, and have you been able to find any evidence that the rats actually have improved spatial precision in that location?
Yeah, that's a great question. We had hoped to look at that in that study. Our behavioral task was a little bit too easy in the sense that we had very, very high performance accuracy And we weren't able to directly identify any kind of behavioral impact of the decoding improvement. We have a new task and some new work we're doing where we think we might be able to dissociate that by making the task a little bit harder and a little more complex. But I think it'd be really fascinating to look at that and see. There's also variability across the rats in terms of how much decoding improvement we saw. So I think it could be really interesting to look at whether or not that animal variability might correlate with some kind of variability in their performance.
Do you see any other sort of passively observed differences? Like, for example, does their running speed change when they get in the vicinity of that region?
Yeah, there are some behavioral differences between the two tasks. So the animals tend to spend slightly more time near the reward zone in environment 2, and they do also tend to run much faster on their trajectories towards the reward zone when they hear the auditory tone. We did some, controls to make sure that wasn't, impacting the coding changes that we saw, but there are some differences in the actual behavior of the animals between the two environments.
What do you think of as the sort of role of that remapping towards the reward?
Yeah, I guess I imagine it as, if I say I have an environment that I'm very familiar with, I have a particular representation of that environment, a particular entorhinal map of that environment. But if one day I find, a really amazing reward that has a high value to me, or, allows me to survive, like a great food source, it might actually benefit me to create a new map of the same environment that has, some resources allocated to representing that reward location. And in some animals, not all, but in some animals, we actually found that there was a sort of detriment in the decoding when the animal was further away from the reward. That wasn't always the case, but it seems like in some cases, there can be sort of a reallocation of resources to representing the space near the reward, sort of at the detriment to positions that are further from the reward.
Right. Well, the questions are starting to pour in, including one from Mike Hasselmo. Hi, Mike. First one by Hugo Spears. Do you think arousal level separate to speed might influence the switching between maps?
Yeah, that's a great question. I suspect arousal probably plays a role. We've been starting to look at pupil dilation to see if maybe we can get some sense of how correlated arousal and running speed are. We might also try some sort of manipulation where we force the animal's running speed to be a certain speed. There was a paper recently that showed that can actually dissociate arousal from locomotion. So I don't know the answer to that question, but I think it's definitely something that we would like to address and better understand.
Jason Moore has a question. If you were to manually restrict the speed by holding the VR ball, would you predict that would induce a remapping? For example, I'm getting it if the speed changes the causal event or vice versa.
Yeah, so we definitely see reorganization in entorhinal maps when we break the ball and just play the visual scene back to the animal. We haven't done like transient breaks, but one of the bioRxiv papers we have online talks about, or I think it may have ended up in the supplemental material, but we actually have some experiments where we break the ball and then we play back the visual scene. And we actually see that entorhinal maps become highly unstable. They're not spatially stable at all anymore. So they do need some kind of pairing between locomotion and landmarks and or optic flow in order to generate stable maps of space, as well as I think probably multiple maps of space.
Right, okay. So Mike Hasselmo asks, did you see any systematic timing relative to theta rhythm phase or change in the theta amplitude at the time of the switch between maps in the study by Lowe et al.?
Yeah, that's a great question. We haven't looked at that yet, but that's something that we are starting to look at now, both seeing if we can detect, how well we can detect the activity of the neurons relative to theta and whether or not there might be any kind of sequences or changes in sequences or changes in phase precession. But yeah, great question. We haven't done that yet.
Question by someone I don't recognize, DFM. Did you see any indication that switching of the maps might be related to some perceptual ambiguity in the environment?
Yeah, that's a great question. We initially were a little concerned about that because we were primarily using this Q-poor environment, but there had been reports from Malcolm's previous work using tetrodes, which had a much more Q-rich environment, as well as other work in the lab using Q-rich environments. And then we also saw that in the Lowe et al. Paper, where we could use a much richer environment in terms of it having many more cues, visual cues, and we still saw the same amount of remapping or switching. So, so I don't think it's just because there's not access to a large number of cues. My suspicion is that, you know, if you were to give access to many, many multi-sensory cues, so when the animal, for example, is navigating in an open field and has access to, say it has access to many multi-sensory cues, olfactory cues, locomotor cues, you know, vestibular cues, it becomes harder for behavioral state to influence the representations in the same way that it is in the VR. I don't know that for sure, but I think the sort of more landmark input you have and the stronger the landmark input is, the harder it's going to be for, say, running speed or behavioral state to kind of lower that barrier for map switches to occur. So I think we do have a paradigm in which we probably see more switches than you might see if an animal was out navigating in a highly cue-rich naturalistic environment.
Yeah, that makes a lot of sense. Just sort of a question of my own here. Did you, if you observe, like, for example, over a time period, did you observe that switches became less frequent? Did animals sort of drift towards anchoring on one?
Yeah. Yeah, that's a great question, Brad. We definitely looked across the entire session because animals become somewhat sated towards the end of the recording session. We saw some hints that in some sessions, it seemed like there were more map switches, like as the animal became a little bit less engaged in the random foraging task, but we didn't see anything that was sort of highly consistent. And then we're also, because we're placing the probes every day, it's a little bit harder to say how things might track across days. We definitely saw a lot of variability. So, you know, there was a lot of variability between animals and between sessions in terms of the number of maps that would emerge, the times that we would, you know, the number of switches that would occur, whether or not both maps were spatially stable or only one was spatially stable. So there's definitely some heterogeneity to this map switching that we don't yet fully understand.
And you think it's the actual dynamics within the entorhinal cortex itself that define it, or do you think there's some other area that's sort of the causal?
You know, I don't know. I think much of the remapping dynamics are perhaps due to, you know, attractor-like dynamics in entorhinal cortex, but I also think the hippocampus likely plays a role. I mean, we know that the hippocampus provides a huge source of input to the entorhinal cortex and has a lot of influence on coding in entorhinal cortex. So, you know, it could be interesting in future studies to do simultaneous, you know, hippocampal and entorhinal recordings to see, you know, if map switches are coherent or if one precedes the other in terms of hippocampal versus entorhinal representations.
Go back into the Q&A here. Ricardo Beltramo asks if you saw switching maps in visual areas that project to the cortex or the entorhinal cortex.
Yeah, that's a great question. We looked at a few regions outside of entorhinal cortex. So like the neuropixel probe kind of goes through a few regions just above entorhinal cortex, and we didn't see map switching there. In the Campbell and Attinger bioarchive paper, we looked specifically at visual cortex, and we definitely see much lower rates of spontaneous remapping in V1 compared to entorhinal cortex. V1 is much more stable in terms of kind of having stable representations across these longer sets of trials.
All right, another question then is, are animals doing anything particularly interesting during the dips in running speed at the remapping events?
I don't know what I would define as particularly interesting. We haven't really looked at that so much, but you know, sometimes they will stop to kind of like quasi-groom. I don't think we've quantified at all what they're really doing specifically during the slower running speeds, and or if that behavior would be of interest. But it's a good thought and good suggestion. We do have video for many of these recording sessions.
Yeah, there's a sort of follow-up to that is like, are there any cases where you sort of expect to see a remapping, but like they might sort of fall back into the same map?
Oh, definitely. It's not like every time they slow down, you always see a remapping. So, you know, we kind of envision it as the slower running speed predisposes the system to remap, but it doesn't necessarily, you know, predispose it to the degree that it's always, you know, as soon as the animal slows down, there's a remap. There's definitely a strong effect of running speed, but it's not like every single trial where the animal's running slower, you always see a remap. There are occasions where the map stays in the same map.
And sort of, you know, thinking about what might happen over time again, are there cases where, for example, it'll be switching back and forth between two maps and then a third map will emerge?
Oh, yeah. Yeah, there were also a few 3-map sessions, and I believe there's even one 4-map session. So, and that map, we didn't see anything, you know, we didn't see anything clear where it was like always map 1, 2, 3, 1, 2, 3. You know, it seemed pretty heterogeneous as to like what map would actually emerge when.
Mm-hmm. Do you think that this might have anything to do with, I don't know, sort of maybe switching between like egocentric and allocentric reference frames? Like sometimes, you know, if we're like navigating out in the world, sometimes we sort of think about where we are in one sort of frame or the other.
Yeah, I hadn't really thought about that, to be honest, but that's a good suggestion. It's possible that it could represent some sort of switch in reference frames. I'd have to think a little bit more about exactly what to expect if it's switching to an egocentric reference frame, but that's a really interesting suggestion in terms of how to think about this data.
Well, it's fascinating. Yeah, obviously we could go all night giving you 20 questions about how the system works. It's so fascinating. I really love the whole navigational system. It's such a great paradigm. And, you know, now with these new techniques, you know, it's just amazing the questions that you can ask and answer.
Thanks. Thanks very much.
So is there sort of like big dream direction that you're heading next in terms of like a different area integrating between like entorhinal and something else?
Yeah, I mean, we've started to look at other brain regions. You know, we're looking a bit at retrosplenial and V1 and trying to understand, you know, what might be unique about entorhinal cortex versus, you know, what might be more universal across these brain regions that support navigation.
One more quick question popped in, and this will be our last one, and we might get cut off at any second here as the next webinar spins up, but let me go ahead and ask it quick. Can the switch of maps correspond to a switch of phases, as in 1D, the same grid cell at different phases would be represented differently?
Oh, that's a great question. We didn't do the analysis where we sort of fit like one-dimensional representations to a two-dimensional grid, but it's possible that they could be different slices. I should emphasize, though, that we see this in what we think are not just grid cells. They're you know, we're not specifically identifying grid cells. We're just looking at spatially stable cells. So this is also probably capturing some cells like those non-grid spatial cells or border cells or other types of cells. So I think it's probably not just a like phase shift in the grid pattern, but that's something that we could probably look into a little bit more.
All right. Well, okay. Thanks again, Lisa.
Thanks, Brad.
Yes.
Yeah, and thanks to everybody who's out there in the virtual world.
All right. Bye, everybody. Enjoy the rest of the conference.
Thank you. Do I just log off now, Brad?
Yeah, I'll just end it and then we're done.
Okay.
All right.
Hey, great.